our honorable chairperson for the session.
Good morning. I welcome you all for today's morning session, Medicinal Botany and Phytochemistry. It's my pleasure to introduce our chairperson for the session, Dr. Arnab Singh. Professor Arnab is working in the Department of Botany and as a coordinator of DBT Bioinformatics facility in North Bengal University, NBU. He is in charge of Bisho Bangla Genome Center, North Bengal University. He has done PhD from Northeastern Hill University, Nehu. Sir has been honored and awarded with different awards like DBT Crest Award, DBT Overseas Award, DBT Fast Track Young Scientist Award. Sir is working as a principal investigator Pancreatic Genome Project sponsored by US Department of Energy. He is honorary chief editor of Sikkim State Biotech Hub, also a member of PhD Defense Board, University of New Hampshire, USA. Professor Arnab is editor for Indian Journal of Microbiology, published by Springer Publication. Along with this, Sir is a guest editor for Indian Journal of Biotechnology, published by CSIR Publication. Sir has completed 15 microbial genome projects and has supervised 28 research scholars. Till date, Sir has published 2 books and 162 papers having total impact factor of 173.85 and total citations 1745. We are very fortunate to have such a knowledgeable person with us. Again, I welcome you, sir. Now I request Dr. Shweta Puri to join on screen and introduce our today's featured speaker, Dr. Sanjeev Ray. Over to you, Shweta.
Thank you so much, Sheetal ma'am. A very good morning to everyone, and I am welcoming Dr. Sanjeev Racer on the very second day of our This Knowledge Mahakumbha International Conferences. The technical session of today is on medicinal botany, and you will be amazed to know that Sir is the professor in zoology department, University of Burdwan and West Bengal, yet Sir has the area of research specialization on medicinal plants like antioxidants, phytochemical extraction from the traditionally used medicinal plants. Sir has pursued his MSc and PhD from Northeastern University, and Sir has 19 years of teaching experience and specialization in molecular biogenetics and cytogenetics. Sir has full of bucket of awards like DBT Crest Award, DBT Overseas Award, Young Scientist Award. Sir also has completed several research projects related with UGC major research project, SAP, DST FIST, and Sir also chaired the session of PhD Defense Board of University of New Hampshire, USA, and Sir also was the principal investigation in Frankia Genome Project. Sir also supervised around 28 students, PhD students, and published around 162 research papers and completed 15 microbial genome project. And we all know the importance of medicinal biobotany these days, that everyone knows medicinal plants has therapeutic values due to their active principles. And in this time, we all now much aware with all CADA and all application in this COVID area. So I'm welcoming Sanjeev Racer for this very first technical session of today. Welcome you, sir.
Thank you for your introduction, but little correction maybe needs some data is not up to the correct. I will give my self introduction.
Sir, okay, sir.
Yes, Sanjeev, we will correct it.
Sanjeev sir, I'm welcoming you to chair this technical session.
It is Arnab sir, not Sanjeev sir. Professor Arnab Sen, please chair the session and start the.
Okay. I think yes, there is some goof up, but no problem. First of all, I must thank the introducer. I think the Shital, for introducing me in such a nice way. Though I don't deserve this, but anyway. And also, I would like to thank Gopa and other organizers for inviting me for in such a nice, podium to chair this session. And Sanjeev is actually like a younger brother to me, because when I've just joined North Bengal University as a lecturer, then Sanjeev came to me and asked about the Northeastern Hill University and also the laboratory of Professor Anupam Chatterjee, where he got a chance to do PhD research. And I told Sanjeev that it's a very good laboratory, one of the best laboratory of NEHU. I said, you can go there. And as per my advice, probably, Sanjeev has joined Northeastern University, Shillong, and then he has done some excellent work over there. Got his PhD over there, and then he has joined, I think, the Bardon University, if I am not wrong, Sanjeev, there, and he is illustrious professor over there now. And rest, Sanjeev himself will introduce himself about his.
I think that's better. Dr. Sanjeev, I think it's better you introduce yourself.
Yes. Sanjeev will introduce yourself. Self-introduction, Sanjeev. Don't mind. Go ahead.
Thank you. First of all, I would like to congratulate this Guru Nanak Khalsa College for organizing this wonderful seminar, international seminar. So I would like to convey my sincere thanks to organizers, all the college team who are doing this great job, and all the members present today these events or this session. So nothing to say much. I have done my master degree from North Bengal University, PhD from NEHU, and presently working in Bardhaman University, Department of Zoology. This is little bit my introduction. So I'll start my presentation directly. Whatever presently I am doing this field, I know this is very challenging task to say something in presence of this major botanist community, as my background is from zoology. So I'll try my level best. So I think you will all cooperate to me to complete my session. So I will take, I think, get maximum 10 to 15 minutes. So accordingly, I will finish my talk.
Yes, there are a lot of presentations today.
Yes, so I'll take only 15 minutes.
Yeah.
Gopa, how much time Sanjeev is allotted?
15 minutes to 20 minutes.
Oh, 20 minutes. Okay, Sanjeev. You
Go ahead,
please
Sanjeev.
conclude within 20 minutes, okay?
You have sent me the presentation?
So I'm sharing from my side.
You share yourself.
Okay, right.
I think it's
Yes.
visible.
Yes, your skin are visible. Go ahead,
Okay.
Sanjeev.
Hi, good morning.
Good morning.
So today's my lecture title as Deciphering Colchicine-like Antimitotic Effects of Claritin Diterpenoids. So first of all, I will explain a little bit about these three important terms like Claritin Diterpenoids, antimitotic effects, and about colchicine. So antimitotic effects, mainly we know that this substance or compounds or components which interacts mainly with microtubules, which ultimately results in stopping of mitosis or cell division. They are mainly used as anti-cancer treatment as well as some other anti-inflammatory disease like gout or other things. As we know that it mainly disrupts microtubule formations, or in some cases, it may be stabilized microtubules. As for examples, we know that Taxol is now presently used as anti-cancer drugs. Colchicine is renowned substance for long time it is used. So in our work, we are trying to find some alternatives of well-established colchicine substance. So whatever we have started our work, we have found out some carotenoid diterpenoids, which are very much similar type of work or functions as anti-mitotic substance is working. So my work, we are trying to establish that alternatives of colchicine as sclerotin diterpenoids. Regarding colchicine, these are the few terms are there. You know all that this is the oldest remedy still there is used. It is extracted from Colchicum autumnal plants and mainly used as to treat gout, Baches disease, pericarditis, familiar Mediterranean fever, and in our cytogenetics laboratory mainly we use to arrest metaphase cells at metaphase and for karyotyping analysis, or in plant breedings we know that they use for synthesis of polyploidy. So from this point of view, as we are trying to establish antimitotic effects, so we know that the spindle fiber is the main target. So spindle fiber disruption results into maybe two outcomes. One may be apoptosis, which may be related to your tumor or cancer treatment, or it is induced polyploidy in plants, maybe plant breeding. So how to check this one? How to assess? For assess cytological that parameter we generally see, like micronuclei, background chromosome, chromosomal aberration we assess. Most importantly, C metaphase. C metaphase means colchicine-induced metaphase. In this stage, we will see that chromosomes will arrange such a way that disperse in the metaphasic plate. So as metaphase arrest will be there, so we can say anti-proliferative property is there. Now this mode of action of colchicine, or this is well known for everyone. So as we know, this colchicum autumnal from bulb, we can extract colchicine. It targets microtubules, alpha and beta tubules. They bind and depolymerize this one. As a result, consequently, we see like this chromosomal plate is very useful for our karyotyping analysis. In case of plant breeders, they produce polyploid plant species. Or in our drug system, we can see that mainly it is now used in case of gout. Now gout, its main medicine is as used as colchicine, as well as Baches disease. Baches disease is one type of disease. Generally, our blood vessel endothelial inflammation is there. Now these are the some crystals. That crystal is uric acid crystals. Ultimately creates this gout, and if you treat this colchicine, inflammation, everything will be stopped. So these are well established fact. Mode of action like colchicine, how it prevents. Like as any inflammation, your neutrophil macrophage, they need to move, and for movement, endothelial cells, they need to interaction like E-selectin or L-selectin of neutrophil, they need interaction. As well as we know for our anti-inflammation, inflammation related IL-8, IL-1, all are become stopped or negative impact, or we can say colchicine has like these properties. Right, generation of ROS, TNF alpha NOS, all can be stopped with colchicine. So in our research, if we can find some chemicals, alternative chemicals, less toxic from different group, as colchicine is alkaloid, we are trying to, as nitrogen is there, we know a little bit poisonous. So we are trying to, so terpenoids which are without any nitrogen group may be less toxic. So we are trying to establish that colchicine alternative from terpenoid groups of chemicals. So for this purpose of work, this is very familiar plant for everyone. We know Clerodendron viscosum or Infortunatum from Lamiaceae family. So from this plant, initially we started work where initially we have seen that colchicine induced tumor-like structure in case of onion root tips. So in our crude extract also we have seen that same type of root swelling showing. That means morphologically similar. That means there is maybe some chemicals are there in these plants. That one we have to identify. So first observation in 2007, when I was working in St. Edmund's College, so I observed squashing of this crude extract preparation from these onion root tips. So if this slide, you can see that arrangement of chromosome, dispersed chromosome, that is C metaphase type property, as well as you can see here some dead cell, apoptosis cell, and there is polyploidy. So this slide indicates that there must be some chemicals which have your colchicine-like substance. So stop searching this one in cell proliferation. We have established this one metaphase arresting and cell creating properties is there in this plant. So now main other work, cytological work we have done, and we have seen that different type of cytological abnormalities all indicates, and we have compared with colchicine-like phenomenon. So this is normal one, this is colchicine treatment. We can see C metaphase, and our treatment crude drug also seeing C metaphase, as well as we can see polyploid nucleus, micronucleus, all are indicating antimitotic effects. So that's why several slides are there, very nice slides. You can see polyploids, how these polyploid cells are dividing. So onion root tip, it is very easy and to score that different cytological, morphological, nuclear morphological aberrations. So we have scored this one. After scoring, we have published one paper, that is mitotic abnormalities and macronuclear inducing potentials of colchicine and leaf aqueous extract of Colchicum cladodendron viscosum. So from this work, now our question is that whether it can induce apoptosis or not. So in Dalton lymphoma cell, we have crude extract, we have checked through fluorescence microscope that dead cells indicate apoptotic cells. So it is indicating, showing that it has good apoptosis inducing property. So apoptosis and polyploidy both indicates that it has antimitotic effects. So now our question is, what is the active principle? Is this the colchicine or colchicine-like antimitotic substances? Whether it is microtubule stabilizer or destabilizer. These are the main questions, and we are working on these questions. So after rigorous fractionation, skinning, isolation process, from this plant, we could isolate two phytochemicals. One is phytochemical, these are terpenoids, cladodendron diterpenoids. One is cladodine, another is dihydrocladodine. This is cladodine and dihydrocladodine. So today's discussion will focus on this cladodine first, and if time allows, I will discuss with another family of this, another member of this family, that is cladodendron inermi. From there also we could isolate another chemical, Copycrepin. They have as the same skeleton, they are showing same morphological as well as molecular target and same effects. So this is the process through which we have could isolate around 90, this is 95% pure substance. We can crystallize this substance, and it has the polyploid inducing property. And these chemicals in market is marketized only by Sigma. You can see cost is very high cost. 1 mg is around 27,000. Another chemical from this plant, only dihydrocladodine, only difference is there is, you see, this is unsaturated, it is saturated. This bond was saturated with 2 hydrogen. So these two chemicals we could isolate, and we filed patent, and this is only this last year we granted this patent. Another chemical also we isolated same way. This is 3-epicreptin, and also this is granted. Now first, cladodine, I was just focusing cladodine, how it works. If this panel showing that normal cell division phases, prophase, metaphase, anaphase, telophase. But while you see in our treated pure cladodine treatment, we could find out some cells with polyploid nature. This is nothing to say, we can just see the polyploidy, how induced, and multi-nuclei. Incomplete cytokinesis is showing here. As we know that for cell division, cell plate formation, microtubule is very, very important. Microtubule degradation needs to form cell plates. As cell microtubules are not forming, so cell plate is not possible to form as incomplete cytokinesis, we can see. Later on, as our topic was deciphering colchicine-like action, so we have to have the comparing effect between clerodine and colchicine. And from this, this is panel showing your untreated phenotype of our cell division phases. But in different condition, we have compared with colchicine. These are all colchicine effects, and these are all our clerodine effects. Now, here also we can see some polyploid cell with colchicine, this is our drug, clerodine. So here multipolar anaphase, we can see multipolar anaphase. That is all phase-wise, whatever the different types of cytological abnormalities showing by colchicine, we want to establish that whether our chemical showing same or not. Now another question will come that our chemical is more efficient, less toxic or not. So first we establish that clerodine and our colchicine showing similar effects. This paper is directly showing that similar effect, at least cytological basis. All point, if you see, incomplete cytokinesis, here also incomplete cytokinesis. This is called C metaphase, here also C metaphase. Like this, 1-to-1 we have correlated. Now all these are different types of cytological abnormalities which we have scored, and we have published also this paper. DNA degradation genotoxic effect, if you see, this is colchicine induced, and this is our dose dependent induced. So genotoxic effect also showing a little bit similar type. This is the matter of concern. And we have different cell line we have used to arrest cells at metaphase. As we know that antimitotic drug will arrest at metaphase. If metaphase arrest, definitely there will be G2M arrest. So flow cytometric analysis in different cell like HEC, ACT, MCF cell, we have tried to show that whether the tendency is there or not. And everywhere we have seen dose dependent, the tendency of G2M arrest is showing. You can see at 32%, it becomes 41%. Here is 26%, at least 33% is there. Next question comes whether there is apoptosis or not. There also we have seen that inducing apoptosis tendency also is there. Different cell lines we have used in cancer cell like HEC, ACT, everywhere showing that tendency of your increased apoptotic cells. Of course, we need to standardize that time duration. Later on, directly we wanted to target whether it is showing any depolymerization effect or not. That's why we have in HEC cell lines fluorescent microscopic study we have seen. And this panel, first panel, we see the control one. So nicely we can see tubulin proteins, but when we have treated, that tubulin formation is not there. Rounded appearance showing that there is no tubulin formation. The ACT is 116 also showing same pattern. This is confocal microscopic, very nicely showing that your tubulin depolymerization. And directly we have field trial in Raphanas sativas, that is in Bengali we call as mula, that we have also seen that it has polyploid inducing effect. This is the control, and in our treatment showing polyploid plants. So for polyploid morphological, as we know that these are stomatal density or size. This is the morphological parameter generally is poor for polyploidy. That also satisfies our result. We have done flow cytometric analysis. Here also we have seen increased increased number of DNA content in cells. That's also established. Later on, directly we have gone for docking analysis, also showing that colchicine binding site it is interacting. And affinity also almost similar or a little bit better. That means it binds in colchicine binding site. Up to this is okay. And same way in another family, we know Cladrona inarni. From this plant also we have seen initial root extract that showing root soiling pattern. So we expected that we must get something, but after isolation purification, we have seen one chemical which is the 3-epicariotene with just only one acetyl group is extra. Other everything is same. So you can say as it in the same family, the same type of chemicals is there. Only variation is there. Only one acetyl group is there, and also sharing same colchicine-like effect. And this is my parameter, mainly root swelling pattern. You see, this is your crude extract. In crude extract, we have seen root swelling. So we have to purify this chemical, and after that, we have to show that whether root swelling is there. As standardization, all parameters we have standardized, so it is not difficult for us to analyze. And with this new compound 3-epicarioptene, also you can see that parameter. I am just finishing within 1 or 2 minutes. So just you see this crude extract, also you are showing this cell division pattern, multi-nuclei condition, incomplete cytokinesis. You see multi-nucleus. So this picture directly says that these molecules has some role in interfering your cell division or interact with our. And this arrangement is C metaphase or colchicine-induced metaphase. This is crude extract. All type of we have scored, and we have all the data we have compared with crude extract as well as our purified compounds. So this is purified compounds. 3-epicarioptene we have isolated and patented this compound already. Isolation process we have patented, and we have shown that these also showing colchicine-like root swelling effects. And we have compared with colchicine, all different plant models. We have seen that polyploidy induction is there or not. And this story is almost a repetition. So I won't take much time. These are our chemical analysis. 92% we are showing purity. So 92 to 95% purity we could attain this with molecules. In this process also we needed to do like NMR, H1 and C13. In all the chemicals we have done this up to NMR analysis. So if you want to see that actually how the data is coming, I don't think I have the time to explain, but one two data just showing you that showing root growth retardation effects. So this is morphological appearance. So root growth retardation, inhibition, if you see that dose dependent little bit increase is there, dose dependent increase is there. And
Sanjeev, we have 2 minutes to conclude.
Yes. My main part is over. Just now, excess part just showing here. Cell cycle kinetics, like as we have compared with colchicine, in colchicine, say metaphase arrest, we can see 82%. In our case, it is 66%. But when we give the release of these molecules, in our colchicine showing 41%, but our compound showing 54%. That means retention of this molecule inside this cell is showing more. This is indicating here. This is all our data we have analysis. Like mainly your metaphase frequency indicates the dose dependent increase as well as just showing similarity with colchicine. In 3-epicarioptene also, just like clerodine, all different type of anti-mitotic effects related cytological or nuclear or mitotic abnormalities we can score. You see, multiple nuclei, micronuclei, polyploidy. Okay, this is polyploidy, very clear. That means our colchicine, clerodine, both acting same way. Another molecules is there, dihydroclerodine. That data I am not showing here. Also showing similar trend of this. That means 3 compounds has some competition. Now we have to decide which one is the best, which one has the less toxicity, and which can compete with colchicine. For these slides, you see 3-epicarioptene showing nicely dose dependent, and this is colchicine. Colchicine induced C metaphase or drug induced C metaphase showing better results. So as it is better result, as microtubule research, it has the scope to work in antivirus, antifungal, and your inflammation related disease, heart disease, or your cancer disease. There is scope to work with these molecules as it's showing that activity is there. So same way, metaphase arrest we have established in cancer cell line also we have shown the dose-dependent cell death or all toxicity parameters we have tested, MDT test through. They are also showing same microtubule this stopping property. Apoptosis induced these molecules also DNA degradation is there, and colchicine binding pocket also is acting as these molecules. Their skeleton is same, almost one acetyl group difference and working the same way. And in the plant also we have tested polyploidy induction properties there or not. And from our laboratory, within few years, we could publish some papers like material science, cell proliferation, industrial crops, JK, mutagenesis. At this part we have done from NEHU. Karyologia, within 1 year, 10 publication we have done in these journals. Cytologia, 8 theses we have compiled, 2 MPhil theses from these 4-6 plants. And this is acknowledgement for my students. They have the hard work and of financial support we have got from UGC MRP, DST Inspire or CSIR, UGC SAP, DST Pass Feast, all the different scholars, all are JRF students, they work with me. And in the upper panel, my 2 mentors, and I am the connecting link. So thank you for your nice patience. In shortly, I have tried to level best to show that what type of work we are doing. The few molecules we could isolate, purified, and patented. Now our time has come to work molecular targets to explore more widely, so as we can convert it as medicine, just like colchicine, and we can replace colchicine. That will be our great success if we can do. So anyone interested to work with us, we are open-minded to collaborate, so that we can progress and this molecule can be converted into drug in your future. Thank you.
Thank you, Sanjeev. It's an excellent presentation. Sanjeev's work is worth patented, and he has rightly patented it. And the plant that has chosen this Cladribanium is actually I believe it is a wonder plant, because recently we have published quite a few papers on Cladribanium against COVID-19, because we have found that this one particular chemical, that is terazidol, which is found in Cladribanium in army, which is very effective against this COVID pandemic, COVID spike protein and other proteins. So this plant has become really become a wonder plant these days. Now it is open for discussion. Only a few questions I can take because we are really running out of time, and maybe 1 or 2 questions from the audience. Yes, Hiranjit.
Yeah,
Hiranjit.
Professor Sanjeev.
Yes, sir.
I would just like to say one of my students is working on a chemical, phytochemical, that is biochemical synthesis of camptothecin. And this camptothecin is actually anti-cancerous because it shows topoisomerase activity.
Yes, yes.
So I think if we can just collaborate, because if the chemical is synthesized here, I can collaborate with you and Sir Arnab to have
no problem.
Very nice. We can work together, no problem. Very good.
Cancer cell line, if we can.
Yes, cancer cell line. Shoji, I think, do you have cancer cell line?
No, some part work I have done from collaborating from other laboratories. Still, I'm trying to establish. I think
Anshuman can help you. Anshuman has some cancer cell line.
Yes.
So that you can go there. Anshuman, do you have anything to say?
Nothing to say. If you give me chemical, I will test it.
Okay, we have just started the thin experiments. So moment I have sufficient this camptothecin production, I'll definitely get in touch with you all. Thank you.
No problem. Please, please have a meeting together and
we can have online meeting. There is no problem.
Yes.
Okay, thank you so much.
Other than NEHU group, there is any other questions from the other audience? Because I know that this is very heavily interested with NEHU. But if not, then thank you very much, Sanjeev, for your excellent presentation once again. And congratulations for your patents. This way you have done so many patents on this particular thing, and hope that you can do more work, better work in future. And we can certainly collaborate with you, no problem at all. I am very much open to collaborations. So that is there. Yeah.
Thank you,
And
sir.
okay, Sanjeev. And over to the Gopa for the next rest of the presentations.
So once again, I want to convey congratulations to the college authority as well as organizer, and for giving me chance to share my little whatever experience I had gained in this platform. Thank you for all.
Sanjeev, we request you to be there as a resource person also, as a chairperson, sorry.
Yes, yes. Sanjeev,
Thank you so much.
Because I have to leave for some time.
Okay, sir.
Thank you so much, sir. I request Dr. Sanjeev Ray to kindly join the session as a chairperson along with Professor Arnab Singh. With your kind permission, let me brief you about the session. We have invited research presentations under medicinal botany and phytochemistry theme, and we received total 37 video presentations. Technical team of medicinal botany, Ms. Neha, Ms. Umera, Ms. Preeti, Ms. Yasmin, Mr. Niraj, along with Mr. Adil, who are our MSc student, has compiled all the videos. We will be playing all these videos in 2 slots of 20 and 17 each. The score sheet for the 37 presentations is already shared with both the chairpersons. At the end of session, you are requested to give valuable suggestions and remark for the presentation. With your kind permission, I request our technical team to start with the presentations. Thank you.
Okay, no problem. Sanjeev, are you ready?
Yes, sir.
Okay, fine. You can, Shital, you can start.
Yes, sir. Just give me a second.
Good evening. Myself, Anusaya Ramdas Kapse. I am a research scholar from Dr. Babasaheb Ambedkar Marathwada University. My research article is Quantification of Nutraceuticals in Chenopodium ambrosioides and Cicer ariantinum vegetables. Introduction. Nutraceutical is a food or a food product which provides health and medical benefits. It is used for prevention and treatment of curing various diseases. Nutraceutical is also the portmanteau of the word nutrition and pharmaceuticals. Nutraceutical may be isolated nutrients, dietary supplements, and diets to genetically engineered designer foods, herbal products, and processed foods such as cereals, soaps, and the vegetables. The present review has been devoted to get acquainted with the nutraceutical value of vegetables. Importance of nutraceuticals are generally in India about 43.5% of children under the age of 5 years are malnourished. Malnutrition, hidden hunger, and deficiency are the major problems in the developing world. For malnourishment, consumption of vegetables is generally considered to be associated with several positive effects on health. The low consumption of fruit and vegetables is related to more cardiovascular diseases. Vegetables are the rich source of carbohydrates, vitamins, proteins, minerals, hence they are known as a protective food. Green leafy vegetables are also the rich source of vitamins such as beta-carotene, ascorbic acid, folic acid, and riboflavin. A World Vegetable Survey shows that 402 vegetable crops are cultivated worldwide. Leafy vegetables, of which the leaves or young leafy shoots are consumed 53%, vegetable fruits 15%, and vegetables with below ground 17%. The materials and the methods. The vegetable was analyzed for different types of the nutraceuticals present in the selected experimental plants. In the analysis, significant amount of nutraceuticals, vitamins, beta-carotene, fiber, nitrogen, protein, and chlorophyll content was found. Total chlorophyll chlorophyll A, chlorophyll B, and carotenoids contents were found in a good amount in the selected experimental plants. Content: the selected leaves of plants were collected from Gaurai district, Bid, in Maharashtra during the month of December 2020, and all the plants were taxonomically identified. The plant material collected in bulk amount and it was shed dry. Then the fine powder was prepared using mortar and the pestle for the further analysis. The experimental analysis of all the selected plants was carried out from the MIT College Center for Analytical Research and Studies, Aurangabad. The experimental result shows, table number 1 shows the amount of a mineral which was found in the Chenopodium and the Cicer erianthum. Table number 2 shows the amount of the vitamins which was found in Chenopodium and the Cicer erianthum. Table number 3 shows the carbohydrate, fiber, nitrogen, protein found in the Chenopodium and the Cicer erianthum. Table number 4 shows the total amount of the chlorophyll A, chlorophyll B, and carotenoid which was found in the Chenopodium and the Cicer erianthum. Table number 5 shows the different nutraceuticals which was found, that is the isoflavone, anthocyanins, lycopene, which was found in the Chenopodium and the Cicer erianthum. Conclusion: in both the experiments planned, it was revealed that the content of calcium, potassium, iron, magnesium, phosphorus, sodium, and zinc found in a remarkable amount, as well as the content of vitamin A, B, C, and beta carotene also found high level. The carbohydrate, fiber, nitrogen, and protein percentages also got in significant amount. So it was the future need to focus on the plant nutraceuticals, which plays as a significant role in human health and the diet. These are the references of my research articles. Thank you.
Hello everyone, I am Alvaro and I currently doing PhD under supervision of Professor Sonjib's Department of Geology, the University of Bardwan. My topic is assessment of antiproliferative, cytotoxic and antioxidant potentials of leaf aqueous extract of Mesia macrophylla 1 EDC. The traditional herbal medicine have been widely used for thousands of years in
Shital, can you please increase the volume a bit?
It's a various pharmacological activities such as antidiabetic, antioxidant, anti-inflammatory, it is as 43.34 plus minus 8.79 and 84.32 plus minus 6.65 mg in 430.60 plus minus 15.21 mg ascorbic acid equivalent gram of aqueous extract.
Actually, this is the way they have recorded.
Activity of
Okay, I understand. Yes, okay, no problem.
Cells. The freeze-dried immersed root of 1.5 to 2 centimeter length of alien cepa were used for the study of growth inhibitory effect of ADM. For the treatment, the roots were exposed to the different concentrations for 3 different time intervals. After the end of each exposure time, roots lengths from each bulb were measured and percentage of root growth inhibitory effects were calculated. In addition, the result showed that at 24 hours, the control inhibition percentage is 0, while at 4 mg per ml, the inhibition percentage is 92.89 plus minus 2.8%. At 72 hours, the inhibition of 4 mg per ml is 98.457 plus minus 0.146%. The acidity value was calculated at 0.31 mg per ml at 24 hours. There is a picture of ADM cepa at 72 hours. This is the control and this is the 0.25, 0.5124 mg per ml concentration at ADM cepa. From the We calculated the cell cycle kinetics in alien separ typical meristem cells at 2-hour microtreat index of control and 4 mg per ml is 8.26 plus minus 0.77 and 4.48 plus minus 0.52 respectively. At 24 hours, the mitotic index control and 4 mg per ml is 7.91 plus minus 0.09 and 0.33 plus minus 0.01 respectively, which is showed that mitotic index is decreased significantly at 24 hours as compared to control. Here, prophase metaphase and anaphase telophase cumulative frequency. The prophase metaphase cumulative frequency was found increased at 24 hours as compared to the full hour, whereas the anaphase telophase frequency was found decreased at 24 hours as compared to 2 hours. The average cell percentage was found to be significantly increased as the time and dose increases. As you can see that the percentage of abnormalities of control is 1.8 plus minus 0.06 at 2 hours, 4 mg per ml it is 7.04 plus minus 0.33. At 24 hours, the percentage is increased and it is 14.05 plus minus 0.37 per 4 mg per ml, which is significantly increased from control. From different type of abnormalities, sticky chromosome found to be highest, followed by anaphase base. Other type of abnormalities such as polar deviation at metaphase, polar deviation at anaphase, variant chromosome, Napper chromosome, host cell hyperplasia also seen. This is the graph showing total apparent cells, anaphase base, sticky chromosome, polar deviation at metaphase, polar deviation at anaphase, host cells, and hyperplasia. Picture showing production of different type of mitotic abnormalities in alien separ. Here is the major findings of the study. Data indicate a concentration-dependent increase in the FAB activities of the LEAM as shown by the decreasing OD values. The half maximum effective reducing power concentration for ascorbic acid was found to be lower than LEAM. LEAM showed a concentration-dependent increase in the Wph free radical scavenging activity. The EC50 of LEAM was required nearly 1.5 times higher than standard ascorbic acid. LEAM also showed relatively higher total antioxidant capacity in terms of ascorbic acid equivalent content, total phenolic content as gallic acid equivalent, and total flavonoid content as quercetin equivalent was found to be in higher amounts. LEAM induced a dose-dependent root retardation effects in alien separ root tip cells. Mitotic index depression biosyscol anti-proliferation revealed that LEAM treatment could reduce mitotic index in alien separ root typical meristem cell and reduction rate of anaphase telophase cumulative frequency and increasing rate of prophase metaphase cumulative frequency. Microscope study revealed that LEAM treatment could induce different types of cytological and chromosomal abnormalities in Uranian root tip cells like sticky chromosome, anaphase bridge, variant chromosome, C metaphase, polar deviation, ring chromosome, host cell hyperchromasia, giant. So here is the conclusion. Meso macrophylla is a rich source of phytochemicals and it has been active components for antioxidant activities, root length inhibition and reduction of mitotic index induced by the plant extract on the root tip cells of alien separ indicates its anti-proliferative and anti-mitotic activity, thus preventing cell reaction. A dose-dependent increase of abnormality percentage indicates the cytotoxic potential of the plant. The presence of studies suggested the possible therapeutic use of meso macrophylla, though further investigations are required for determining the active function and understanding the molecular mechanism of isolated compounds in vivo. And here are the selected. Here are some of my. Thank you.
Hello, myself Apura Prashant Bhatkande. My topic for today is antenatal activity of polyherbal formulation on Malassezia fruit. Topics which I am to cover are introduction, aim and objective of the study, the different materials and methods which I am using, then the result and discussion of the study and the references. Cosmetic and personal care product has become the integral part of our day-to-day life. But from where the word cosmetic has come? The word cosmetic is originated from Greek word cosmeticos, means adorn and preparation. But Federal Food, Drug and Cosmetic Act, which has come in 1940, defined the cosmetic as it is the article intended to be rubbed, poured, sprinkled, or sprayed on, introduced into, or otherwise applied to the human body for cleansing, beautifying, promoting attractiveness, or altering the appearance. As we all know, there are different types of cosmetics and personal care products which has three different types. The products which are the rinse off products or the wash off product and the applied ones. Rinse off products are the products like the toothbrush or the shampoo, which we just apply to the hair and wash it. Whereas the leave-on products like a cleanser or the lotion or the sunscreen lotion, etc., we are applied on the skin and it is remain for certain period of time. Whereas the applied ones, that is the makeup product, like the mascara, eyeliner, lipstick, are come under the applied ones. But all these products are not a single made up from single component. In order to form this cosmetic and personal care products. There are the different colorants which are responsible to give the color to that product. There are the different preservatives which are necessary to increase the shelf life of the product, also to maintain the stability of the product. There are thickness to give its uniform consistency to that product so that you can uniformly apply it to your skin or the hair. Then there is the surfactants like sodium lauryl sulfate, LS. These are mainly found in the shampoo so that there is the formation of foam. Then silicone. These products are mainly used in the hair care treatment in which gives a shiny appearance to the hair and it makes it easy to maintain your health of your hair. The ingredients used in cosmetic products are mainly derived from the synthetic source and they are made from harsh chemicals. Prolonged exposure of such cosmetic products which contains harsh chemicals is not good for your health. It may be carcinogenic and it can lead to the toxicological effect to the human health. To get rid of such products, there is a different alternative which is called a cosmeceuticals. What are the cosmeceuticals? Cosmeceutical is nothing but the hybridization between the cosmetic and pharmaceuticals, which not only cure the alignment but also improves the health conditions. Cosmeceuticals are different than cosmetic products because ingredients which used in a cosmeceutical products are derived from the nature. They are plant-based or microbe-based. So there is a no added chemicals or preservatives or colorants in them. Then my study as we all know, dandruff is the most abundant health problem all over the world, which is caused by the microorganism called Malassezia furfur, which is nothing but the fastidious growing yeast. But instead of using shampoos containing harsh chemicals, we can use the plant to get rid of this dandruff. Plant material which I use for this formulation is the pumpkin pea, orange pea, and the currant plant. As we all know, these plants are rich source of phytoconstituent, and to procure this phytoconstituent, its extraction is required. There are different extraction techniques available to extract the phytoconstituent, like rotary shaker, maceration. But in my study, maceration gives me the best yield of the all plant materials. Further, study proceed with the conducting a risk diffusion method in order to find out the inhibitory activity of plant extract on the microorganism. The assay is conducted by using the disk diffusion method, where all three plant material Along with its formulation, it's subjected to the microorganisms, amongst which formulation gives the larger zone of inhibition as compared to the individual plant matter. These are the images where you can observe the formulation gives the larger zone of inhibition as compared to the individual plant extract. Also, study proceed by conducting the millimetry inhibitory concentration test in which confirms the minimum concentration of a concentration required to inhibit the growth of microorganisms. This is the 96 well plate which we used for the analysis of the minimum millimetry concentration test. So here I can conclude that adapting pathogens develop the resistance against antibiotics and antifungicidal agent, which creates challenges in order to find out antimicrobial activity of plants and their metabolites. Therefore, the study also aids in utilizing fruit and vegetable peels which are thrown into the environment as agro-waste and can be considered as remedy in the form of anti-dandruff dry shampoo. It can be considered as an economical, eco-friendly, and preservative and effective in dandruff management. These are the references. Thank you.
Hello everyone, my name is Ashutosh Satish Zadav. Currently, I am an MSc student having a specialization subject as analytical chemistry. I am doing my master's from Guru Nanak Khalsa College. So today, I will be presenting about my research project which we have performed. Due to the pandemic, our department opted for an in silico approach, that is, molecular docking through various softwares, tools, and websites. This gave us an exposure to various new concepts, methods, and techniques. In this presentation, I will be discussing about our research which we performed. Starting with the title, our theme of the research is in silico anti-inflammatory activity, evolution of phytochemical through molecular docking approach. Starting with the introduction, the main aim of the experiment is to explore the phytopharmaceutical potential of various phytochemicals and their application, and using achieving the desired anti-inflammatory effect. So if we look into the actual inflammation, what is inflammation? So inflammation is a result of our body immune system reacting to foreign harmful objects such as pathogens, bacteria, virus. It results in pain, swelling, redness, burns, which is considered as sign of foreign invasion. If we dive into the cellular level, we can see, as shown in the following figure, there is a gush of blood flow in the affected area, which results in warmth of the region. Due to entry of foreign substances, body immune system kicks in, which results in release of inflammatory cells. Upon release of inflammatory cells and cytokinase is a substance that stimulates inflammatory cells. As shown in the figure, our WBC gathers to the affected area and releases cytokinase, which results in inflammatory cell release. As this cell causes an inflammatory response, it traps the bacteria and other offending agents and starts the healing process in our body. So as we learned earlier, inflammation is simple, yet it can also cause hazardous effect if not attended properly. So in this study, our main objective is to look for a solution. So we achieved our results by using various softwares that help us to simulate our target protein and desired drug. So in our study, we mainly considered two proteins that are cyclooxygenase 1, that is also known as COX-1, and indicible nitric oxide synthase, that is also known as INOS. So as we see on the previous slide, we can see that there is a proper procedure in which we carry out our research. So first of all, starting with, we need to search the structure of protein in order to bind it. So we downloaded our protein structure from Protein Data Bank, which is also known as PDB. It is a website available for everyone. The next step was to search for the desired drug. The desired drug was taken from Dr. Duke's database. Dr. Duke's database is an online cloud-based drug site. To download the drug structures, we used PubChem. It is also a cloud-based website. After gathering of data, we need to have some desired softwares in which we can carry out a further docking process. So the first software that we used was Wph Discovery Studio. The next software was Ufsc Chimera. The third software we used was Pyrax. After that, to analyze the data, we used some online sites that were Swiss ADME and Protox 2. So the first software we use is Wph Discovery Studio. So basically, this software, or you can say tool, is used in analyzing and modeling molecular structures, sequences, and other data of our desired protein. So basically, this tool is used in editing, viewing the data. It is also used to perform some basic data analysis. So in our research, we use this tool to remove unwanted water molecules and ligands that were bound to our protein. So the next software we use is Ufsc Chimera. So basically, this tool is used for interactive visualization and analysis of molecular structure. So in this software, we basically added hydrogen atoms to our protein, which will help us in further docking process. So the last software which we use is Pyrax. Pyrax is a virtual screening software. It is used to screen libraries of compounds against potential drug targets. So in our research, we basically dock our desired drug with protein with the use of this software. After gathering the data from the following softwares, the next step is to analyze this data. In order to analyze this data, we use these two websites that are Swiss ADME and Protox 2. So starting with Swiss ADME basically gives us information about various parameters such as bioavailability, drug likeness, and lipophilicity. The next is Protox 2. Protox basically helps in prediction of toxicity. After analyzing this data, we found out that after docking for 150 phytochemicals, we found out that 59 had the highest binding affinity, which ranged from -8 to -11. So in these 150 molecules, basically, we found 58 molecules that had desired binding affinity, which we want. In this 59 drug candidates, we found out that 10 phytochemicals obeyed Lipinski rule of 5 and had the toxicity in the desired range. So in our research, we found out that COX-1 had 6 such phytochemicals and INOS had 7 such phytochemicals out of 10, which were suitable drug candidates for our research project. The following graph represents the drug candidates that were docked with COX-1. These are the drug candidates which are in the desired range. So moving towards the conclusion, the drug candidates such as bavacillin, bermestine, alkylidine, masic acid, etc. are some of the molecules that we think are best for the future studies in the inflammation. As I said earlier, we found out that these 10 molecules obeys the Lipinski rule of 5, and the toxicity was also in the desired range. But even if the toxicity was in desired range, out of this, we found out that some of these drug candidates had toxicity level between 3 to 5. So to tackle it, we can increase the LD50 value. LD50 value is basically high tolerance to poison. The following diagram shows the 3D and 2D image of the drug molecule docked with our COX protein. Moving toward the recommendation, we recommend these 10 molecules for further studies. These drug candidates satisfy our desired results, which we expected. These results will help us in tackling a simple yet dangerous problem, which is inflammation, by neutralizing it and removing it from the host. Even if some drug candidates are mildly toxic, they can be used by slightly modifying them. These are the references which we took. I have mentioned some of the sites of the softwares and websites which we referred. I thank all the dignities and my dear friends for hearing me patiently. Thank you.
Very good morning. I'm Bikushni Bakusa, a research scholar from the Department of Life Sciences, Manipur University, and I'll be presenting phytochemical screening, antioxidant activity, and GCMS analysis of Xanthoxylum armatum. Medicinal plants have been used in indigenous herbal practices since time immemorial. They are rich in bioactive constituents, providing both health and nutritional benefits. Xanthoxylum of the family Rutaceae comprises of about 250 species worldwide, with 13 species known in India. Xanthoxylum armatum is an aromatic plant of about 6 meters tall, commonly called as the two-egg tree or dimum. It is extensively used in the indigenous system of medicine as a carbonated, stomachic, and emphyematic. The bulbs are used to treat asthma, diabetes, diarrhea, and cholera. The fruits are used as an analgesic, antiseptic, deodorant, and disinfectant. It is used to treat fever, tonsillitis, colds, and indigestion. The fruits and seeds are used to treat rheumatism, muscle cramp, varicose veins, and varicose ulcers. The objectives include screening of phytochemicals present in the leaves and pericarp, and to determine the total phenolics, flavonoids, tannins, and anthocyanins content in the leaves and pericarp, and to identify the different compounds that are present in the methanol extract of the leaves. Materials and methods: collection of plant materials. Fresh leaves and fruits were collected from Spati district of Manipur. It was authenticated at Botanical Survey of India, Shillong. The samples are washed, air-dried, and ground to fine powder. And the powder is used for extraction. Extraction of samples was done following Yadav and Agarwal 2011. Qualitative phytochemical analysis. For qualitative phytochemical analysis, Hobby 1973, Superwoman 1993. Following CO calculus method according to Kavita of 2014. Determined by aluminum chloride colorimetric assay according to Koyo in 2020. Anthocyanin content was determined according to Ibrinosa et al. 2013. And the total tannin content was determined by following CO calculus method according to Tambay in Bamboo 2014. For antioxidant activity, two radical scavenging activities were carried out. First, the DPPH radical scavenging activity, which was done following Islam et al. 2018. And the DPPH radical scavenging activity, which was done following Gray et al. 1999. GCMS analysis to methanol extract of the leaves for cell central instrumentation laboratory, Punjab University, and an in silico study was conducted on the identified compounds to predict their toxicity and mutagenicity using test software. Results and discussions. The qualitative phytochemical screening of the aqueous extract shows the presence of proteins, carbohydrates, phenols, tannins, saponins, glycosides, steroids, terpenoids, and alkaloids in the leaf extract, while proteins, carbohydrates, phenols, tannins, saponins, glycosides, steroids, and terpenoids are present in the pericarp. The quantitative phytochemical analysis shows that the methanol extract of the leaf has the highest total phenolic, flavonoid, tannin, and anthocyanin content, which is followed by the methanol extract of the pericarp. Antioxidant activity. From the graph, we can see that the methanol extract of the leaf has the highest antioxidant activity, with an IC50 value of 89.29 for Wph and 55.81 for Apts, which is comparable with ascorbic acid, which is an IC50 value of 29.78 for Wph and 24.97 for Apts. The GCMS analysis of the methanol extract of the leaves resulted in the identification of 26 compounds with antioxidant cyclohexyl acid, Metastatic octadecanoic acid, cyclohexene, and cyclohexanol, two amino methyl are some of the major compounds. Of the 26 compounds that were identified from GCMS analysis, reputations were removed and 20 compounds were screened for mutagenicity, of which 90% showed negative result, with 10% showed positive result for mutagenicity, and 22 compounds were screened for toxicity, which 65% were moderately toxic, 15% slightly toxic, 50% practically non-toxic, and 5% very toxic. From the above, we can conclude that the methanol extract of the leaf showed relatively higher total phenolics, flavonoids, tannins, and anthocyanin content. The methanol extract of the leaf has the highest antioxidant activity. Xanthoxylum armatum contains phytochemicals with antioxidant activities, and further research of the species is required to aid in the discovery of new compounds and future drug discovery. In silico prediction can provide significant understanding in the validation, safety, and regulation of herbal recipes, and search for novel leads for drug discovery. Estimation of toxicity can be a criteria for selecting the drug candidates. These are some of the references I used. Thank you.
Good morning to one and all present here. I am Ms. Bhakti Sawant, ex-student of Guru Nanak Khalsa College of Arts, Science and Commerce, would like to present a topic in the category of medicinal botany and phytochemistry. The topic is screening of phytochemicals and in vitro seed culture of selected Asteraceae mango. Plants are reservoir of different kinds of bioactive chemical agents and often being utilized either in the form of traditional preparation or as pure active compounds. Plants contain compounds such as phenols, flavonoids, quinols, tannins, terpenoids, essential oils, and alkaloids, which act as antimicrobial agents individually or in combination. Thus, therapeutical phytochemicals have been used as model candidates for synthesis of chemical drugs and pharmaceuticals. In the present study, different members of Asteraceae family were used. In the first half of the study, plant perennial flowers were used. The different flowers were Aster species and of Vegeta species. Preparation of extract was the first step. Petals were dried for 5 days. 0.5 gram was dissolved in 50 ml of water and boiled, and a concentrated extract was created. This extract was subjected to quantitative analysis, which included the test of tannins, saponins, reducing sugars, alkaloids, terpenoids, flavonoids, phenols, cardioglycoside, anthraquinone glycoside, and thiogenetic glycoside. Aster showed the presence of saponins, reducing sugars, terpenoids, flavonoids, phenols, cardioglycosides, and cyanogenetic glycoside, whereas Digita showed the presence of tannins, saponins, reducing sugar, alkaloids, terpenoids, flavonoids, phenols, cardioglycoside, and cyanogenetic glycoside. The main aim of the study was to isolate a certain phytochemical because phytochemicals have different medicinal uses. For example, cardioglycoside is used to arrest sodium and potassium pump. Similarly, lutein is also being used as an medicine to cure eye diseases such as cataract and macular degradation. Thus, to isolate and lutein, an economically cheaper method was used, that is column chromatography. After a lot of standardization, the stationary phase was used as silica gel, and the solvent system was hexane and ethyl acetate in the ratio of 19:10. The extract obtained from column chromatography was subjected to TLC, where the RF received as 0.88 and 0.71 in Aster and Vegetas respectively. This was compared to a previous research paper, and the presence of lutein was declared. The second half of the study was to inoculate the callus culture. The main motto of this study was to increase the phytochemical content in the plants by using illiciter. For this, inoculation of callus was the first step. The seeds which were used as Aster, Dahlia pinnata, Zinnia elegans, and Chrysanthemum indicum. In this method, MS media was used, and the main hormone which played a role in induction of callus was 2,4-dichlorophenol acetic acid. After weeks, callus was seen in Chrysanthemum indicum, Astus species, and Xenia elegans. Conclusion: the study of phytochemical screening of Tagetes and Astus showed the content mainly flavonoid, alkaloid, glycoside. Presence of these bioactive compounds hold great medicinal value and have definite physiological significance on human body. The isolation of lutein compound using column chromatography is economical as compared to other method of extraction. The quantification, purification, and bioassay of bioactive compound, especially lutein, holds great promise in therapeutic application. Thank you.
Hey to all. I am C.K. Bhavini, Research Scholar, Department of Microbiology, Mohammed College of Arts and Science, Chennai. My presentation topic is phytochemical analysis of Alvaro species. Marine is a huge hub of biota and recently exploring source of potential compounds having many applications in various fields like medicine, environmental, food and pharmaceutical, etc. Antimicrobial resistance is one of the major global threats which needs better solution from the natural sources. Seaweeds are one of the potential marine algae with a variety of phytochemicals to explore. The edible green seaweed Ulva species was chosen for the present study. The objective of the study is sample collection, extraction preparation, and phytochemical and GCMS analysis of the sample. The fresh samples were collected from the coastal region of Mandapam, Tamil Nadu, and the seaweeds were thoroughly washed with seawater to remove the epiphytes and impurity. In lab, the samples were cleaned with water and shade dried. The dried seaweeds were grounded to powder to make the extract. Crude methanolic extracts were subjected to standard phytochemical qualitative analysis and also sent for the GCMS analysis. Methanolic extracts were subjected to qualitative analysis for phytochemicals like alkaloids, phenol components, terpenoids, flavonoids, and tannins, steroids, and safenins. Yellow precipitation indicates the presence of alkaloids after adding hydrochloric acid and saturated citric acid solution to the extract. A reddish brown green coloration of the interface indicates the presence of terpenoids. The dark brown color after adding ferric chloride to the extract indicates the presence of phenolic components. The flavonoids and tannins and glycosides also were present in the phytochemical analysis of the Alvaro species. The steroids and safenins were absent in the present phytochemical analysis. The methanolic extract was sent for the GCMS analysis. GCMS analysis revealed the presence of different phytocomponents with various biological activity like antioxidative, antimicrobial, antibiofilm, and antifungal. The previous research shows that fatty acids like dodecanoic acid, pentadecanoic acid, and hexadecanoic acids are potential components with anti-inflammatory, anti-proliferative, antioxidative activities. This present table shows the major components which have been identified in the GCMS analysis with their structure, retention time, molecular weight, and molecular formulas. The present study is an attempt to explore and identify the potential components of seaweed Alvaro species. Phytochemical and GCMS analysis of Alvaro reveal the presence of different components and metabolites with a wide range of biological activity. The preliminary study paves the way to isolation and identification of these bioactive components to further explore the applications of the metabolites as a remedy for antimicrobial resistance. Few references to support my study. I would like to thank my guide, principal and management of Mohammed College for giving me the permission to do this presentation. I would also like to thank the organizers for giving us a wonderful opportunity. Thank you.
Hello everyone, my name is Chetana Shetty and under the guidance of Dr. Maitri Shah, I am going to present a part of my research work titled Comparison Between Two Phytochemical Extraction Methods. Introduction. Medicinal plants known to be a rich source of drug-developing chemical constituents. One such medicinal plant is Xanthellus pereira, which is also commonly called as abuta. They are rich source of polyphenols, flavonols, anthocyanins, and many others. Now these phytochemicals, they help to produce physiological action in the body. Xanthellus pereira is one such plant which has got many therapeutic value. They help to reduce or it helps to reduce fever and relieve pain. Xanthellus pereira is also a well-known blood purifying agent and it has got anti-inflammatory properties. There are various types of extraction methods which we have seen, like Soxhlet extraction method, microwave assisted method, cold and hot extraction method, and so on. Now they depend on various factors. Factors which really matters are their availability, time, cost, feasibility, and ease. There is one such method which has been recently upcoming or popular method, which is ultrasound extraction or sonication method, that is by using sonicator. Now, improved extraction techniques will definitely lead to better utilization of antioxidant from the plant. So it is necessary that we come to a better model of extraction for all the plants. Now, thus the study includes effect of sonication treatment on plant extract. The study will help in identifying and finding out the feasibility of using sonicator and sonication method as one of the extraction techniques. Thereby performing extractive value and phytochemical screening of solvent extracts. Material and method. Fresh Xanthellus pereira plants were collected from Thane region. They were authenticated by Platter Herbarium. Now these plants were washed and later dried in a oven at 40 degrees Celsius for consecutive 5 days. Aerial parts of the plant, that is leaves and stems, were considered for study. Dried plants were powdered and stored for further study. Extractive values were calculated by standard methods. Same way, phytochemical analysis were also done by standard procedure. Methodology. Cold extraction method or maceration or by maceration. Plant powder, that is around 5 grams of plant powder was taken in 5 conical flasks containing solvent like methanol, alcohol, dichloromethane, petroleum ether, and water. Each solvent was around 100 ml. Samples were kept overnight in solvents. The filtrates were evaporated using a water bath and then they were stored in a refrigerator at 4 degrees Celsius for further phytochemical screening. Same way, for ultrasonic assisted extraction, 5 gram plant powder was taken in 5 different conical flasks containing 5 different solvents, 100 ml of 5 different solvents. They are methanol, ethanol, dichloromethane, petroleum ether, and water. Later, samples were sonicated for 30 minutes in a sonicator. Temperature was controlled by changing the water periodically. The filtrates were evaporated using a water bath. Later on, they were stored in a refrigerator at 4 degrees Celsius for further phytochemical screening. Observation. filtrate obtained after cold extraction method, and second table shows filtrate obtained after ultrasonic assisted extraction method. Observation: extractive value these are the results. As you can see, water, that is the aqueous medium, showed highest yield for cold extraction method as well as for ultrasonic assisted extraction method, that is 22 for cold extraction and 20 weight per person for ultrasonic assisted extraction method. These are the observation: phytochemical screening by cold extraction method and phytochemical screening by ultrasonic assisted extraction method. These are the results. Like I mentioned before, extractive value maximum for extraction method for aqueous extract, same way it was maximum for ultrasonic assisted method for aqueous extract. Percentage yield was maximum for aqueous extract by conventional method. Similarly, the rate of extraction was found to be maximum in aqueous extract for the modern method. Now, the data obtained for phytochemical analysis were similar. However, the extractive yield varied between the process. So, this is what my final conclusion are. Using minimum solvent, ultrasonic assisted extraction could be an efficient time-saving technique, but standardization of each variable is must, which will lead to better optimized extraction of phytoconstituent by the ultrasonic extraction method. Standardization of extraction procedure is important for obtaining the quality outcome plan. Further study include standardization of each variable. References. Thank you.
Hello, respected teachers, organizing committee of the Khalsa College. My research topic is medicinal uses of plants practiced among the tribals in the Birbhum district, West Bengal, India, a quantitative ethnobotany approach. Myself, Dr. Swarnil Manon from MSc Women's College, Bardhaman. Introduction. Ethnobotany is the multidisciplinary subject which deals with the relationship between man and a culture, plants and environment. Among the different subdisciplines, ethnomedicine is the subdiscipline which is related to cultural heritage of knowledge, beliefs, experiences, practices related to food drugs. Like any traditional knowledge system, documentation of traditional knowledge is essential prerequisite for preservation of this knowledge system, for biospection, drug discovery, as well as conservation of the medicinal plants. A large portion of the tribal people over the world practice and use medicinal plant effective curing of different diseases and ailments. This essence of knowledge is still largely unexplored in terms of their scientific documentation and pharmaceutical prediction. For past few decades, quantitative ethnobotanical tools are being used in ethnobotany to understand the richness and levity of herbal wisdom. From India, this kind of quantitative tools are being used for documentation of restoration of plants. However, from Birbhum district of West Bengal, this quantitative approach is not used as much as possible, but the conventional techniques are used for documentation of the knowledge. Here, we got some scope to study in this approach. Objective of the study is to document the traditional herbal knowledge of the studied area related to phytoresources used by the tribal people of the Birbhum district, which will be helping in the building of the state as well as national level ethnography. To analyze the ethnobotanical data by conventional ethnobotanical approach, to identify the culturally important medicinal plant species employing suitable quantitative ethnobotanical indices, and to incorporate the new information on ethnobotanical uses to existing data bank of ethnobotany. This is the study area. We have explored 3 forest pockets, tribal dominated forest pocket, where mainly Santhal people are there, and the total forest area of the district is 159 square kilometer. Among the people, 70.68% literacy is existing. Materials and methods. Regular ethnobotanical pilgrims were made from 2018, and total 80 tribal knowledge holders and knowledgeable persons were interrogated by purposive sampling method, and prior from consent was taken. GPS data of the place surveys were recorded, and during the survey, semi-structured interview guided to technique in local evaluation were employed to collect the data about the local traditional knowledge and the medicinal plants, and their tribal name, parts used, and mode of administration, disease code, etc. Have been tabulated. To confirm the authenticity of the collected data, cross-checking were made by employing inventory intelligence. Plant specimen identification visualization. Standard literature have been followed. Consultation of herbarium made in Central National Herbarium, Kolkata. Consultation of International Planting Index and Planting Database have been followed in the website. And finally, the herbarium specimen were reserved by standard technique in the NHMS College Botany Department. This is the methodology which I have followed from the walk-in interview method. We have followed the inventory interview and in loco identification. Guided tool technique is also followed, semi-structure interview and visual simile method to cross-check the data. Categorization of the disease categories is followed by Cook method of Cook in 1995. 14 broad disease categories have been registered. For data analysis, we have followed FIC and FL technique. FIC is the information consider factor, where U is the number of use report, NP is the number of taxa. Here, fidelity level means NP by N into 100, where NP is the number of respondent, the number of respondent that use the plant for a specific disease, for any disease. Results. We have documented 40 medicinal species, among which 34 are from dicotyledonous and 6 are from monocotyledonous species. This is the tabular presentation of the drugs used in different medicinal purposes. Mainly roots are used. Here you can see 50% of the plants used for their root rhizome suitable, and herbaceous plants are mainly used for medicinal purposes. In the informants, it has been found that the horizontal knowledge transfer is actually prevailing, and mainly males are dominating in the culture of medicinal practices. They belong to the age group mainly 50 to 75 years old, and the middle age group 13 to 49. Literacy is 35%. This is the tabular formation form of the presentation of the different plants used in different phytotherapies like Crenum aceticum, Diaspora abilifera, This is the continuation of the previous TV. Wph Scavenging Activity, Giacomo, Theodorm, Decibel, Jacopanana is used mainly in dietary purpose. The root is used in oral root. Here, finally, it has been found that several plants are actually undocumented in terms of their medicinal uses. We have actually consulted different literatures, and it has been found that Ufsc is use of the Geodrum, Tlc Profiling, and Tlc Profiling. All these are found very much new to Indian ethnography. This is the pictorial presentation of the different medicinal plants: Wph Scavenging, Carrier Arborea, Pancreatic, Parigala Total, Geodrum, Brensicodum, Placidium, Multilecturity, Ufsc. This is the tabular form of the FIC data, where it has been found that for injuries and for poisoning, quite high percent, quite high value of FIC is found, and for poor lactation, it is quite high also, 0.777. For fertility level, it has been found that in case of the Premna herbacea, it is mainly used in gout, and the consent of this use is very much high among the informant, that is 93.75%. Summary of the finding is altogether we have documented 40 ethnomusician plants that belong to 32 families, and this knowledge transmission is actually vertical mode, and mostly herbaceous plants are used. Roots parts are mainly used among the other parts, and ethnomusician uses are very much new. Some of the ethnomusician uses are very much new. Again, ethnomusician FIC value indicates that diseases like menstrual problem, muscular pain, diarrhea, dysentery, snakebite, etc. occurs very frequently in the tribal society of the district. 7 plants species got highest fertility level, that is 100%, like Distranthus folinus, Glaucidia, Multilicula, etc. And 11 plants out of the 40 plants got above 70% fertility level, like Hemitus, Lucinicus, Premna herbacea, etc., which may be considered as the potent plant, because these plants may be in future can be used as potent in case of biospection study. From FIC value, it may be considered that the ethnomusician plants are facing high exploitation pressure. That means whenever some plants got high percentage of fertility level, it may be possible that this plant is exploited very much in a higher rate. from the forest localities. In near future, there is every possibility of these plants may be rare from those districts, and so they need priorities for early conservation. Conclusion: knowledge of the medicinal plant, which is based on the regular experiences, treatment of 14 different disorders, measurement of local knowledge, ethno-directed validation of the technology uses, quantitative ethnometric analysis provides a better option to select the medicinal plants for searching active principles, synthesize new and comparatively cheaper, effective, eco-friendly biomedicine. Measurement of the use pressure in the impinging on a species, priorities for conservation for the forest product of the district. So these are the literature which I have consulted. Thank you, Organizing Committee of the Khalsa College, for giving the chance for presenting my research work. Thank you.
A very good morning to all the demonstrators. Myself is Alvaro. Today I'm going to present a paper presentation based on ethnomedicinal plants used as home remedies in Sutana village, East India Hills district of Meghalaya. Medicinal plants are considered to be a source of traditional remedies that can be provide a wealth of information for the creation of contemporary medicines for the betterment of future. The use of natural ingredients available in the home for treatment of common ailments is known as home remedies. Alternative medicines are becoming increasingly popular as the public interest in health and wellness grows up in daily life. More than 3.3 billion people in less developed nations use medicinal plants on a regular basis, making medicinal plants a local heritage with global significance. Coming to the objectives, the primary goals of this research was to document the traditional knowledge of home remedies used by Sutana village residents. Coming to the methodology, in order to document the utilization of indigenous medicinal plants as a home remedies, a survey was carried out during the month of September to December 2021 in different localities of Sutana village. The geographic coordinates are 25° north and 92° east. And multiple inquiries were made to understand their knowledge, diagnostic methods, and disease treatment, and data were collected on the specific parts of the plant use, collection, method of uses of the drugs, and dosage of administration. The medicinal plants specimens were collected, and a herbarium was created for the future conservation by following guidelines given by Jain and Rao 1997, Maiden 2004, Sesha G. Rao 2016, and for identification, we are using specimens, authentic specimens, journals, and other sources, and for confirmation, we took it to the Botanical Survey of India. And this is the map depicting the location of the research area. Coming to the results, there are about 51 ethnomedicinal plants that are beneficial to our prior health. The most commonly presented families were Phytochemical, followed by Plc, Phytochemical, and so on. The graph represented here the core form analysis of medicinal plants, in which herbs made up the highest proportion, represented by 26 species, 50.9%, followed by three species, 35.2%, then shrub, 3 species, 5.8%, and then fiber in wine, 2 species each, which is of 3.9%. The herbs being the highest uses is due to the easy availability and high effectiveness in the treatment of various ailments in comparison to other forms. The local people use different parts of medicinal plant species for the preparation of traditional remedies. Mostly they use fruits and seeds, in which 17 species, 3.3%, live, 16 species, 31.3%, the entire plant, 7 species, 13.7%, roots and rhizomes, 4 species each, 7.8%, the flower, about 3 stamens. This table represents the 51 ethnomedicinal plant species, in which botanical name, family name, common name, units, mode of preparation is given. From these tables, we can say that the above-ground plant parts are utilized more for medicines than the underground parts. The most common methods of preparation were decoction and base of plant parts. Plant decoction is extracted by flushing plant parts, and sometimes plant parts are boiled with water and the liquid is decanted. The most common health problems are stomach disorder, blood clotting, hypertension,
hypotension, cardiac, and other elements. It is also observed that most of the remedies consisted of a single plant parts and more than one way of preparation. However, some of the remedies consisted of different portions of the same plant species to treat one or more diseases. The remedies are prepared either from freshly gathered plant parts or dried plant parts depending on their intended function and are either administered orally. For example, ginger, which is a very common species belonging to Zingiberaceae family, in which a paste is made and applied on the head for 1 to 2 hours and wash it, and then it is also cut into small pieces and mixed with sugar for puff and hoarseness of voice. Coming to the conclusion, herbal medicines are likely blessing in rural areas where modern medical facilities are insufficient. However, knowledge of these herbal remedies is declining day by day as the young generation in rural areas is not much interested in it. Therefore, more efforts must be made to document such knowledge before it vanishes. Here is the reference which I have taken. Thank you.
Good morning, everyone. My name is Gloria Mintan, supervisor at Ufsc Chimera from the Department of Botany, Nagaland University, Manila. Today, for the presentation on Internet and Business Session, a conference on novel approaches and right practices organized by Department of Botany in collaboration with Ufsc Chimera and Taxa College. My topic is critical scavenging and antioxidant activities of intermediate expressed in two wild continuous species in Nagaland. India. My category is medicinal botany. This is my presentation outline, introduction, objective, measures and methods, debate and discussions, and conclusion. What are free radicals? Free radicals are highly reactive non-stable atoms, ions and molecules, and they are formed during the normal cellular metabolism. However, they are also biologically very important part of our body. However, when this concentration increases, they initiate oxidative stresses and causes damage to cellular contents and is also responsible for many metabolic disorders, which include cancer, diabetes, neurodegeneration, etc. However, studies have found out that antioxidant supplements or antioxidant containing foods may be used to help the human body to reduce synthetic antioxidants that present are butylated hydroxyamines, butylated hydroxy, toluene, propyl gallate, and third butyl hydroquinone. However, they are suspected of being responsible for liver damage and hence acting as carcinogens in laboratory animals. Therefore, research for new products with antioxidant properties and fewer side effects is very active domain of research. Lentinus fishes, they are a genus of Polyphoraceae, and they are edible mushrooms that offer several important medicinal compounds. Studies by Delay Tevitic shows that they have become widespread as it has shown exhibiting antioxidant, antibacterial, and anti-tumor and also antiviral, immune modulating, also cholesterol regulating effects. Therefore, these very bioactive compounds and biological activities of this lentinus species have received much attention in recent years because of the increasing interest in human health. We have selected two various species, Lentinus pygonus and Lentinus hydrocardus, for this very study, and they are edible wood residuomycetes, and they are consumed by local communities in Nagaland. These two very figures shows the cumulative appearance of word growth and also trend topics since 2006 till date and shows its relevance for the present study. This data has been taken from Scopus bibliometry. The objective of the study is to evaluate the antioxidant property by the following assays: Wph free radical scavenging assay, hydrogen peroxide scavenging assay, phosphomolybdenum assay, and reducing power assay, and also a couple of flavonoids were quantified. Materials and methods. Wph free radical scavenging assay is a widely used method to evaluate antioxidant activities in a relatively short time compared with the other methods. The hydrogen peroxide scavenging assay determined by Rich and co-workers, reducing power assay by Oyazu and co-workers with some modifications, phosphomolybdenum assay by Brito and co-workers as follows. Also, termination of total permanent content was determined by the aluminum chloride colorimetric method as described by Chang and co-workers with modification. Preparation of mushroom extract. The extracts are prepared isoprene, 300 ml of methanol at 150 rpm for 24 hours. Were filtered and then concentrated in watery evaporator, and fully dried extracts were sealed and stored in -20 degrees Celsius for further experiment. Resultant. For the resultant discussion, Table 1 shows IC50 values of botulinum, carbonate, and intermediate hydrocarbon. The antioxidant, the PPPH, free radical scavenging assay of maintenance, carbonate, and maintenance hydrocarbon exhibit significant free radical scavenging potential of IC50 of 126 microgram per ml and 150 microgram per ml respectively. Maintenance carbonate displays strong hydrogen peroxide scavenging activity of IC50 value of 55 microgram per ml, whereas maintenance hydrocarbon exhibited IC50 value of 144 microgram per ml. The antioxidant activity of both the species of phosphomolybdenum assay was not significantly different. However, maintenance hydrocarbon showed that in total antioxidant capacity, that is IC50 value of 2.78 microgram per ml, while that of maintenance hydrocarbon was 2.34 microgram per ml. In table 2, it quantifies the flavonoid contents of maintenance carbonate and maintenance hydrocarbon through spectrophotometry. Significant amount of flavonoids are present in both species, with 5.355 mg quercetin equivalent per gram of dry extract in maintenance carbonate, and that of 4.15 mg quercetin equivalent per gram of dry extract in maintenance hydrocarbon. In figure 2, we see the antioxidant activities of maintenance in both maintenance carbonate and maintenance hydrocarbon. The results from this various graph shows the various antioxidant capacity. FA reveals that the antioxidant activity of the mushroom extracts was concentration dependent, and all extracts exhibited strong antioxidant properties. The scavenging effect of ascorbic acid in all the graphs shows that ascorbic acid are higher than all methanolic extracts of mushroom species. Same situations were also reported from previous studies as well. These results reveal that methanolic extracts of mushroom species were free radical scavengers, acting possibly as primary antioxidants. The antioxidant activity of the mushroom extracts was concentration dependent, and all extracts exhibited strong antioxidant properties. Both lanthanous species extracts could terminate free radical chain reaction in vitro. Lanthanous carbonate exhibited more antioxidant activity compared to lanthanous hydrocarbon. In conclusion, we would like to conclude that today antioxidant properties of the plants have become of great interest due to their possible uses as natural additives to replace synthetic ones. The results of the present study shows that all extracts exhibit potent antioxidant activity, and methanolic extract exhibited high potency for free radical scavenging activity, which is highly related to the presence of hydroxyl groups in the phenolic compounds. Thus, present data suggests that methanolic extract can be used as a good source of natural antioxidants for health benefits, and further isolation of bioactive compounds is required for identifying the unknown compounds to establish a pharmacological properties. For explanation, I am thankful to the head department of botany, Nadian University, for the infrastructural facilities provided for present study. I am also thankful for the financial assistance funded by DBFund, Institute of Technology, Environment and Development, and National Institute of Chemistry of Environment, Forest, Climate Change, Government of India, to the University Grants Commission for providing recipes DRS1, DRS2, and DRS3, and Center for Biodiversity Studies with Department of Botany, Nadian University in India. Thank you.
Salutations to the committee. I am Khushi Jain. I have worked on the paper titled Mechanism of Plant-Derived Alkaloid Paclitaxel as an Antimitotic, along with Rohit Sankar as co-author and Dr. Pamela Jha as a guide and corresponding author. The category of my paper is Medicinal Botany and Phytochemistry. Without further ado, I would like to welcome you all to the paclitaxel mechanism or review. Taxa is a drug which is made by the wood of Pacific tree, Taxus brevifolia. and is used to treat breast, lung, and ovary cancer along with Kaposi sarcoma. Its capacity to stabilize microtubules and induce microtubule bundling is the basis for its biological activity. On a larger scale, the discovery of Taxol was the only outcome of NCI joint USDA's venture effort. Despite screening over 1,14,045 plant extracts over nearly 2 decades, Taxol was the only item the algorithm could create. It was also significant due to large sums of money spent on research. Taxol was developed with a $25 million investment from the US government. The FDA authorized Taxol to treat ovarian cancer in December 1992. Following further clinical trials, FDA approved Taxol to cure breast cancer in 1994. Then, in 1997, the FDA approved it for the cure of AIDS-related Kaposi sarcoma. Because of Taxol's success, additional organizations and businesses have joined together to increase Taxol research. Taxol has the potential to provide more life-saving treatment options when paired with other cancer treatments. Patients with metastatic pancreatic cancer were studied in the Metastatic Pancreatic Adenocarcinoma Clinical Trial (MPACT). Patients who received nab-paclitaxel, a kind of paclitaxel in combination with gemcitabine, lived longer than those who had gemcitabine alone, according to the researchers. Based on the findings of MPACT, the FDA authorized nab-paclitaxel along with gemcitabine for the patients with locally progressed pancreatic tumor in 2013. Therefore, Taxol provides cancer patients with better therapeutic efficacy, a longer life expectancy, and a higher standard of living. Because of the availability of a limited amount of unattached kinetochores, the therapy causes cells to enter mitosis. Stimulation of the mitotic checkpoint, the key cell cycle management method that functions during mitosis to avoid chromosomal missegregation, causes paclitaxel-induced mitotic arrest. The mitotic checkpoint prevents chromosomes from dividing until the mitotic spindle's two ends have formed solid links. Chromosomes begin mitosis as repeated bivalence of chromosomes of sister chromatids, and the outcome of this design is that every daughter cell will acquire a copy of each chromatid. Kinetochores build protein complexes on centromeric sections of DNA to connect chromatids to spindle microtubules, and the unattached kinetochores, which lack strong linkages to microtubules, stimulate a signaling cascade, which will inhibit the anaphase enhancing complex, which will in turn delay mitotic advancement. After 10-year follow-up period, researchers showed that adding PTX to doxorubicin plus cyclophosphamide significantly reduced the probability of recurrence and death in 3,121 node-positive postoperative patients. HER2 plus tumors were found in 65.2% of patients responding to taxanes, while HER2 tumors were reported in 35.5%. So, as a conclusion, PTX operates through a signaling pathway that is specific to HER2 plus cancers. PTX stimulates both the cancer suppressive protein P35 and CDK inhibitor P21WAF1. However, because cancer cells lack this tumor suppressor, the usual activation of P35 does not activate P21WAF. So, in BC cells, it increases antigen-presenting cell maturation by attaching to the toll-like receptor on the BC surface. In NK cells, higher levels of perforin, a critical effector protein in NK activity, were associated with increased cytotoxicity, pointing to the fact that PTX increased the NK cytotoxicity in a dose-dependent manner. By assisting in the construction of alpha and beta tubulin subunits, the building blocks of microtubules, the drug decreases the critical concentration of tubulin necessary for its assembly. Mitochondrial outer membrane permeabilization (MONP) is a characteristic of the pathway that results in the discharge of cytochrome C and other pro-apoptotic proteins in the cytoplasm. Capsaces are activated, which results in cell death. Capsaces are exclusively found downstream of mitochondria and are required by MTAs as apoptosis mediators.
Taxol-mediated apoptosis in MCF7 cells were not as efficient in eradicating apoptosis. Taxol-induced apoptosis was reduced in RAS-transformed RAB1 cells by dominant negative MEK. Like other drugs, paclitaxel has concentration-dependent consequences. The immediate drastic actions of increasing paclitaxel amounts on mitosis and cell death were previously assumed to be involved in its success in cancer, which is understandable. Nevertheless, fundamental scientists' inability to obtain patient samples was a significant lag in discovering that lower doses, which take longer to elicit cell death, are statistically important. Due to inconsistencies in its clinical use, it is critical to improve its qualities in relation to reducing cancer cell resistance to PTX, increasing its success in cancer therapy, and alleviating symptoms associated with standard PTX use. Though endophytic fungi have shown promise in Taxol synthesis, the biochemical process is yet unknown and the yields are still modest. Its significance in tumor immunotherapy should be further investigated. With this, I would like to conclude my presentation. Thank you each and everyone for taking all the time to go through our presentation, and special thanks to Khalsa College and Journal of Plant Science Research Committee for providing us with a platform to showcase our skills. Thank you very much.
Greetings everyone. My name is Krisha Ravi, and along with my co-author Aditi Nambiar, we have written a review paper titled Endophytic Fungi-Based Production of Taxol and Advancements in Clinical Studies. This was written under the guidance of Dr. Pamela Jha. I'll be presenting this paper today. What is Taxol? Taxol was extracted from the roots and bark of Taxus brevifolia in 1962 for the first time. It is to be noted that it is a very important and useful secondary metabolite. Now, the reason why it's important is because it is a highly efficient anti-neoplastic and antimitotic agent with low toxicity and has been extensively used for the treatment of many cancers such as uterine cancer, breast cancer, lung cancer, and is also effective in the treatment of Kaposi sarcoma, which is a deadly tumor usually seen in AIDS patients. I'd like to briefly talk about the preclinical developments of paclitaxel. It was in 1962 that the samples collected from Taxus brevifolia by botanists Arthur Barclay, from which in 1967 Mansukhani and Munro Wall were able to isolate and identify the active ingredient, namely taxol. Paclitaxel structure was published in 1971. National Cancer Institute (NCI) confirmed the anti-tumor efficiency in the mouse melanoma B16 model. Animal models showed activity against MX1 mammary, LX1 lung, and CX1 colon tumors. After these studies, paclitaxel was selected as a candidate for clinical development, and hence the clinical trials began in 1983. Paclitaxel is made up of an 8-member taxane ring and a 4-member oxytocin ring, which is connected by an ester side chain at position C30, and this ester side chain contributes to its anti-cancer properties. Paclitaxel is a diterpene alkaloid which acts on microtubules as its site of action. Paclitaxel promotes microtubule assembly and stabilizes microtubule polymers, preventing cell proliferation. A homochiral ester side chain is an active portion that binds to the microtubules, stabilizes tubulin bundles, and enhances the disassembly of microtubules in a GDP complex. Cell growth is inhibited as a result of the cell cycle stalling at the metaphase-anaphase boundary and the creation of an incomplete metaphase plate of chromosomes. Based on this, it is determined that the taxane ring and the ester side chain are required for activity. In our review paper, we have stated a detailed table summarizing the clinical developments of paclitaxel. As we know that everything comes with its own pros and cons, so does the taxol. You can see on the slide the 3 main problems with traditional extraction of taxol, which are: first, the production cost is expensive. Second, the method is environmentally non-sustainable, which leads to the third problem, that is, it has caused endangerment of taxus species. Limitations with respect to formulation of a drug. One major limitation of Taxol is its low solubility in water, which is just 0.1 microgram per ml, and its extreme hydrophobicity complicates its drug supply and makes it a difficult drug to formulate for human use. It is difficult to formulate drugs from active compounds or ingredients derived from medicinal plants, and the supply of these compounds can face challenges such as limited amount of phytocompounds, a constrained plant growth rate, confined localization of active pharmaceutical ingredients in specific organs, and preservation of natural resources. To address this issue, various formulations were made, which are discussed in depth in the review paper. To tackle the problem of low yield from taxa species and the endangerment of those species, researches have been done and are still underway to understand the mechanism of biosynthetic pathways, detection, and isolation of taxa from the taxa-producing endophytic fungi. About 200 endophytic fungi have been identified as taxa producers from more than 40 fungal genera. On the right side of the slide, you can see Metarhizium amnisoplai, which has the highest taxa yield concentration of 846.1 microgram per liter. Detection of taxol in the fungal extracts are carried out by advanced chromatographic and spectroscopic techniques like Hplc, NMR, Baldetrov, and other than these methods, PCR-based methods have been proven to be reliable methods for the screening and identification of taxa-producing fungi. Genomic mining is also used to identify the fungi associated with taxa-producing properties. Various methods to increase taxa production. Incorporation of host cells that have some distant phylogenetic relationships is a technique called protoplast fusion. And this can give us long-range hybridization of cells and expansion of the recombination range of genetic material. A mixture of mutational and protoplast fusion techniques is efficient in producing high taxa yield. Other scientific approaches like agrobacterium-mediated transformation, electroporation, and genome editing are also employed. The new advancements in genetic engineering techniques like selection of appropriate vectors, cloning of key enzymes have also improved the taxa yield enormously. At last, I'd like to conclude that PTX is one of the most efficient anti-cancer drugs to be ever developed from a phytochemical plant. The various phases of clinical trials conducted wherein combination of natural agents like Taxol with chemotherapy drugs in patients with benign or malignant tumors may lessen the toxicity risk produced by chemotherapy, making this a rather accessible and safer method for cancer control and management. One of the emerging methods for increasing targeted metabolite production is the genetic alteration of endophytic fungi. Given the current state of affairs seen in the global market, more efforts are being made to increase taxa production by endophytic fungi. And in future, large-scale industrial developments of taxa production by endophytic fungi will acquire an adequate amount of attention. These are some of the references. Thank you for your time.
Greetings to one and all. This is Ms. Lakshmi Sri Jingala, Assistant Professor from Department of Botany, Chief Norman Women's College, Delhi. The topic of my presentation is effect of storage condition on total phenol and sunlight content in dry stem powder of hemiparasite in Rhabdi falcata variety pubescent. MAC has been using plant as a source of healing agent. This healing property is determined by the type, quality, and concentration of phytochemical present in them. Pre and post treatment, as well as storage circumstances, can have impact on the potency and efficacy of this method. Delphi falcata, also known as mistletoe, belongs to agent like cooling, bitter, astringent, antibacterial, aphrodisiac, narcotic, and diuretic properties. Objective. The present study was carried out to assess the effect of storage period on the stability of total phenol and sunlight content in dried stem powder of Dendrophila falcata, variety pubescent growing on different hosts under room temperature. Plant sample Depichosa, people belonging to family Moaceae, is a native plant and also an unusual host of Dendrophila falcata. Method. Samples were collected. It was dried, shade dried, and in dark. The samples were 6 months in a year. The sample, the estimation of total phenol and total flavonoid content method by Singleton and Rosen, 1965. The total flavonoid was determined by aluminum chloride method by Wph in 1998. The statistical tool used was t-test and p-value was kept at 0.01. The statistical analysis was carried out by Excel Stat Pen software. Results. The samples were abbreviated as DFM, DFN and DFP. DFM for hemiparasite stem collected from Mangifera indica, that is mango. DFN, hemiparasite sample stem collected from neem, that is Melia azadera. DFP, sample hemiparasite sample collected from people, that is Ficus religiosa. Results of total phenol. Dendrocti pyrata, variety pubescent, collected from mango after 6 months of storage showed the total phenol content to be 91%, and after an year of storage it was found to be 81%, a significant decrease of 80.25%. A slight reduction was observed after 6 months of storage with the retention of approximately 96.6% of total phenol in DFN. After a year, it showed stability of 84.8%, that is 15.25% decrease. Whereas DFP showed 88.5% after a storage for 6 months, whereas after a year, it showed 71.15%, that is a decrease of 28.92%. The results of total flavonoid. The flavonoid content of DFM for 6 months old sample during storage showed retention of 85.9%. A year later, retention had decreased to 62.14%, a significant decrease of 38.8%. DFM showed 91.6% of total flavonoid after storage for 6 months, whereas after a year it showed 72.6%, that is a decrease of 27.4%. DFP showed 93.3% of flavonoid after storage for 6 months, whereas after a year it showed 77.6%, that is 22.8% was observed. The decrease in total phenol and flavonoid could be related to degradation of phenolic compounds and flavonoid due to storage, duration, and temperature. The results are found to be in agreement with the report of other authors like Rea et al. 2015 reported significant decrease of 50% in TPC and 65% of total flavonoid after storage for 4 days at 25 degrees centigrade was observed in different parts of Finlanthus nuclei, and he corroborated with duration and temperature, whereas Deltaro et al. In 2015 demonstrated approximately 97 and 95% stability of phenol and flavonoid, which stood at -20 degrees in amber bottles for 60 days of various parts of Anoplosus californica, and he assumed that it is an influence of light and temperature, and the degradation would be due to change in structure of polyphenols. Other authors also reported a similar kind of results and corroborated the results to oxidative degradation. Decrease in flavonoid can be due to oxidative stress, resulting in production of reactive oxygen species, either due to inappropriate storage temperature, or loss in individual phenolic and flavonoid compounds due to active enzyme system in the tissue. To summarize the result, the present study shows significant reduction in total phenol and flavonoid content. The rate of reduction of these phytochemicals was found to differ in same hemiparasite going on 3 distinct hosts. The stability of TPC was found to be more in DFM, whereas stability of total flavonoid was found to be more in DFP, that is hemiparasite collected from people. From the above result, it can be observed that flavonoids have decreased more compared to phenol after a year of storage. To conclude, the decrease in total phenol and flavonoid content observed in the present study could be due to deterioration or degradation in the TPC and THC value facilitated by storage temperature and time, along with a possible influence of their host. These are my references. Thank you so much. I thank my guide Dr. Geeta Menon from R.K. Kalreja College and Khalsa College for giving me this opportunity. Thank you so much.
Hi everyone,
I'm Lydia from the Department of Botany University. I'm a research scholar and I'll be presenting on the topic Faith and Food, a religious view on food waste markers, and my category is medicinal botany. So traditional food gastronomy, it reflects the interactions between the environment and the local peoples or the society that produces it, and it helps sustain the traditional food items, and it implements food sovereignty, and the food waste or the preparation and the consumption pattern of the food. It captures perceptive endowment and it strengthens the community affiliation that helps shape the cultural identity, personal goals of communities and communities in society. So the themes involved in this presentation involves lessons that were gleaned from the oral traditions from ethnic patterns of family ties, experiences, the religious beliefs, the food availability and the use of resources, the culinary skills and the ingredients used. So the gastronomy of the Nagas, it parallels the age-old traditions and practices of the past, and food and medicine were synonymous to our way of living. We Nagas, we were once known as warrior tribes, but now our bravery, it is marked in the unique cooking and eating food habits that our people produce and create under the nose of our neighbors. Why I mention the nose of our neighbors is because this part of our country, the northeastern part, we are known for our unique food habits that is culturally and socially very different from the rest part of our country. So the diverse forms of culinary preparations that our people produce, it interlinks the rich ancestral knowledge, traditional knowledge, and it reflects the versatile nature of the food, particularly implicit on the human health. So foods that to improve metabolism to foods cancer are consumed by our people to face the daily challenges of life. Christianity is the established religion of our state, and our social structures and practices are hugely burdened by our religious belief. And the religious context, it heavily influences the food choices that we make, as it creates an organic output of community living, creating the ties, cultural ties between at various levels between the individuals or the homes, or even between the various tribes that are found in our state. And this food choice also provides a unique camaraderie between the food that has been served, or that has been prepared, and which is going to be served to the individual person towards healing, as prayers are always said before serving the food or before the food is consumed. So this table, it represents the various foods that are used as medicinal agents against hypertension, diabetes, and cancer. So these are some plants that are used against the safe diseases. The next is the foods that are used as medicinal agents against headache, fever, cold and cough, too as well. So this type, the preparation is mostly curry or chutney. These are some examples of plants that are used for fighting against headaches and flu and stuff. So the next one is foods used as medicinal agents against digestive and stomach ailments. Preparation is mostly boiled or rice. So these are some example of plants. Then the last table is foods used as medicinal agents against various other diseases. Here, interesting example is the ginger and the fermented soybean. This is very popular among the Sumi tribe. We have 60 tribes in Nagaland, and Sumi tribe is one of the major tribes. And this particular chutney is also very famous when we travel, because it is said to have it is said to be effective against nausea and headache as well. So this is the figure. The figure B is the ginger and fermented soybean chutney. Then the black turtle bean is used against the mouth ulcer. Another common example is the use of the papaya leaves, which is grounded and crushed, or it is boiled, and it is effectively used against low blood platelets. So
Another community were woven by thread of religious occasions, festivity, and visitation by our neighbors, family, and relatives, and each occasion is celebrated by preparation of festive foods, and the various dishes that we creates a multiculturalism facet among the others, and it is represented by totemic or emblematic dishes that signify a particular tribe. So food connoisseurs, social identities, and central to food waste is the sharing of the common goal of healing, which seek to improve individuals' well-being, are the food culture in Latin. It has emerged to be a product of the centuries, strongly influenced by religion, while at the same time it strives to maintain its cultural identity. And one classic example of how religion had really, really impacted our culture is the abandoning of the use of alcoholic rice beverages, which were once an integral part of our culture, but now it is being replaced by tea or other herbal beverages. So these are the references, and thank you.
Good day to one and all present. I'm Marcel Chalumana with the scholar, Department of Methodology, one of the scholars of Partial Science, Tourism English. I'm going to present on the topic antimicrobial activity of mangrove endophytic fungal activity. Mangrove ecosystem has unique characteristics due to its high salinity and organic matter. And mangrove endophytes are known to produce wide variety of bioactive metabolites which possess antimicrobial activity. In recent years, it is being reported that marine-based microbes exhibit new bioactive compounds with various biological activities. Fungi have aroused much interest as they are an important natural source for the discovery of new bioactive compounds with potential biotechnological and pharmacological benefits. The objectives of this study are collection of sample and solutions, isolation of endophytic fungi, food compound extract, antibiotic extract, and Tlc analysis. Methodology and from the isolate crude was extracted, and the extracted crude was used collected from the collected samples were processed for surface sterilization. So the surface sterilization was done by standard methods, and then the explants were cut into small pieces, sterile aseptically, and then it is placed on photodextrose agar medium for the isolation of endophytic fungus. And this slide shows the growth of endophytic fungus from the leaves, bark, and flowers of the mango plant, and these are the pure isolates. The crude extract, the isolated seeds were inoculated into the photodextrose rod, and the crude extract of the medium was done or extracted with double volume of ethyl acetate in a separating funnel. Then the crude result extract was used for antibiogram studies. And this slide shows the antibiogram activity of the isolates against Candida species, Mucus species, and Aspergillus. And this slide shows the antibiogram activity of the isolates against the bacterial species, and this is Shigella, Salmonella, and Staphylococcus. And this graph shows the antimicrobial action of the isolates. So the isolates shows, some of the isolates shows much action against the given pathogen when it is compared to the commercial antibiotic fluorochemical. And this is the Tlc analysis of this isolate. And mangrove, conclusion of the study is, mangroves are the host to plant-associated microorganisms, including fungi. And this mangrove forest has a distinctive habitat adapting with marine and terrestrial environment. And this mangrove endophytic microorganisms have complex structure when it is compared to the other microorganisms.
since it is exposed to the extreme environment, which helps them to produce novel secondary metabolites. The current study concludes that the purified endophytic fungal extract acts as a promising antimicrobial agent. In future, this compounds will be purified and further studies will be done. Thank you. So now I thank my guide, Dr. Rajan, for giving me this opportunity and the organizers of this conference for providing us a wonderful opportunity. Thank you one and all.
Good day, everyone. I am Nagavini from Botany Department, Bangalore University, Bangalore. I am going to present a paper titled Metabolic Profiling of Medicinally Important Compounds, Beta-Cytosterol and Alpha-Amarin in Ergolaneta. Let me introduce the plant first. It's a medicinal plant which belongs to the family Amaranthaceae, or commonly known as Polpala. It is well known in Ayurvedic medicine for a lot of diseases, namely renal diseases, viral fever, cough, lithiasis, and also recorded as diuretic in Ayurvedic literature. And many people in ethnobotany regard the whole plant to be edible, as it gives protection against evil spirit and also known for good luck for hunters and safeguarding the well-being of widows. These reports, the various reports on its multiple uses, attracted our attention to work on this plant. So let me describe the plant taxonomically. It is a herb with a base hard as wood and branches erect or creeping. Leaves are alternate, duly tomentose, 1.3 to 2.5 centimeter long. Flowers are greenish white, minute, borne in axillary panicles. Fruits are greenish, round, compressed verticals. Seeds are kidney shaped and very small. The chemical constituents which attracted our attention are alpha-amarin, campesterol, beta-cytosterol, beta-cytosterol palmitate, and 4 flavonoid glycosides. So we are actually basically focusing on the profiling of the chemical compounds beta-cytosterol and alpha-amarin. So the material and methods used are Hplc and Tlc. So the plants were collected from the Department of Botany. We had maintained them in our botanical garden. And then the TLC test model goes like this. 2 grams of powder material was taken on 50 ml stoppered conical flask. 10 ml of methanol was added to it. The mixture was kept for overnight and to decanted the extract, which was later filtered and solvent was removed under reduced pressure. This process was repeated thrice, of course, to get comparative results. The extracts were concentrated, percentage yield was calculated, and they were then subjected to TLC profiling studies. TLC is done only as a qualitative test and not a quantitative test here for alpha-amarin. So we created a standard reference solution by dissolving 1 mg of alpha-amarin and in 1 ml of chloroform. So 5 ml of each test and reference solution was applied on a pre-coated silica gel 60 F254 TLC plate from Merck, and the plate was developed in the solvent system of N-hexane, chloroform, methanol in the ratio 2:7.5:0.5 to a distance of 8 centimeters. So visualization, the plates were sprayed with anisaldehyde and sulfuric acid reagent and heated at 105 degrees till the color bands appeared. They were observed under UV light at 254 nanometers. So the observations will be shown in the results slide. The HPLC analysis module. So we created mobile phase of water and acetonitrile 15:85. The column used was Phenomenux C18 Luna 5 micrometers 150 into 4.6 millimeters. Detection was done under 202 nanometers. Flow rate 2 ml per minute. And standard solution was prepared. 10 mg of beta-cytosterol was dissolved in 25 ml of methanol and heated on water bath for 15-20 minutes. The solution was made up to volume of 25 ml with methanol. Sample solution 100 mg of the plant extract was dissolved in 25 ml methanol and heated on water bath for 15-20 minutes. Solution was made up to the volume of 25 ml with methanol. So how do we run through the column? We are injecting 20 microliters of
the sample and the standard separately to the column, and then we calculate the area of the standard as compared to the sample and calculate the assay using the formula. So the formula is here as shown on the slide. So the results, coming to the results, HPLC analysis of beta-cytosterol. The wild plant is showing 0.021% as compared to the standard. The tissue culture plant is showing 0.0099%, which is equal, almost equal to the standard sample used. And herbal data, which is influenced by a chemical mutagen, is showing 0.043% of beta-cytosterol. As you can see, this figure A or the graph A represents the standard solution of beta-cytosterol with 8.490 content, whereas figure B represents the wild plant where beta-cytosterol is comparatively lesser, and we can find the in vitro sample of herbal data, which is showing similar to the standard procedure is 8.433. But whereas the in vitro herbal sample influenced by a chemical mutagen is showing a higher content in the figure 4, that is D. So when we discuss these results, so based on our results, there is a growth change in the normal and in vitro conditions. The normal in vitro conditions where you actually use the basic media fortified with growth hormones, it is showing 52.85 lesser than the beta-cytosterol content than the wild plant. This decrease can be attributed to the size of the leaves obtained from glycine plants. However, extracts of in vitro plants influenced under chemical mutagen is showing a significant increase of 104.76%, which is to be noted. Then we come to the Tlc profiling, which is a qualitative test. You can see the bands on the right side. A, B, C, D name. A is the tissue culture plant of herbal meta, normal conditions. Then B is wild plant. C is influenced by chemical mutagen, and the D is the standard. So among the three, you can see the bands on the screen available in all the four. Of course, the D is the standard. So when you compare A and D, that is tissue culture, and the last one, the standard, they show similar length of bands, whereas B is showing a little increase, which means the wild plant has more content of alpha-amarin compared to the standard, whereas C, the in vitro plant influenced by chemical mutagen, is showing a higher content of alpha-amarin with the bandwidth and darkness as shown in the band. So this was a qualitative analysis. So when you see the qualitative analysis as I've described again, the bands are visible in both reference and test solution tracks in all the three plant samples. So this is confirming the presence of this compound in all the three plant sets. However, the bandwidth of in vitro plants is thinner than the wild, and it is the plant which is influenced by chemical mutagen showing a wider and darker band than the wild plant. These length and depth differences in bands can be correlated with the increase or decrease of particular content, but a theoretical assumption cannot be proved unless and until a quantitative study is done. So based on the results obtained, here are the conclusions. So chemically influenced in vitro plant has shown considerable increase in the volume of secondary metabolites, namely beta-cytosterol and alpha-amarin. This increase can be enhanced, and this can be attributed to the mutagenesis. So decrease in beta-cytosterol or no changes in alpha-amarin is actually proving that there is no clonal propagation or no genetic or physiological or any other kind of changes in the in vitro grown plants. Further, the methods used to enhance secondary metabolites can be experimented on other plants to obtain varied results. Thank you.
The research title is Observation and Screening of Phytochemicals Inhibiting the Agaricus biscolus mushroom primacillus skin whitening activity by in silico approach using molecular doctrine. Skin whitening cosmetics is one category of advanced cosmetics and it decreases pigmentation, generally known as blots, freckle of the skin caused by the solar ultraviolet rays. So, tyrosinase, which has an active center formed by dinucleocopper, that is multifunctional enzyme, play an important role in the pigment production. The cause of the pigmentation and darkened skin color is due to the pigment called melanin, and this pigment is found in the cell or organelle called melanosome, which is found inside unique skin cells. The pigment melanin is formed by catalyzation of tyrosin in the presence of enzyme tyrosinase, where the hydroxylation of monophenols to orthodiphenols and then oxidation of orthodiphenols to orthoquinones generates the dopachrome. Then the dopachrome produces melanin through polymerization. So, due to critical role of tyrosinase in the melanogenesis, many researchers are trying to discover new tyrosinase inhibitor from both natural and synthetic. So, tyrosinase can be obtained from various sources like microbes, bacteria, fungi, plants, and animals. So, in addition, the amino acid sequence identity between human and mushroom tyrosinase is 23%. So, tyrosinase enzyme found in the agaricus by spores are chosen due to their high activity extracts without extensive purification. As the mushroom tyrosinase resembles the human tyrosinase, so therefore this enzyme is used for in silico studies for developing the skin whitening substances. The aim is to evaluate the inhibitory effect of phytochemicals on mushroom tyrosinase using in silico studies. The objective is to study the tyrosinase inhibitory activity to introduce the concept of multi-nature and inhibits the mushroom tyrosinase. These are the softwares which I have chosen for the research finding. For the retrieval, the three-dimensional crystallographic structure is taken from the agaricus bispores, that is mushroom tyrosinase. They were downloaded in the PDB format from the protein data bank, and then the protein was prepared for autodock through UCSF Chimera. The ligands, which were phytochemicals, they were retrieved from extensive literature survey, that is, they must possess the antioxidant properties, and and this downloaded in the SDF format from the PubChem database. The docking procedure aimed to identify the correct position of ligands in the binding pocket of a protein and to predict the affinity binding between the ligand and protein. So molecular docking was carried out by using pipettes. So for pharmacokinetics, bioavailability, and drug likeness prediction, ADME involves the absorption, distribution, metabolism, and excretion, which evaluate the pharmacokinetics of a drug molecule. So Swiss ADME was used to take account of this prediction. Toxicity, there could be a measure of any undesirable or adverse effects of chemical, and the toxicity prediction was carried out through the Protox tool, which is a virtual lab for the prediction of toxicity. Then, in order to get the idea of how ligands are bind to the protein in 2D and 3D format, the Discovery Studio was used. The modulation of melanogenesis is an important strategy for the treatment of excessive skin pigmentation. So, with the help of in silico studies, the possible phytochemical inhibitor which are involved in inhibiting the tyrosinase catalytic activity, they can be tested in a short period time for their efficiency and efficacy rather than working on the cell and animal tissue. So 213 compounds, they were isolated from various sources like Indian Plant Pharmacopoeia, PubMed, Journal of Phytochemistry, and etc. By performing the molecular docking, out of 213 compounds, 23 phytochemicals were docked and possess significantly for its ligand binding affinity, and they were shown maximum binding affinity as well, and therefore they were selected as the potent inhibitor. Through Swiss ADME, 23 natural phytochemicals having high binding affinity values, they were investigated for pharmacokinetic characteristic and the drug likeness prediction, where these 5 compounds were chosen because the radar plot is within the pink region and it can be considered safe to use as a drug. Then the Lipinski value is also 0, which indicating none of the requirement have been violated. Log S indicates the solubility, where the solution C is found to be having less value, which indicates more soluble. Log P indicates property of lipophilicity, where the infacting is having the lowest value. Kp is the skin permeability. The more negative value, then it will be less permeable. The synthetic accessibility indicates how easy it is to synthesize chemicals, where one value will indicate it is easy to get it and then indicates how to get it. Then toxicity prediction was carried out using Protox 2 for 23 phytochemicals, and through ADME interpretation, the 5 compounds were coming under the class 4, where the LD50 value should be in between 300 to 2000. And this shows the 3D interaction of ligand protein shows how ligands are bound to the protein and interacting with the amino acid residues. So delphatine, neodine, nimbandiol, as well as nimbanine and sembutane C, all of them are interacting well with the amino acid residues and hence they were binding the tyrosinas. Then in conclusion, by using computational tools, the potent tyrosinase inhibitor were discovered and evaluated. Through this data, this reduced the time and cost for developing novel pharmaceutics. Then by in silico method, molecular docking with the agaricus bisporus among 213 phytochemicals, 5 potent inhibitors were found, which are sembutane C and delphatine belonging to alkaloids, neolin, nimbanil, and nimbandiol belonging to other class of compounds. So this research work will be useful to medicinal chemists working on melanogenesis, especially on tyrosinase protein, to identify the novel inhibitors with drug-like properties. These are the references which I have taken for my research finding. Thank you.
Hello everyone, I'm Pawan and Punya, PhD scholar and co-author of the Scientific Topic. I would like to thank the organizing committee for doing this topic. Today I'll be giving a presentation on anti-chemical assay of chemicals happening in the development and people coming to medical. Those nebulensis is a very less detailed plant found in areas of northeastern part of our country, India. It has been reported to be found in pocket areas in northeastern part. The Taxa tribe Nabalan, India has been using the plant indigenously for treatment of rheumatoid arthritis. Array has been found to be associated with inflammation in the affected area on joints. This study now it is to confirm the effectiveness of C. nebulensis in treatments. Information compared with standard chemicals. Preparation of sample for tests. Now the needed samples were collected from the specified location in the beginning of the year, where it is mostly vegetative. The collected leaves were washed and dried executively selected on the vacuum evaporator and oven. Dried leaf samples were grinded to very fine powder. Couplet apparatus along with microwave was used to completely extract the physical compound by using boiler over methanol. Methanol was completely removed by pouring the bubbler. For anti-inflammatory assay, isolation of food extract will be good. 50, 100, 200, 400, 800 microgram per ml and a solution of Tlc per ml. Each 250 ml of PSA plus 1.5 PPA solution was added and added up to techniques were heated to 70% centigrade for 5 minutes, followed by a heat market. On taking the distilled water as a negative control, and Ultrasonic acid has a positive control temperature for setting at 15 nanometer. The good sample at 800 microgram per ml was taken to replicate the accurate percentage of triplicate means taken average of the they also exploded to check the regression rate. That assay it is performed as per the protocol used by the modification in the configuration. Concentration of 100 to 140 gram per ml for the third and 24 and 80 and 20 gram per ml for activity. For the solutions for these with 2.5 ml EPS and 2.5 ml protection rate diameter. It was intubated systemically followed by rapidly for use. PCA was added to all the full facilitation and 5 kg. Now, the 2.5 ml supernatant was taken and 2.5 ml the water for 4.5 for solution. Control of 1 ml is the water was allowed to stand for 10 minutes. Now, that's been situated and as usually, for example, was added for the in vitro anti-inflammatory assay, the regression value as per on the graph was calculated as 0.971. Now the percentage inhibition compared to acetylsalicylic acid at 800 microgram per ml was found to be 7.6%. Now here acetic acid is and because of and percentage what we learned from the essay is that medicinal plants are known to exhibit anti-inflammatory and antioxidant properties in the presence of flavonoid. It was found that denaturing the 7.2% of bacterial flavour. Now, as A is one of them, we can denaturing agent. Now, the regression value of 0.97 on the acidity of samples is 6.89% of acetic acid, fat acid. Now, further research can be proposed to explore bioactive hence, it is justified that T evidences DC extract is support.
Hello all, I am Pravit Warrior, pursuing PhD from Department of Chemistry, Ramnarayan Ruya, Autonomous College, Mumbai. I am pursuing PhD under the guidance of Professor Dr. Madhuri Badoli. The title of my presentation is GCMS-based phytochemical profiling of N-hexane extracts of Curcuma angustifolia rocks rhizomes. The category is medicinal botany and phytochemistry. Curcuma angustifolia rocks is commonly known as East Indian arrowroot and narrow-leaf turmeric. It is an indigenous herb native in India, found in wild in Bengal, northeast regions, and western coastal plains and hills. It is also found in abundance in Chhattisgarh, Orissa, and Andhra Pradesh, and highlands of Tamil Nadu and Kerala. It is from the family Zingiberaceae and the genus is Curcuma. The plant is highly nutritious, used for remedial purposes according to local healers. The rhizome is a source of starch. The current work provides information on phytoconstituents present in the N-hexane extract of the rhizome. This slide depicts the materials and methods used for the study. The rhizome was received from Jorhat, Assam, and was planted in a home garden. The plant was authenticated at CSIR NBRI Lucknow. The rhizomes were cleaned, cut, dried, and ground into a fine powder. For the GCMS studies and the extractive value, the rhizome powder was extracted using a Soxhlet extract apparatus in N-hexane. For the phytochemical screening, the rhizomes were reflexed with N-hexane. The GCMS instrument used was Shimadzu GCMS QP 2010 Plus model coupled with GCMS QP 2010 Ultra Mass Spectrometer. The solution software was GCMS solution software, and NIST 11 library database was used for the identification of components. This slide depicts the results and the discussions for the study. Out 27 components were detected in the N-hexane extract of Curcuma angustifolia rhox rhizome by GCMS. Out of the 27 compounds, 10 compounds had area percent greater than 3, and the remaining had less than 3. This slide here depicts the tabular form of all the components in the study. Those highlighted in the bold are with the major components. The slide here presents the tabular format for the major components along with the biological activity reported for each of the components. Also, the table shows here the phytochemical screening tests, wherein it is seen that terpenoids, steroids, phenolic compounds are present. Also, the extractive value in N-hexane is denoted here. From the presentation, I conclude that the data reported in this study shows the presence of phytoconstituents having biological activities in the N-hexane extract. This study provides the latest information on the extract of rhizome with N-hexane using Soxhlet apparatus and details the molecular weight structure of the phytoconstituents present in the extract. The current work is a small step in fingerprinting the chemical phytoconstituent of the rhizome. This study augments the prospects of using rhizome part of the Curcuma angustifolia rhox plant as a herbal drug in the future. These are the references used for my study. I would like to thank my guide, Professor Dr. Madhvi Badole, for guiding me during the study. I would also like to thank the head of the department, Chemistry, Professor Manish Hate, and thank the organizers of this conference, the Botany Department of Khalsa College, for providing me an opportunity to present in this conference. Thank you.
Respected chairperson and all the participants, we have done with first lot of 20 video presentations, and we will start with another slot of 17 videos in next 5 minutes as we are changing with the system. We will start with the next 5 minutes.
Yes, Shital.
Yes, thank you.
Yes, and thank you very much. There's some excellent presentation for last, almost 24, if I can remember properly. Around 25 presentations we have already seen, and some of the presentations are very good. Some are, of course, they need to brush out these presentations. But one problem is there, is that there is a sound problem. When you are recording, So your sounds may not be very good. So you should check first that whether the sound system is okay or not. First of all, another thing is that you must speak a little bit slowly, because in online presentation sometimes your voice is okay, and then sometimes no, you cannot understand. The audience cannot understand what you are saying. So these problems are there, but nevertheless the presentation is good, and some of the papers are very good, and some of the topics that have the speakers have covered, it's very good. And the organizers, and of course me and my co-chair Sanjeev, we have evaluated your presentation, each and every presentation, and in due course of time we'll let you know whose presentation is the best in this lot. Of course we'll go to the second lot. Yes.
Arnabda, some more presentations are there in this session.
I know. In the second half is there. Presentation is there. I'll be there. I probably will be starting within 5-10 minutes, I believe. Gopal?
Yes,
in 5 minutes, sir. Yes.
Okay. I'm chatting a little bit with the presenters. Most of the presenters must be present over here. But I must say it's quite encouraging, and many of them have done very good. Thank you.
Yeah.
Yes, Sanjeev.
This session is, I think, whatever we have seen is a successful session yet. 20 presentation is there. I have seen lots of area they have covered, from traditional knowledge to scientific validations, as well as modern approach, and that current trends of isolating or screening some effective substances with in silico model. Everything they have covered till now, from different source microbes as well as seaweeds or whatever different study materials already covered. In that point of view, it is very till now whatever it's very good session. Okay, thank you. I think we can wait for the next session.
So with your kind permission, we are starting with a next presentation.
A very good afternoon to the chairperson, all the dignitaries and participants present. Myself, Priyank Gowri, a research scholar from GN Khalsa College, with Dr. Manjiri Bhavet, Assistant Professor Botany, GN Khalsa College, would like to present our work, Studies on Flavonoid of Melitharia zapote fruit, under the theme Medicinal Botany and Phytochemistry. The content of the presentation contains introduction, review of literature, outcome from the review of literature, objectives, materials and methods, observations and results, future scope of study, acknowledgement and bibliography. So let us start with the introduction. The first question that arises is, what are flavonoids and what is its importance? Flavonoids are the most common group of polyphenolic compounds in the human diet and are found ubiquitously in plants. Polyphenols appear to be important metabolic modulators by virtue of their ability to influence several cellular pathways. Flavonoids, a subclass of polyphenols, are a group of phytochemicals that are among most potent and abundant antioxidants in our diet. Now, in addition to the internal antioxidant system which already exists in the human body, an external source of antioxidants, especially the naturally occurring ones, would be of great importance in combating various diseases. Now, according to the difference in the functional group and the relative position of the 15 carbon skeleton, flavonoids are classified into several subgroups. Although more than 4,000 flavonoids have been identified, several appear to be important components of many fruits and vegetables. So now let us talk about the plant which is used in the study. We have been endowed by nature with a marvelous flora and fauna which has modified our life. Manilcara zapodi, commonly known as Sapodilla chiku or sapota, one of the wonders of nature, belongs to the family of Sapotaceae. Chiku fruit holds great nutritional value because of good source of sugar varying from 12 to 14%. The plant has promising medicinal and nutritional properties. Now, somehow, directly or indirectly, presence of phytochemicals, especially flavonoids, have a great impact on medicinal and nutritional properties of a plant. Thus, this investigation aims to study the flavonoid profile in the fruit of Manilcara sapoti. One must be wondering, why did I choose this topic? So I stay at a place known as Anu, which is known for its chiku plantation. So I had an interest to study about it and also an emotional connect. Then I found enough research material to refer, and it was quite achievable and time bound with my academics. So first, the review of literature was done. Various papers were studied to check the phytochemical content of different plant parts, especially flavonoids. The plant also showed antimicrobial, antifungal, antibacterial, anticancerous, and other different properties. So now what was the outcome from review of literature? Although reports show presence of different ethnobotical properties, antioxidant properties, and presence of polyphenols, flavonoids in the different plant parts of Manilcara sapoti, the work on flavonoid content of the fruit was scanty or was not found. With all the benefits and popularity of chiku fruit for having its own flavor, study of its phytochemical content, especially flavonoids, is of utmost importance. Objectives. So the first objective being to extract flavonoids from chiku fruit with and without peel, and the second objective was to study flavonoid profile using HPLC. Let's move on to materials and methods. The collection of sample was done from Nagpur, Maharashtra. The chiku fruit was dried and made into powder by using sun drying method. The profile study of flavonoids was done using HPLC from Anglo, Mumbai. Initially, preliminary tests were conducted to confirm the presence of flavonoid in the sample. Later on, for HPLC studies, the sample were named as follows: Manilcara sapoti fruit with peel, Manilcara sapoti fruit without peel. The extraction was done using ethyl acetate bisonication method. The stationary phase used was 10 by 10 Tlc silica gel plate. The mobile phase, ethyl acetate, methyl ethyl ketone, formic acid, water in the ratio 5:4:1:1 gave best results. Derivatizing agents were used, namely natural product A, which is flavonoid specific, and the second one used was 10% methanolic sulfuric acid, which is an universal reagent. The sample were loaded in the following method in the tracks 1 to 7 with increasing concentrations. Observation. The complete procedure of HPLC analysis for the sample was done, and we found after development 4 bands were seen in both with pale and without pale samples. After derivatizing with natural product A, we saw intensified 3 bands in both with pale and without pale. After derivatizing with 10% methanolic sulfuric acid, we found in total 7 bands. All the pictures were taken in 366 nanometer wavelength. Image under 366 nanometer after derivatizing with 10% methanolic sulfuric acid reagent showed the bands at the following RS. With peel and without peel, both the samples showed in all 7 bands of different colors and different intensities. So let's see the result of the analysis conducted. In all, 7 bands and different RF of different colors and different intensity can be seen after derivatization. This proves that a fruit of Manilcara sapoti with peel and without peel is rich in flavonoids. The future scope of study is optimization studies for extraction of flavonoid, identification of isolated flavonoids, and nutraceutically important natural product development using chiku fruit. Acknowledgement. The authors are grateful to Principal G.N. Khalsa College, Head of the Department of Botany, G.N. Khalsa College, for their kind support. The acknowledgement is due to the Director and Chrome Private Limited for facilitating the research work at their premises with generous support of their analysts for HPLC analysis. Following research papers were referred for this study. And now thank you for your patient listening, and your questions are most welcome.
Good day to all the respected teachers and everyone present. My name is Rida Sharumbai, a research scholar in the Department of Biochemistry, Nehu, Shillong. The topic for my presentation is in vitro and in vivo antioxidant potential of Gentiana porphyria bloom. Oxidative stress and imbalance between the pro-oxidants and antioxidants results in molecular and cellular damage that leads to various diseases such as diabetes, cardiovascular diseases, and etc. Hence, increasing antioxidants level is of great importance because antioxidants reduce oxidative stress by controlling autooxidation of free radicals. Medicinal plants are best known for naturally occurring antioxidants that can scavenge free radicals. Therefore, studies on their antioxidant potential is of great interest. A selected plant, Gentiana porphyria, is found and collected from here. It is locally known as Yangao in Jante Hills and Jamyang in Khasi Hills. It has been traditionally used for many years for its beneficial properties in lowering blood pressure. This is the picture of the whole plant of Gentiana porphyria bloom. These are the various methods used for preparation of extracts. Leaves, stems, and flowers were separated, shaped, dried, grinded, and extracted in vitro according to the method of Alvaro. For in vitro study, Wph, ABTS, phospholipidem, and FRAP assay were done. For in vivo studies, the activity of superoxide dismutase and catalase enzyme were determined in liver and pancreas of different groups of mice. And now we come to the results. Number 1, Wph scavenging activity. As we can see from the graph, with increase in concentration, the percentage of inhibition also increases. And the IC50 of methanolic leaf is less than methanolic flower and methanolic stem as compared to the ascorbic used as standard. Less IC50 indicates high scavenging activity, hence methanolic leaf has high scavenging activity than methanolic flower and methanolic stem. The same with ABTS scavenging activity. As we can see from the graph, with increase in concentration, the percentage of inhibition also increases, and the IC50 of methanolic leaf for ABTS is less than methanolic flower and methanolic stem as compared to the standard, which indicates that methanolic is high ABTS scavenging activity as compared to the flower and stem. In phospholipidem assay also, the reduction potential of molybdenum 6 to molybdenum 5 is more in methanolic leaves than methanolic flowers and methanolic stems as compared to the ascorbic acid used as standard. With ferric reducing antioxidant power assay, the reduction potential of ferric 2,4,6 tripyridine S triacin to ferrous 2,4,6 tripyridine S triacin is also high in methanolic leaf than methanolic flower and methanolic stem as compared to the ascorbic acid used as for in vivo studies. The superoxide dismutase and catalase enzyme activity were determined, and as seen from the graph, the enzyme activity is significantly decreased in diabetic control of both liver and pancreas as compared to the normal control. However, the enzyme activity is only increased in diabetic mice treated with ascorbic acid and diabetic mice treated with 250 mg of methanolic leaves extract as compared to the diabetic control. Hence, in conclusion, through all these findings, we can conclude that Gentiana porphyria methanolic leaves extract have a higher antioxidative potential. Therefore, Gentiana porphyria could be a potential source of natural antioxidants, having great importance in the prevention and treatment of stress-related degenerative diseases. Thank you.
Hello everyone, my name is Robin P. Matthew and I am going to present on the topic in silico study of anti-diabetic effect of cinnamon meal. This work is co-authored by Ria P. Matthew. It's a computational biology work which is associated with the phytochemicals of cinnamon verum and the disease diabetes mellitus. This is the abstract of the work. The figure indicates or shows the plant. The hypothesis of our work is phytochemicals of cinnamon verum show anti-diabetic effect, and some of these phytochemicals can be used as a drug to control blood sugar levels in diabetes patients. The materials and methods are shown in the table. We use phytochemical collection screening using IMPACT, a comprehensive online database. We did target acquisition for drug and disease-related effects using BizGeneNet electronic database. We did construction of drug, active ingredients, and disease target networks by Swiss Drug Design Database, KEGG, and BizGeneNet database, which was visualized in Cytoscape version 3.9.1. We did protein-protein interaction network construction and food gene screening by string database. We also did a enrichment analysis by the data which was obtained from Spring database, we did homology modeling of the set protein targets in Swiss model, and the quality of the protein models were checked using all probability and cumin disco. Before molecular docking, we did protein and ligand preparation in different softwares like SPDB and Avogadro, respectively. We also did protein molecular docking in the Swiss doc. This is the first image which is generated. This is called as the phytochemicals of the cinnamon venom, and it's a network analysis of all the targets. We then segregated to make it simple for the phytochemicals of cinnamon venom with the gene protein targets related to diabetes mellitus. So the green ones are the phytochemicals of the cinnamon venom, and the blue ones are the genes or protein targets associated with the disease diabetes mellitus. Then we impose these data into the string database to find out if there is a connection between them. We found that except live, BCHE and GCGR, rest all the proteins are associated, or the signaling pathways are associated with each other. The colored ones indicate those 8 key proteins which are associated with very important pathways associated with diabetes mellitus. The enriched cake pathway shows you the data which gives you cross confirmation of the same. Then we did a molecular docking of all the said proteins, key proteins, with the ligands, out of which 3 important dock poses were found. We found that the highest scores were attained by certain protein docking poses of minus 6.88 as in case of AKR1B1 and cinnamide alcohol, minus 7.11 as in case of AKR1B1 and methyl cinnamate, and minus 6.72 as in case of MAPK8 and cinnamide acetate, with major interactions which are shown in the lower figure as in over here, with distances of less than 5 angstroms. All these molecular docks are composed in a single image. These are the RMSD values. So what we did is we superimposed the docked proteins with the energy minimized crystallographic key protein targets, and we found out what the root mean square deviations for them are. As you can see, the root mean square deviations fall under a range of 1 to 3 angstroms, which is considered to be a good RMSD value. And the final conclusion of the said work says that the present study investigated gives you effective active ingredients which are found in cinnamon venom and their molecular mechanisms to study antidiabetic effects. The active ingredients of cinnamon venom have an antidiabetic effect which includes cinnamide alcohol, methyl cinnamate, and cinnamide acetate, which is considered to be the topmost. And out of the 25 gene targets, we have AKR1B1 and MAPK8, which were considered for the dog doses. The signaling pathways in the treatment of diabetes mellitus included type 2 diabetes mellitus signaling pathway, insulin signaling, insulin resistant signaling pathway, age-led signaling pathway. And further, the results of this study shows or provides a new way to study the mechanism of cinnamon venom bioactive in the treatment of diabetes mellitus. These are the said references which we have referred. Thank you for a patient listening.
My name is Roshan Rajesh Paul. My topic is related to screening of toxic agrochemicals and studying the impact on nuclear receptor through in silico studies. First, we are going to see the introduction. Agriculture practice first began around 10,000 years ago. Eventually, agriculture became a lifestyle as various food crops were cultivated in different parts of the world. Today, with the advancement in agricultural science, still damage from the pest and disease-causing organisms help reduce the agricultural productivity. So there is a great need to find out the ways to overcome this problem caused by the pest, which eventually lead to the discovery of insecticides. Insecticides are not only able to control the pest but also able to increase the economy by increasing the food production. But with the excessive use of these insecticides, various harmful effects have been seen on the humans. In this study, we are going to see the effect of insecticides on the nuclear receptors with the help of in silico approach. In silico approach, various computational tools were used for the analysis. There are various methods involved in the in silico approach. We are going to study one by one. First is the protein trapping method. The targeted protein molecule was a required method. Protein determined and analyzed under discovery study of visualizer and carbon, and the protein was prepared for the doctor. These are the correct protein used for the analysis. Next, we move towards the selection of ligand. The ligand are the insecticides which affect the human nuclear receptors. which are retrieved from the PubChem database into the SDF format, which are then converted from SDF to PDB-UT format for further docking. Molecular docking was performed using this ligand structure and with the protein by Vena Wizard 2 Vena Pyrex software in order to find the best binding affinity with a targeted protein. Next is the bioavailability. Bioavailability of this ligand was analyzed using space advanced software, and the oxidative prediction was done by using Advent Lab 2.0. The thereby analysis of oxidative prediction was a neurological or central nervous system was done using blood-brain barrier 2. Various docking results was done. We will going to see one by one each result of it. First, we are going to see the estrogen receptor. Estrogen receptor belongs to the family of nuclear receptor, which are located in the hypothalamus and mammary glands. The function, the potential immunosuppressor inhibits the proliferation of cell as well as opposes the ER alpha action in the reproductive tissue. 77 agrochemicals were selected, out of which top 9 ligands were selected as they should good binding affinity with all the receptors, not only with the estrogen receptors. Highest binding affinity was exhibited by clothianafine and flotinoxorine with the highest docking score, and the lowest binding affinity was exhibited by alpha cytomethylene. So these are the graphical representation of the result and the 2D and the 3D structure of it. Next, we move towards the talking of androgen receptor. So receptor is a nuclear receptor located in the X chromosome of the male human species. Its function is to conserve and regulate the development and growth of the prostate. The range of binding affinity of this compound was between -6.4 to -9.9. Pyridine shows the highest binding affinity of -9.9 kilograms per mole, whereas alpha cypermethrin shows the lowest binding affinity of -6.2. These are the graphical representation in the 3D and the 2D structure of it. Talking of thyroid hormone receptor also belongs to the family of nuclear receptor. They are present in the nucleus of most of this tissue. Function is to control transformation and regulate development, growth, metabolism, and cardiac function. Range binding affinity of this compound is between -6.8 kcal per mole to -10.6 kcal per mole. Out of the selected 9 ligands based on the binding affinity, highest binding affinity for thyroid hormone receptor was exhibited by hexathioxox, while the least binding affinity was exhibited by alpha cypermethrin, respectively. These are the graphical representation of the 3D and the 2D structure of it. Next, we move towards the docking of progesterone receptor is a protein located in the female reproductive tissue. This function is to regulate the target gene expression in response to hormonal stimulus. Binding affinity ranging from -7.6 kcal per mole to -10.3 kcal per mole. Out of the selected 9 ligands based on the binding affinity, the highest binding affinity for the progesterone receptor was exhibited by prolifactone and fluoxetine, while the least binding affinity was exhibited by transferrin and alpha cypermethrin respectively. These are the graphical representation of the 2D and the 3D structure of the docking result. Similarly, the other receptors docking were performed and the result were concluded on the basis of this. And then we move towards the conclusion that these ligands which we are using, these 9 ligands which we get, are the most harmful to be used as a in agrochemicals and regular practice, and they should be removed from the practice. These are the bibliography. Thank you.
Respected organizers, speakers, and all the participants, I'm Rokunya Kocho, a research scholar from Nagaland University, Lumami. The title of my presentation is Use of Medicinal Herbs by the Indigenous Tribes of Nagaland, Northeast India. Category is Medicinal Botany. So, since time immemorial, people have been dependent on plants with therapeutic properties, and medicinal plants are not only considered as a rich bioresource, but it also plays a very important role in the development of human culture. And studies also reveals that about 80% of the world's population still depends on traditional medicine over the modern system of medicine. Northeast India is blessed with high medicinal plant diversity due to its variance in topography and physiognomy, and it also supports 50% of the India's biodiversity. Nagaland, being a part of the Indo-Burma mega biodiversity hotspot, harbors a huge diversity of medicinal plant species and also a number of plants which are of economic importance. The knowledge of practicing the use of medicinal plants is very much respected, and it forms a fundamental part of the social, cultural, and economic life of the Naga people. But since this valuable knowledge and ways on using medicinal plants is being passed down from generation orally and through traditional practices. So in the process, many detailed information is lost. So therefore, the current study was conducted to document the use of medicinal herbs used by the indigenous people in Nagaland. Nagaland, a mountain state, lies in the northeastern part of India, inhabited by 16 major tribes, and all these tribes have their own traditional method of preparing medicines using different plants. So field survey was conducted in different districts in Nagaland, and the first-hand information was collected during the field survey from the local people, elders, villagers, and the local healers on the uses of the medicinal plants, their local name, and plant parts used for treating various diseases. So coming to the results and discussion, in the present study, 118 medicinal herbs used by the indigenous people were documented, and family Asteraceae was reported with the largest number of species. The plant parts are mostly used by the leaves, shoots, roots, rhizomes, seeds, flowers, and the fruits. And traditionally, the mode of preparing this medicines is by using either fresh or dried plant materials, depending on the type of treatment and also the ailments. And most of the plants are processed by macerating, grinding, or decoction made with other herbs, cooked with local aromatic rice, and also mixed with other spices and condiments. So this is a table showing the list of medicinal herbs, along with the scientific and vernacular names, the family, plant parts, use, the preparation, and its purpose. Due to time constraint, I won't be explaining everything. So this is Gynora bicolor. The vernacular name is called Ligeno. The plant parts used are the aerial parts and the young leaves. Mode of preparation: the young leaves are boiled and taken with rice, or it is also cooked with other spices and condiments. Uses for constipation, diabetes, post-labor recovery, migraines, and gastrointestinal problems. This is Hutunia cordata. The vernacular name is called Gatha. Here the whole plant is used. Mode of preparation is it is eaten raw, or a fresh plant is also mixed with fermented soybean, and the uses are: it is used for detoxification, hypertension. It also improves appetite, and for heart problem as well. Plantago major, the vernacular name is called Gappa. Plant parts used are the whole plant. The mode of preparation, it is boiled and eaten with other vegetables. Its uses: it is used for treating stomach congestion, and the plant paste is also applied on cuts and burns. This is Passiflora edulis. The vernacular name is called bell. The plant parts used are the leaves and the fruits. Mode of preparation: it is consumed either steamed, boiled, or fried, and fruits are eaten fresh or fermented. Uses: it is used for treatment of asthma. It is used as a liver tonic for heart problems. The leaf paste is also applied for snake bite, and it also maintains high blood pressure. Polygonal mole, vernacular name is called gaje. The plant parts used are the leaves and the young stem. Mode of preparation is the fresh leaf paste is applied on infected skin. The young leaves and stems are eaten raw. The leaves are also cooked with rice and made into porridge, as it is shown in the picture. And its uses: it is anti-inflammatory, used for the treatment of diarrhea, for skin rashes. It also maintains high blood pressure and for gastrointestinal problems. We mentioned Fortigan. The vinegar name is called Dunia. Here the whole plant is used. The mode of preparation, it is consumed fresh or cooked with other vegetables as well. It is used for the treatment of earache, deworming, and it also improves appetite. So in conclusion, it is evident from the study that medicinal plants play a very vital role in aiding to the health of indigenous people in this region. And many of the people in the studied area still depends on medicinal plants for their daily healthcare routine over the modern system of medicines. But over the years, this system may also deteriorate due to developmental activities, deforestation, overexploitation of medicinal plants, and also due to lack of interest. Therefore, necessary steps should be taken up to encourage the upcoming generation, and also proper documentation should be done for future references. Several conservation techniques like in situ conservation in home gardens and on-farm cultivation should also be encouraged for socio-economic and sustainable growth. And lastly, scientific validation of these plants should also be done to attain global health issues. Several references. Thank you.
No elegance is possible without perfume, and so elegant is our research article, that is formulation of oil-based alcohol-free perfumes and deodorant using herbal sources. Myself, Ms. Sampada Patil from Department of 5-year Integrated Course in Bioanalytical Sciences of Guru Nanak Khalsa College, Matunga, and the category for which we are applying is phytochemical. Introduction. The Latin word perfumes, which means through smoke. But nowadays, the perfume which we use is a creative blend in alcoholic solution. This alcohol is mainly ethanol and its concentration varies from 50 to 95%. Another alternative to perfume is deodorant, which serves to eliminate the body odor and also act as a sweat controller that called as antiperspirant. And both of these contains the synthetic chemical. The synthetic chemicals used in perfumery is linked to cause allergic reaction. Sometimes the carcinogenic agents associated with it lead to have reproductive toxicity also. So coming to a natural solution to it is essential oils. As we all know, essential oils having the aromatic nature, antimicrobial nature. Today the consumer wants something that is natural and organic based. Aim was to develop a freshness inducing deodorant and perfume, and which we further step into. First was extraction of essential oils, then to check its purity, and to develop the perfume as well as deodorant, and to look for the market, what market need is and what customers want from us. For which materials and methods, we go through many research articles in order to get what extraction method we should use, what essential oils should be considered for making our perfumes, and we came up with several. So the selected sources was our lemon, aniseed, and lemongrass. The extraction method which we used was hydrodistillation and solvent-solvent extraction. In solvent-solvent extraction, we moved towards soda wax, so that the solvent which we used and we recycled and reused for the next batch. The analysis of the essential oil was gas chromatography, in which we take control as a standard marketed essential oils. Further which we move for the formulation of our perfumes as well as deodorants. These four perfumes and deodorant were then subjected for physical tests, in which we do proximate analysis and solubility. In sensory evaluation, we do texture for deodorant, viscosity for our perfume, and smell for both of them. In order to reduce the bodily odor, we check for controlling the microbial flora which causes the odor, bodily odor. So for that, we collected samples from oily skin type, dry skin type, and combination of both, and we do well diffusion method for perfume as well as deodorant. In safety test, we do both, that is repeat open application test and patch test. In survey reports, we go for how our product appears, how its appearance is, how long it lasts, what smoothing and moisturizing effect is this, and what customer want us to change in our product. Coming to the result and discussion part, after we have completed all our steps, it was found that the retention type of our standard as well as our extracted oil in gas chromatography was same, showing its purity. Then the microbial analysis which we have done shows slight zone of inhibition for all the deodorant as well as the perfume. In safety test, there was no redness, irritation, or allergy notice, stating its skin-friendly nature. The deodorant was easily soluble in chloroform, having a pH of 7, which makes justifying that it's neutral in nature. In proximate analysis, the whole compound was charred and there was no residue left. giving us the statement that is completely organic in nature. In survey report, we found out that the customer was more attracted towards what we have formulated, and and the deodorant as well as perfume were up to the mark what the customers nowadays. These were the chromatographic graphs which we have got for lemongrass, aniseeds, and lemon, having our peak of interest. Into the conclusion part, we can state that efficient extraction of the essential oils, that is from lemon, aniseeds, lemongrass, was done. The product which we developed was safe. Throughout the production, we keep in mind that lesser waste should be generated. Whole product, whether it is perfume or deodorant, or organic in nature, and serve its purpose that it inhibits the odor and give a long-lasting fragrance. The perfume as well as deodorant was safe for use, having no side effects in it, and the quality of the product satisfies the customer deeply. Coming to our future scopes, we are taking our oil-based perfume a little step ahead and formulating water-based oil microemulsion perfume using Cobb's method. After making this microemulsion perfume, we are subjecting it for the particle size analysis by PET, and we will try to reduce the size of the particle as much as possible. Then we will do bioautography so that we can specify what exactly compound leads to the antimicrobial activity. Further, the packaging and the designing of the product is also important, along with the fabric, the strong base of knowledge which we get from these references. Thank you for giving us a chance to present our work on such a big platform.
Welcome to all. Myself, Srinivadi, a student from Department of Physics and Biomedical Sciences from Guru Nanak Khalsa College of Arts, Science and Commerce. The topic of my paper is molecular docking studies of selected phytochemicals as a potent inhibitor for Parkinson's disease. Approach to anti-Parkinson's drug. The topic focuses on Parkinson's disease, which is a neurodegenerative condition where the person suffers because of neuronal death caused by the depleted dopamine level, which is a neurotransmitter which is responsible for the normal functioning of the brain in SM part, which is the central nigra part of the neuron. The cell death in the SM dopaminergic neurons results in striatal dopamine deficiency. The condition is marked by tremor, stiffness, and bradykinesia. Next is the neurodegeneration pathway of the cycle, which is due to the factors like alpha-synuclein aggregation, abnormal protein clearance, mitochondrial dysfunction, oxidative stress, which is ROS, and they have a role in the genesis and the progression of the PD. The biomarkers for the PD which are selected for the study are COMT, which is catechol O-methyl transferase, MAO-B, monoamine B, PINK1, ketone-induced putative kinase 1, E3 parking, E3 ubiquitin ligase, base 1, which is beta-amyloid precursor protein cleaving enzyme 1, and alpha-synuclein. Biomarkers or the proteins selected in this study for Parkinson's are studied because they have some mutations in the structure which either suppresses them or overexpresses, hampering the normal functioning, leading to the factors of neurodegeneration. Next, why must we use phytochemicals if there are previous drugs present? The answer to this is that conventional L-dopa therapy is widely used for Parkinson's disease. There are several limitations for this therapy. It is associated with troublesome side effects, which is common in the synthetic drug, and it does not halt the disease progression. It suppresses the symptoms, and later the patient suffers from resistance to L-dopa. To prevent this from happening, the phytochemicals have become widely studied area as a therapeutic for various diseases, which includes neurodegenerative diseases like Parkinson's, minimize and antioxidant properties are studied. The dietary polyphenols, triterpenoids, and molecular classes of therapeutic benefits in numerous models of neurodegeneration, which helps in neuroprotection. The humoral antioxidant activity is manifested by the ability to inhibit reactive oxygen species, which is ROS, which is effective in preventing the oxidative stress reduced apoptosis. Apoptosis means the cell death. For molecular docking, the database used are PDB, a protein structure database from where the 3D structures were retrieved. PubChem, a database from where the phytochemicals were retrieved, and the software used are Autodoc tools, which is an energy minimization tool, Pyrex, which is a molecular docking tool, Discovery Studio Visualizer, which is a tool for visualizing the protein ligand complexes. Next, the protein preparation step, energy minimization of protein.
is important because it helps in the formation of stable protein ligand complexes, and the minimization was carried out in Autodoc for the 3D structures of CONT, MAOB, alpha-synuclein, BACE1, PINK1, and Parkin. Next, the ligand preparation. The ligand showing the neuroprotective and antioxidant properties were selected. The 3D optimization of the ligand structure was done using OpenRabble, a tool in the Pyrex. The ligands selected are in table number 1. The docking studies were conducted by Pyrex. The search was extended over the whole receptor protein, which is also called as giant docking. Evaluation was done with respect to the binding energy. These are the phytochemicals which were considered for the study, which are 38 in number and were differentiated according to the classes like alkaloid, coumarins, phenolics, and terpenoids. Next, the results and discussion. 38 phytochemicals selected from the docking against 6 protein targets. The results obtained were binding affinities of 228 complexes. Protein ligand binding affinities of 228 complexes were in the range of -4 to -10.4 kilocal per mole. From that complex showing binding affinity greater than -10 was selected best because higher the affinity, best is the complex interaction, which means more negative the binding energy, the more stable is the protein complex. And the table here shows the asparaginine shows -10.4 binding energy with MAO-B, bicuculline of -10.1 binding energy with PINK1, and glycine with -10 binding energy with MAO-B. And these are the interactions of bicuculline with PINK1 protein, which is indicated in figure 1, and the figure 2 indicates the interaction of asparaginine with MAO-B protein, visualized in Discovery Studio Visualizer. Conclusion. From the results, the top 3 phytochemicals, bicuculline from Dicentra cucullaria, asparaginine from Cordia celestina, glycine from Passiflora caerulea were proven to be best in the docking studies. Therefore, bicuculline, asparaginine, and glycine may be helpful in the treatment of Parkinson's disease. However, it is suggested that further in vitro and in vivo assays for toxicology, pharmacology, and bioavailability studies of these phytochemicals have to be validated for the present predictions. These are some references, and thank you.
Good afternoon, everyone. My topic for the presentation is phytochemical profiling and GCMS analysis of N-hexane extract of Lavandula bipinnata leaves. India is a nation blessed with the most diverse traditional medicine culture in the world. It is mostly referred as botanical garden of the world. The World Health Organization supports and recommends the use of traditional medicine provided they are proven to be effective and safe for consumption. In the last few decades, use of herbal drugs has been increased due to their easy availability, therapeutic potential, lesser side effects, and cost. Lavandula genus is an important member of family Lamiaceae or Labidae. It is also called as mint family of flowering plants. These species are mainly grown for their essential oils that can be used in perfume, cosmetics, also in aromatherapy. The most common species which are proven to have medicinal value are Lavandula dente, Lavandula augustifolia, Lavandula dufarensis. However, Lavandula bipinnata is less exploited with respect to its medicinal properties. Lavandula bipinnata is a slender erect herbaceous, which is approximately 40 to 100 centimeters in height. It is found in central and south India. It is useful in anxiety, headaches, depression, as a breath freshener and mouthwash. The paste of roots is used for bites of poisonous animals. Paste of leaf is applied on decay to reduce pain. It is also used in combination with other herbs to treat rheumatism internally. In the present study, we aim to investigate the possible biological active constituents present in N-hexane extract of leaves of Lavandula bipinnata by subjecting it to GCMS analysis. Objectives are collection and identification of plant, preparation of sample for phytochemical screening, preliminary phytochemical screening, GCMS analysis, and identification of compounds. Materials and methods. Plant material Lavandula bipinnata was collected from the campus of Botany Department of Nanded University, Nanded, India, and it was identified and authenticated by Botanical Survey of India, Pune. For preparation of sample, we followed Soxet extraction method, and N-hexane was used as a solvent. The solution was concentrated in vacuum to dryness, and dried extract was stored in refrigerator at 4 degrees Celsius for further use. These are some of the pictures. Preliminary phytochemical screening was done for the secondary metabolites such as alkaloids, tannins, phenols, etc. Standard methods were followed. These were the conditions that were followed for GCMS analysis. Identification of compounds was done using mass spectra of flavors and fragrances of natural and synthetic compounds library, as well as National Institute Standard and Technology Library. The spectrum of the known constituent was compared with the spectrum of the unknown constituent stored in both the libraries. The results and discussion. Preliminary phytochemical screening of N-hexane leaves extract of Lavandula bipinata revealed the presence of phytochemicals such as alkaloids, tannins, phenols, flavonoids, terpenoids, quinones, cardiac glycosides, whereas sterols, saponins, anthraquinones, proteins were found to be absent. This is the result in tabular format. These are the pictures for the preliminary phytochemical screening. The presence of alkaloids suggests the ability of this plant to play a role as a detoxifying agent, and it has anti-hypertensive properties. Tannin acts as an antidiarrheal and anti-hemorrhagic agent. Phenolic compounds prevent oxidative damage in living system, which indicates this plant might play an important role as dietary antioxidants. Flavonoids are known to be highly effective scavengers of most oxidizing molecules, whereas terpenoids have been reported to have potent activity against gastric ulcers. The gas chromatography mass spectroscopy profiling of N-hexane extract revealed the presence of 17 major compounds. The major compounds were butanic acid, 2-methyl, 2-methyl-butyl estrel, fencone, phthalic acid, heptadecane, hexadecane, and others. This is the GCMS chromatogram of the N-hexane leaves extract of Lavandula bipinata. This is the tabular form of bioactive compounds identified in the N-hexane extract with its molecular formula, molecular weight, peak area, and structure of compounds. A total of 17 compounds were identified. In recent times, there is growing awareness in correlating bioactive compounds and their biological activities. Gas chromatogram mass spectral analysis of N-hexane leaves extract of Lavandula bipinata identified 17 phytochemical constituents. Majors are N-hexadecanoic acid, which is also called as palmitic acid. It has antioxidant 5-alpha reductase inhibitor properties. It is also considered as nematicide, pesticide, anti-cancer, and hemolytic activity. The squalene is a triterpene, which is phenolic compound found in latex and resins of some plants. Then dodecane, it is an alkane and an aliphatic compound. It has antibacterial and antifungal activity. Hexadecane has antimicrobial, antifungal, and antioxidant activity. Fencone is a ketone monoterpenoid. It is found in fennel oil and in the oil of Lavandula stapes. It is mainly used in flavor and perfumery. Caryophyllene has potent anti-inflammatory, antimicrobial, antibacterial properties. The anti-proliferative effect of caryophyllene on several cancer cell lines was reported by Dham et al. In the year 2015. Report also showed the caryophyllene suppressed PC3 prostate cancer cell and MCF breast cancer cell proliferation in a dose-dependent manner. Conclusion. In the present study, Lavandula bipinata leaves have shown to have various secondary metabolites which possess many pharmacological properties. The 17 compounds which were identified have activities like antimicrobial, antioxidant, anti-cancer, and many other activities. Therefore, the presence of these bioactive compounds is responsible for therapeutic effect of plant. Further investigation is required for possible development of drugs using these bioactive compounds found in Lavandula bipinata. These are few of the references which I used. Thank you.
Hello everyone, my name is Shweta Pereira. I'm a student of MSc. Today what I'd like to present to you all is the topic extraction of anthocyanins from Goan wild berries. This talk was carried out at the Department of Biochemistry at Parvatibai Chogle College of Arts and Science as a part of a dissertation along with 8 of my classmates under the guidance of Dr. Adulcha Naik. These are the contents of the presentation. So plants have special pigments which absorb light and impart colors. So the 4 main types of pigments are chlorophyll, anthocyanins, carotenoids, and betalains. Anthocyanins are the water-soluble plant pigments and they impart colors to different flowers, fruits, and vegetables. Anthocyanins are also known to have various health benefits as they are potential antioxidants and they also act as natural pH indicators. The aims and objectives of this project were to extract the anthocyanin, to optimize the extraction process, to characterize anthocyanins, to study the effect of pH, and to establish consumer preferences for anthocyanin fortified food products through an online survey. The berries which were used used in the study were Carissa carandas, Ixora coccinea, Cesium cumini, Garcinia indica, Buchania lanson. The features of this are displayed on the slide. The extraction solvents which were used were distilled water, ethanol and water in the ratio of 1:1, ethanol, distilled water and acetic acid in the ratio 49:49:2, distilled water and acetic acid, and ethanol and 0.2% citric acid in the ratio of 1:1. Coming to the methods, the wild berries were first collected, their taxonomical identification was carried out, and then the extraction solvents were prepared and the extraction was carried out. So after the extraction, the TLC and pH and spectrophotometric analysis were done for the extracts. Also, an online survey was conducted to understand the consumer preferences on anthocyanin fortified food products. Coming to the results and discussions. So the anthocyanin extraction was carried out using different extraction solvents, and we found that the extraction solvent, that is distilled water and acetic acid, gave good results. This is shown a picture which depicts the TLC plates of Carissa carandas with the different extraction solvents. For the TLC, the mobile phase which was used to develop the plates were butanol, acetic acid and water in the ratio of 4:1:5, and the stationary phase used was silica gel. The RF value of the plates was calculated, and the results were then compared with literature data. For Garcinia indica, there was one large spot on the plate, and literature data states that there are two anthocyanins present, so it can be assumed to be either one anthocyanin. For Carissa carandas and Cesium cumini, 3 spots were seen on the plates, and literature data also states that there are 3 anthocyanins present. So it can be corresponded to the mentioned anthocyanins. For Buchania lanson and Ixora coccinea, there was one spot on both the plates, and the presence of anthocyanins in these berries is reported for the first time to this study. These are the pictures of the TLC plates of the different berry extracts with the solvents used, the pH and spectrophotometric analysis. So we know that anthocyanins change their color with pH. So from these pictures, we can see that in acidic conditions, the extracts are showing reddish to pinkish shades, and in basic condition, it is shifting towards more bluish and purplish shades. So from this, it is changing. We can see that it is changing color with pH. So it is indicating that anthocyanins are present. Here, red cabbage was used as a standard for the comparison of the berry extracts. These are the absorption spectra of anthocyanin extracts at different pH. So from these figures, we can see that at pH 2, the absorption peaks are ranging from 500 to 550 nanometers for all the berry extracts. But for Ixora coccinea, there are peaks in the range of 400 to 450 nanometers, and this is consistent with the anthocyanin absorption spectra. And there are also variations seen in the graphs at different wavelengths. This is also a strong indicator of anthocyanin being present in all these berry extracts. A survey was conducted using Google Forms to understand the consumer preferences on anthocyanin fortified food products, and the responses were later analyzed. So overall, we got a quite positive response from the survey, and the survey clearly indicated that consumers would like to consume the anthocyanin fortified food products in the form of ready-to-eat products. And among the ready-to-eat products, the majority would like to consume them as ice creams, followed by yogurts and juices. And a minority of them were interested in having them as jellies and carbonated soft drinks. When asked about their opinion on the anthocyanin fortified food product, the response was very positive. Most of them supported the idea of anthocyanin fortified food products. Also, they were asked about paying a premium price for the anthocyanin fortified food products. So, majority of them were not sure of paying a premium price, whereas a minority of them agreed on paying a premium price for the product. So, through the current investigation, we have achieved the extraction of anthocyanins from woven wild berries, which are reported for the first time, that is from Buchania lanson and Ixora coccinea. So, these results indicate the potential use of these anthocyanins in food industry as they are extracted using aqueous solvents which are permitted in food manufacture. So these projects can be further uplifted by characterizing the previously undocumented anthocyanins. Antioxidant assays can be carried out to understand the therapeutic potential, to purify the anthocyanin extract, to study the stability, to develop anthocyanin fortified food products, and also investigate their stability. These are the references. Thank you.
Hi, I'm Pankara, Research Scholar, Department of Botany, Science, Wph University. My topic name is the histochemical and phytochemical study of Tlc Hamiltoni. Introduction. Tlc Hamiltoni is a climber belonging to family of the Cyanaceae. It is used as a substituent of Chemidesmus indicus. Majorly phytoconstitute glycol, beta-amarine to hydroxy for methylbenzene ether. The roots of drug used as a coolant and blood purifier in traditional medicine system, which having properties like emetic, diuretic, anti-inflammatory, etc. Material method. The collection of plant material. The collection of plant material found the site of Wph University in month of September 2018. The microscopic trace plant parts used. The microscopic study were carried by the preparation section of root, stem, and leaf. Sapling stain was used for staining, mounted in glycerin for observation. Thin section were observed under binocular microscope. Photograph at different magnification level taken by histological test. The section of root, stem, leaf were tested in different regions and localization of alkaloid proteins, starch, fat, glycoside, tannin, etc. Primarily phytochemical test. Primarily phytochemical test for determination of alkaloids, anthraquinone, amino acid, carbohydrate, thymine, glycoside, phenol, saponin, steroid, starch, tannin, etc. Then results. The case of food show the phylum cortex and vascular tissue. Starch gain or cortical cells fully loaded with large size. Lenticerous ducts scattered throughout the cortex. Case of stem, epidermis, cortex, and vascular tissue present. Epidermis is a multicellular cell with irregular shape and thin wall. Cortex region is the innermost layer and consists of xylem and phloem. Vascular bundle, different size and number. Teeth, large and hexagonal parenchymatous cells. Case of lip, show lamina and midrib regions. Lamina dorsiventral and shows upper and lower epidermis with polygonal tubular cell with smooth cuticles. Upper epidermis has a unicellular covering trichomes, well developed collateral vascular bundles. Histochemical test. Tlc is source of alkaloid protein, starch, fat, etc. The alkaloid detected by using different two reagents, Wagner and Hagas reagent. Wagner is a reddish brown color and Hagas yellow ppt is present in a root, stem, and leaves. That means the alcohol present root, stem, and leaves. Protein treated with potassium ferrocyanide instance occurs greenish blue color in root, stem, as well as leaf. That means the protein present in a root, stem, and leaves. Starch section treated with iodine and potassium iodide indicated dark blue color in root and stem, absent in leaves. So, starch present in root and stems only, absent in leaves. Another section said, kept in Sudan stem for 20 minutes, show absence of fats in all parts of the plants. Also, glucoside absent in all plant parts. Tannin. The treated with the dilute acetic ferric chloride solution with the greenish color, that means present in a tannin, root, stem, as well as leaves. Phytochemical analysis. Presence of physical metabolic drug was confirmed by the pharmacological action. The quality screening, alkaloid, flavonoids, glucoside were detected, alcoholic extract of leaf, stem, and root. Alcohol in all study plant parts were detected by the Wagner's, Meyer's, Dagerberg, and Hager's reagent. It was found positive and ethanol, and dichloromethane, where negative hexane and aqueous extract. Carbohydrate was present in all plant parts
with two mollusc reagent to positive test indicator was red gum-molybdenum. The presence of flavonol and flavanol was confirmed by the ammonia test. This test is ethanol methanol important imparted color, whereas the negative in hexane, dichloromethane aqueous extract. Glucoside were also present in all parts. Those were subjected to filling test, ferric chloride test, give calcium high precipitation. The test show positive in all solvent except the water extract. Anthraquinone gives pink red color in blotting test. It was positive in hexane aqueous extract but negative in ethanol and methanol. Amino acid and protein were added millions and in hydrant reagent gave the greenish black and blue color in all solvent respectively. Saffron present in ethanol solvent and fats were absent in all plant parts. Steroid were common in all plant parts. Tlc test indicated occurrence lower layer that turned cherry red color. Steroid were found in positive in hexane and ethanol, whereas negative in methanol and aqueous extract respectively. Potassium and potassium iodide reagent gave the bluish black precipitate shows presence of starch in fruits and stem but absent in leaf extract. If stem and leaf extract were added acidic ferric chloride and transiman gave the greenish and blue color respectively. The confirmed presence of tannin and phenol. It was a positive all solvent except aqueous extract. Conclusion: this study is aligned with quality parameters prescribed in Ayurvedic Pharmacopoeia of India and also standard tests by other international agencies. The present study was enormous practical implication to anthraquinone material that has been used as drug trade based on family leukocytosis in general and Tlc in particular. The detailed investigation for treating microscopic feature along with the biochemical investigation shall aid in confirming and further selecting opposite plant-based drug. This study concluded that pharmacological and phytochemical study and declared will be highly useful to determine qualitative and quantitative standard which can ascertain the identity, quality and purity of this plant drug. In turn, it will result standard herbal vision for the betterment of human society. The present investigation thus highlighted urgent need to undertake detailed scientific research, explore this common endemic and least studied plant for its potential in these herbal preparations. Here are a few references. Thank you.
Hello, this is Vaishali Nilmakar. I'm going to present anthocyanin as a natural plant-derived antifungal agent here in the category of medicinal botany and phytochemistry. Anthocyanin, as all of us know, they are the polyphenolic compounds, the natural pigments, and they are the water-soluble pigments. They have the antimicrobial activity and they, especially inhibiting the foodborne pathogens. In the present study, different plant materials were collected and extraction of anthocyanin was done from them. Determination of both qualitative as well as quantitative was done, and its antifungal activity was determined by using the minimum inhibitory concentration as well as agar-well diffusion methods. The plant material used here were beetroot, black carrot, red leaves of Terminella catappa, green leaves of Terminella catappa, red and green leaves of Terminella catappa, marigold, red capsicum, and purplish red cabbage. For the qualitative determination of anthocyanin, the Tlc method was used where about 3 to 4 spots were found in both the extracts, that is acetone as well as acetified methanolic extracts here. So many of these R values were found to be common with each other, depicting that the same compound might be present, but still more studies is to be done on that. For quantitative acid, the pH differential analysis of anthocyanins was done in terms of cyanidin-3 glycoside, where it was found maximum in the red cabbage, followed by Terminella catappa, red leaf, and the black carrot. Antifungal activity was determined by using the minimum inhibitory concentration, where in the acetone extract against penicillium species when it was found, here two extracts, that is acetone extract and acidified methanol extract with penicillium species and aspergillus species was done. So here for the acetone extract, when the MIC was found, it was found maximum in the red capsicum, beetroot and black carrot at 72 hours. For penicillium species. And for acidified methanolic extract against penicillium species, it was found for Terminella catappa, red leaves, again for 72 hours. The acetone extract against aspergillus species, it was found for red capsicum and black carrot, that is the maximum inhibition was given at 72 hours. The red capsicum, their maximum inhibition in the acidified methanolic extract against aspergillus species at 72 hours. The antifungal activity was also checked by using agar-well diffusion method, where it was found that the red cabbage and black carrot, almost red cabbage, black carrot, beetroot, marigold, they get the maximum zone of inhibition, and it was maximum seen in the red cabbage, that is at 72 hours it was about 23, red cabbage and black carrot both against penicillium species. These are some of the peaks, and for the acetyl extract against penicillium species, it was found maximum with terminal catappa red leaves and terminal catappa red green leaves. These are some of the peaks. For acidified methanol extract against Aspergillus species, red cabbage was found to be very potent against Aspergillus species at 72 hours, that is 29 millimeters zone of inhibition was seen. These are some of the peaks. The antifungal activity of acetyl extract against Aspergillus species was also found to be maximum with terminal catappa red leaves, red and green leaves, and red as well as with the red cabbage. These are some of the peaks for that. So in conclusion, it could be said that the extraction of anthocyanin from cheaper substances could be used for industrial applications. The skin, which is mostly treated as a waste product, would be used as a very useful source for colorants. Anthocyanins can also be used as a natural plant-derived antifungal agents. It could be used to prevent the fungal spoilage and deterioration of the processed foods. It could be a safe product with antioxidant as well as antimicrobial potential. These are some of my references. Thank you.
Greetings to one and all. Myself, Meena Desai, under the guidance of Dr. Janvi Bhagwat, will be presenting a research paper titled Red Blood Cell Membrane Stabilizing Potential of Roestonia regia bark extract. Sadeke et al. In his paper mentioned that RBC membrane resembles lysosomal membrane. So, the stabilizing activity of RBC membrane can be extrapolated to the stabilization of lysosomal membrane. But why is stabilization of lysosomal membrane that much important? During inflammation, lysis of lysosomal membrane takes place, which releases hydrolytic enzymes, causing tissue inflammation and organelle damages. These damages lead to acute as well as chronic disorders. Therefore, in our paper, this paper of ours, we have explored the stabilizing activity and potential of Roestonia regia bark extract. Roestonia regia is known as Cuban royal palm, which is natively present in Mexico, parts of Central America, and Caribbean, southern Florida. It is being cultivated in some places, like in India, as a landscape palm. It has various benefits, like seeds is used as livestock feed and a source of oil. It has the lipid extracts from the fruit of this palm has protective effects on prostatic hyperplasia. Palm has a rich array of medicinal use, but its potential benefits to the biological effects in humans has still to be explored. In this paper, these are the objectives which we had followed. As bark was collected, washed, shade dried, and grinded to coarse powder. The powder was then extracted in ethanol for 3 days, filtered, dried, and stored for further analysis. Screening of phytochemicals were done so as to see the presence of alkaloids, carbohydrates, proteins, tannins, etc. In the extract. Antioxidant potential of the extract was seen by Wph Scavenging Activity. Various concentration of extract was used. 1 mg per ml concentration of ascorbic acid was used as a standard. Further ahead, in red blood cell membrane analysis, various concentration of extract with 1, 5, 10, 15, and 20 mg per ml was used. Same concentrations of standard was used. Standard was used as diclofenac sodium, then very well known anti-inflammatory drug. Whole blood from a healthy human being was taken and RBCs were separated. These RBCs were incubated along with the extract or standard, and hemolysis was induced by hypotonic solution, heat, and cold. Are the results of phytochemical analysis? Presence of alkaloids, flavonoids, polyphenols, tannins, proteins, saponins, and carbohydrates were noted. The extracts showed a very high Wph scavenging activity. The maximum of 91.07% inhibition was observed at 5 mg per ml. The percent inhibition ranged from 74.83 to 83.47. But in hypotonic solution induced hemolysis, the bark extract showed a gradual increase. It exhibited highest stability of 95.05% at 20 mg per ml concentration. If, as we can see, it was very near to that of the standards 97.25% stabilization, which is a very nice result. The extract showed in heat-induced hemolysis showed a minimum of 2.32% per ml stability and maximum 66.87% stability. As we can see that the extract showed a sudden increase from 16.2% at 10 mg per ml to 64.43% at 15 mg per ml. We can also note that the extract showed more stabilization than that of the standard at 15 mg per ml concentration. In cold-induced hemolysis, a gradual increase was noted so as that of the hypotonic solution induced. The membrane stability of the extract in cold-induced hemolysis ranged from 80.83% to 62.7%. Thus, the research revealed that the extract contains compounds that have high antioxidant potential, and it also protects the RBC membranes very effectively. It was also noted that extract showed concentration-dependent membrane stabilizing activity. That is, the stabilizing activity of the extract increases with increasing concentration. As suggested by Wph et al. 2004, it might be because of the mode of action which can be connected with binding of RBC membrane with subsequent alteration of the surface charges of the cells. It might also be because the physical interaction was prevented with the aggregating agents or the promoter dispersal by the mutual repulsion of the like charges which were involved in the hemolysis of red blood cells. Sadiki et al. Also joined or connected these stabilizing potential of the extract along with the phytochemicals which are present, like saponins and flavonoids, shows a very effective membrane stabilizing effect on lysosomal membranes. The tannins and saponins which are present in the extract may also have the ability to bind cations, which extends to the stabilization of erythrocyte and other biological macromolecules. Ravipati et al. Likely said that high antioxidant activities possesses good anti-inflammatory activities. Olipano et al. In 2010 also stated that the lipid peroxidation and oxidative stress damages the lysosomal membranes. Thus, if the extract has a better scavenging of free radicals, these damages can be prevented, thus resulting to the stability of the lysosomes. So, we can conclude that the methanolic bark extract of Rostonia regia contains A. higher amount of phytochemical presence, B. a high DPPH free radical scavenging compound, and C. effective membrane stabilization property. These three may also be connected with each other. Thus, it can be confirmed that Rostonia regia is a potential natural source of red blood cell membranes. These are some of the references which we have gone through. Thank you.
Good day to you all. My name is Mona Joshi, and I am here to present my research work on the topic antibacterial and antioxidant activity of the molecules. The work presented here is performed under the guidance of Professor Gupta, research guide, and Dr. Roger Kaur, lecturer JNIT, Jane Khalsa College. All the work presented here was conducted at JNIT Department, Jane Khalsa College. According to Indian traditional medicinal practices, garlic has been a vital part of the various products targeted at reducing heart-related infections, improving gut health, and easing heart-related conditions. It is observed that food of plant origin contains significant amount of bioactive phytochemicals, which may provide health benefits beyond just nutrition to reduce the risk of chronic diseases. In this project, we studied the antibacterial, antifungal, and antioxidant activity of garlic extracts. To start with the experiments, the extracts were obtained by crushing, macerating the garlic in selected solvent and putting it on the overnight protein shaker for the extraction. The extract thus obtained was then filtered, evaporated, and reconstituted in suitable solvents for further analysis. The total phenolic contents were estimated using Collins-Revolto method. The reagent contains phosphomolecular phosphoric acid complexes. The method relies on transfer of electrons in alkaline medium from phenolic compounds to form a blue chromophore constituted by phosphomolecular phosphoric acid complexes, where the maximum absorbance is related to the phenolic content of the extracts. The results were reported as the garlic acid equivalents. The content present in garlic was found to be in the range of 80 to 89 milligrams. The minimum inhibitory concentration assay is a 96 title plate method, where we use the multifold dilutions of the extracts obtained to study its inhibitory effect against selected microbial strain. The result of MIC is obtained in the form of minimum concentration required to kill the microorganism under the study. The minimum concentration thus obtained by using MIC assay is then further used
to study the minimum bactericidal concentration of the extracts. The minimum bactericidal concentration is established by using an Alvaro assay where we use the MIC concentration and one higher concentration to study the bactericidal effects of the extracts. The results of MBC assays are reported in the format of diameters of zone of inhibition, and depending upon these diameters, the extracts or the antibiotic agents are categorized into the mild, effective, and strongly effective categories of the antimicrobial agents or the antibiotics. The ferric reducing antioxidant power assay is a method that measures the reduction of ferric ion ligand complex to the intensely blue colored ferrous complex by antioxidant in an acidic medium. The result for FRAP is reported in the form of absorbance. More the absorbance, more is the antioxidant activity, and thus more may be the phenol content of the samples under the examination. The DPPH free radical method is an antioxidant assay based on electron transfer that produces a violet solution in ethanol. This free radical, which is stable at room temperature, is reduced in the presence of an antioxidant molecule, which turns the ethanol solution colorless. The result of the DPPH assay is reported as the IC50 values of extractives, that is the minimum concentration required to reduce at least 50% of the free radical present in the solution. The yield of the extract obtained using ethyl acetate as a solvent falls into the range of 20 to 22%. The extract happens to show antimicrobial activity at the concentration of 40 ppm and higher. The extract also happens to be a potential antioxidant agent. Further studies need to be conducted in order to isolate and identify the constituents to which these properties are attributed to. These are my references. As Dr. Kalam said, excellence is a continuous process and not an accident. We all being part of such a process, I would like to wish you all the best in this research journey. Thank you for your patience while listening to me. I would like to thank Dr. K. Mandokar, Principal Sir, GM Taxa College, Dr. Suriva Gupta, my research guide, and our coordinator, Dr. Ravindra Kaur, for supporting me throughout this research work. I would also like to thank the organizers and coordinators of this conference for providing us such a beautiful platform to present our work.
Greetings to all. I, Rishita Hegde, on behalf of my group members Abhishek and Sandhya, will be presenting our group project of MSc Part 2 in silico and toxicoinformatic analysis of aflatoxin as endocrine disruptors. These are the subtopics which I will be presenting in this presentation. Now, what are aflatoxins? Aflatoxins are basically a mycotoxin which is produced by Aspergillus flavus and Aspergillus parasiticus. Aflatoxin generating fungi are generally observed on lifeless and decaying flora, which infects agricultural crops. Contamination of aflatoxin has been reported in food and feed by such as groundnuts, corn, seeds, maize, wheat, rice, and etc. These are some of the most common aflatoxins which are found to be most toxic and carcinogenic. Mostly, aflatoxins are considered as an unavoidable contaminant. Aflatoxin was actually named around 1960 after its discovery as the source in a disease, turkey eggs disease. Aflatoxin contaminated feed was consumed by various poultry animals, which resulted in death of nearly 1 lakh poultry animals. The first image shows the death of poultry animals. In the second image, the left-hand side DNA is the normal DNA. The right-hand side DNA is shown after the aflatoxin is interacted with the DNA. Aflatoxin is also considered as an endocrine disruptor. Endocrine disruptors are usually known as hormonally active agents which interfere with the endocrine system. These disruptions cause cancerous tumors, birth defects, and other developmental disorders. Objectives of this project was to understand the toxic effects of aflatoxin as an endocrine disruptor and which aflatoxin binds best with the majority of the nuclear receptor. Materials and methods. This project was wholly carried out by in silico method, which is done by the computer stimulation. 15 nuclear receptors were selected and docked against 18 aflatoxins or ligands. For toxicology analysis, Web tool Protox 2 was used, and for bioavailability analysis, Sadme Web tool was used. These are the aflatoxins and nuclear receptors which were selected along with their CID number and PDB IDs. For the docking process, Discovery Studio software was used in which the heteroatom and the water atom of the nuclear receptor was deleted. After the deleting of water molecules, Ufsc Chimera was used to do the energy minimization process. After this, the processed protein or the processed nuclear receptor was loaded in the Pyrex software, from which, using the open babel thing of the Pyrex software, the ligands were inserted and one nuclear receptor with all the 18 ligands were docked. The statistical data shows that aflatoxin P1 shows the highest binding affinity. The aflatoxin P1 shows the highest binding affinity with glucocorticoid receptor, which has a PDB ID of 4P6X. The results of the docking showed us that most of the aflatoxins shows good binding affinity with most of the nuclear receptors, from which aflatoxin P1 shows nice binding with the nuclear receptor. Now, aflatoxin P1 is mostly found in contaminated urine samples of humans and animals, and also in dairy products. The 2D and 3D images of aflatoxin P1 binding with glucocorticoid receptor is shown in this slide. When aflatoxin binds with this receptor, it usually results in growth impairment and slow immune response. Toxicology analysis showed that aflatoxin P1 is the most carcinogenic and immunotoxic and mutagenic. Bioavailability score of aflatoxin P1 was 0.11, which showed that it is not sufficiently absorbable via the oral route. From this project, we concluded that aflatoxin P1 shows good binding affinity with major of the selected nuclear receptors, and most of the selected aflatoxins also show good binding affinities with the nuclear receptors. Scope of work was aflatoxins were mostly researched on their treatment or detoxification property and the health effects of aflatoxin. Research is needed on aflatoxin P1 to know its effects of human physiology. These are the various references used in our project. I would like to acknowledge Ajita ma'am, coordinator of MSc Part 2 Chemistry Department, for helping us throughout this project, and Vikas sir of NFB Department for helping us throughout this project. Thank you.
Hello everyone, welcome all of you. I am Dr. Ahide Yogesh Raghunath, Assistant Professor, Department of Botany in the Shri Chhatrapati Shivaji Mahavidyalaya, Shri Gonda, Ahmednagar. And my today's research topic is about the allelopathic potential in Coccina grandis green voids. So basically this allelopathy, so what is it? Actually it is allelopathy. So allelopathy is a chemical process that a plant uses to keep other plants from growing too close or too away from it. So the word allelopathy derived from the two separate words, allon which means the of each other and pathos which means of to suffer. So according to Molish in 1937, he first coined the term allelopathy, which refers to biochemical interaction between all types of plants, including microorganisms. And according to Rice in 1984, he also defined allelopathy as any indirect harmful or beneficial effect of one plant to another plant through release of chemicals into the environment. So these plants so basically, these plant parts release aerochemicals compound into the environment by various processes, like we can say root exudation, leaching, voltarization, then decomposition of plants, other issues of it. Okay, so for this study, I've used the plant in this present investigation. I've used the Coccina grandis, which belongs to family Cucurbitaceae, and which is also known as a irrigard, and Marathi it is known as a thorn liver. So it is a dioecious creeper found to be growing wild on edges of India. All over we can see in India it can be found. It has a very medicinal use also. In this picture you can see the habit of it, the leaf and flowering tip and the root one. Next slide is a this material method used for this research. So the mature seeds of Phaseolus equitifolus, that is a jack variety Abhaya, was collected from the various agricultural shops. So these seeds was surface sterilized with the mercury chloride, that is a 0.1%, followed by the washing and for washing with the distilled water, and carefully dried and used for biopsy for seed germination and seedling growth. So we can say to evaluate the allelopathic activity of Coccina grandis on the Phaseolus equitifolus seeds, two types of biopsy were conducted in the laboratory. The first, that is the phytochemical biopsy, and the next that is leachate biopsy. The first, that is a phytochemical biopsy. In this, the plant parts separately it was taken, that is 100 gram fresh roots, and then stem and the leaves of Coccina grandis was selected. Then it was washed and blinded separately with a distilled water. It was filtered through the Whatman filter paper, and a stock solution was prepared from the ratio of 1:2.5, having a final volume of 250 ml. And this was used as a 5-pressure biopsy. So the dilution was made from the stock solution. That was 1%, then 1.5, 2, and 2.5%. And the last one of petri dish containing filter paper moisture with 100 ml, 10 ml distilled water was served as control one. And the next
Neha, till the time you can present Sakshi's presentation only.
Is there a technical issue, Gopal?
Yes, sir, we are trying to sort it out.
Yes, yes, no problem. You take your time.
Give us few minutes. I think the video is not playing.
আমরা আমাদের লাঞ্চ খেয়ে আসি বুঝলি
দাঁড়াও 2 মিনিট দাঁড়াও হয়ে যাবে
তুই তো খাওয়াবি না
তো এটা
তুই খাওয়াবি
চলে এসো কেন কেন খাওয়াবো না চলে এসো
ইমেলে পাঠিয়ে দে
ইমেলে পাঠানো যাবে না চলে এসো খাওয়াবো
কোপা এখন অনলাইনে সব অর্ডার করে দেওয়া যায়
এইটা রংশুমার একটা কাজের কথা বলেছে রংশুমার একটা কাজের কথা বলেছে
সবাই কেন বাদ যাবে
Good morning, everyone. Myself, Sakshi Bhatte, research scholar from Botany Department of Tlc College. My topic is evaluation of adulteration, caffeine content, and antioxidant activity in different plants of plant. So, as we all know, tea is obtained from the Camellia sinensis plant, which belongs to the Tlc family. So the adulteration is a process which introduces the low quality or almost similar quality components to the original product, which result in quality degradation and ultimately harmful consequences on the customer's health. Nowadays, tea is one of the most consumed beverage worldwide after the water. Tea also contains some amount of caffeine to it. So the caffeine is a member of xanthan family. It is odorless and colorless compound when it is submerged in the hot waters. Caffeine occurs naturally in tea plant. For example, Camellia sinensis. Then tea also contains L-theanine, which gives the feeling of calmness and the relaxation. And L-theanine is only found in a tea plant. As we all know, tea is a rich source of antioxidants. So the antioxidants, the detoxifying effect of the antioxidants protects cells from the free radicals, damage that can lead to blood clot formation, cancer, brain dysfunction, and cardiovascular diseases. So the daily permitted average consumption of the caffeine is around 400 mg per day. Therefore, in this research, we have quantified caffeine as well. So these are some adulteration tests: coal tar dye, then sand particles, iron fillings, artificial color, azo colors, cereal starch, and chicory. So these are the results for the adulteration test as we took 5 different market brands. That is brand 1, 2, 3, 4, and 5. Then comes the caffeine content. See, it is said that black tea generally has more caffeine content as compared to the other tea. But it is difficult to state which tea variety has the absolute highest and the lowest quantity of caffeine because there are so many different techniques to measure caffeine. There are plenty of criteria that go into determining the caffeine level of tea. And any tea manufacturer can pick one and claim that it is the highest caffeine containing tea available, even if it is a taking data from an ideal period in time that changes as it is processed. So in this method, we have used chloroform method to determine caffeine content from different tea brands. So these are the caffeine crystals which we got from the different tea brands. And this is a photo that is caffeine separation using separating format. Yes. Then comes the antioxidant property. Tea is a rich source of polyphenols or flavonoids, that is catechins, then theaflavins, then thealurogens, which act as antioxidants. We have used the Wph method to study antioxidant activity from the tea brands. So this is the result for the caffeine content and antioxidant activity. So yes, the caffeine varies from 0.71 plus or minus 0.09 to 1.20 plus or minus 0.09. And the TPPH method shows the antioxidant activity ranging from 3.29 plus or minus 1.03 to 15.96 plus or minus 1.21. These are two graphs we obtained. The first one is caffeine content and antioxidant activity of the tea or brands. Now the second one, it is a correlation between the caffeine content and antioxidant activity of the tea brands. So yes, this is our team, myself Sakshi Khate and my guide Dr. Bobita Rana, Assistant Professor from Guru Nanak Khalsa College. So these are some references and yes, thank you.
অর্ণব শোনা যাচ্ছে?
না শোনা, we are seeing there is some technical problem.
So we are having last video here. We are playing that.
Okay, okay.
Having a final volume of 250 ml, and this was used as a phytoester biocide
Of LSD department.
To be growing wild on edges of India. All over the, we can see in India it can be found. It has a very medicinal use also. In this picture you can see the habit of it, the leaf and flowering tip and the root one. Next to that is a this material method used for this research. So the mature seeds of Phaseolus acetifolus, that is a jack variety Abhaya, was collected from the various agriculture shops. So these seeds was surface sterilized with the mercury chloride, that is a 0.1%, followed by the washing and for washing with the distilled water, and carefully dried and used for biopsy for seed germination and seedling growth. So we can say to evaluate the allelopathic activity of Coccina grandis on the Phaseolus acanthophilus seeds, two types of bioassay were conducted in the laboratory. The first, that is the phytoextract bioassay, and the next that is leachate bioassay. The first, that is the phytoextract bioassay. In this, the plant parts separately it was taken, that is 100 gram fresh roots and then stem and the leaves of Coccina grandis was selected. Then it was washed and blended separately with the distilled water. It was filtered through the Whatman filter on paper, and a stock solution was prepared upon the ratio of 1:2.5, having a final volume of 250 ml. And this was used as a phytoextract bioassay. So the dilution were made from the stock solution. That was 1%, then 1.5, 2, and the 2.5%. And the last one, a petri dish containing filter paper moisture with 100 ml, sorry, 10 ml distilled water was served as control one. And the next, in this petri dishes, each petri dish containing 10 seeds of Phaseolus acanthophilus seeds were kept in a triplicate form at the room temperature, which was wrapped, this petri dish was wrapped with the brown paper, and to avoid with the direct sunlight, and the data of seed germination, or that is the hypocotyl, and the radical length were recorded. Next, that is the leachate bioassay. So in this, leachates are what is actual leachate? It is the removal of substance from a plant by the action of aqueous solvent. So to find out the effectiveness of inhibitors leaching out from the Coccina grandis, 100 grams of healthy and clean plant parts were soaked separately, that is the root stem leaf, in equal amount of distilled water for about 72 hours. So each leachate was filtered through this Whatman filter paper, and this filtrate was brought at a volume of about 250 ml with the addition of filtrate used for the biopsy. And this next method, that is was HPLC. So this HPL, that is high performance thin layer chromatography technique, was followed for the qualitative analysis and confirmation of phytochemicals present in it. The result and discussions. So the present innovation clearly demonstrates the inhibitory effect of root, stem, and leaf of Coccina grandis on the seed germination as well as radical and the hypothetical growth. So first, that is the phytoextract bioassay. You can see that in this image. So the root, the stem, and the leaf phytoextract bioassay indicates that major inhibitory activity was caused by the ratio of 1:2.5% of phytoextract of each part. So it is also observed that the major toxicity was caused at 2% and 2.5% of phytoextract of all the three parts. So the root extract shows greater inhibitory as on seed germination and seedling growth of Phaseolus. In this picture, you can see this. That is the root phytoextract bioassay, then seed phytoextract bioassay, and leaf phytoextract bioassay. This table shows clearly indicates that 2% extract concentration of 2% and 2.5%, radical length and the hypocotyl length was lesser in that area, inhibited. In the stem also, stem phytoextract, the same situation was found, that is in the 2% and the 2.5%. And the same was followed by the leaf extract also. Now the second, that is a leachate bioassay. So the leachates of roots, stem, and the leaves significantly affect the seed germination and seedling growth of test crop. So it is observed that root leachates fully suppresses the seed germination as well as overall seedling growth of Phaseolus. So the root leachate shows more inhibitions than stem and the leaf leachate, as you can see in the image 2. That is, the first image shows the control and the root leachates effect of, which shows the effect of this plant leachates on the seeds of the Phaseolus. The second, that is the stem leachates, and the third is the leaf leachates. So these tables, you can, in this table also you can see this reduction in the germination is found in this majority is found in the root leachate only as compared to the stem and the leaf one. So this reduction in germination might be due to water-soluble chemicals present in the phytoextract as well as in the leachates. So this proves that there are some chemicals present in it. So with the help of HPTLC techniques, lipule phytoconstituents were identified and confirmed from the root and the stem of Coxiella grandis with the authentic samples and as well as terpenoids and steroid in root, stem, and leaves of Coxiella grandis. However, the above said when we discussing about this, thus phytochemicals are phytotoxic one. So the presence of these chemical compounds in the plant parts of Coxiella grandis are well correlated with the degree of their phytotoxicity. So these results indicates that the allelopathic activities exist in the Coxiella grandis are because of these phytotoxic chemical present in it. So these are some selected references for my research. The last one, thank you very much. Thank you very much for the Guru Nanak Khalsa College of Art, Science and Commerce, Matunga, Mumbai Department of Botany for organizing this international conference on novel approaches in the life sciences and giving me opportunity to present my paper in this conference. Thank you very much.
Greetings of the day to one and all. I, Yusra Khatib, Assistant Professor of Botany from GM Moment Women's College, Bhiwandi, presenting today under the guidance of Lakshmishri Chengala. This presentation is designated to the theme Medicinal Botany and Phytochemistry, with the title Extraction of Essential Oil from Fresh Flowers of Polyanthus tuberosa, its characterization and TLC analysis. While roaming around the city for a research idea, I stopped at the florist shop, where I found many of the well-known plants like mogra, lily, marigold, roses, aster, some of them with their well-known products in the market. Among them, I found tuberose, with sweet and indolic fragrance and intense white floral. It is commonly known as gulchadi or rajnigandha. Its scientific name is Polyanthus tuberosa, belonging to the family Asparaceae with a spike inflorescence. It is a popular and prized product. The essential oil from the flower is extracted and used as antidepressant, antispasmodic, expectorant, mucolytic, and antidiarrheal too. It also increases blood circulation. So the aim of this project is to extract essential oil from fresh flowers of Polyanthus tuberosa, its characterization and TLC analysis. The first objective is to successfully extract the essential oil from fresh flowers of tuberose with suitable method. Second is to characterize it for the qualitative analysis with the help of some physicochemical parameters. And third is to know several key components with the help of thin layer chromatographic analysis. The fresh flowers of tuberose was collected from the local market of Bhiwandi, Maharashtra, and deposited to the Blatter Herbarium at St. Xavier's College for the identification and authentication purpose. Two methods were chosen to extract essential oil. One is hydrodistillation using Clevenger apparatus, as discussed by Agarwal and Paridhavi in 2012, with some modifications, where the ratio between flowers and distilled water was kept 1:2 and temperature was set at 60 degrees initially and raised to 100 degrees Celsius for 8 hours. In the second method of extraction, N-hexane was used as a solvent and rotary evaporator as apparatus, where the ratio between the material and N-hexane was kept 1:2. The whole setup was covered with aluminum foil and left at room temperature for 24 hours. Resulting two layers were separated and upper layer was subjected to rotary evaporator at 60 degrees Celsius to yield concrete essential oil. The concrete essential oil is then treated with ethanol to yield absolute essential oil. The characterization of essential oil was done considering the following physicochemical parameters: color, density, specific gravity, refractive index, acid value, saponification value, ester value, and peroxide value. And also the TLC analysis was carried out to know the several key components. Those components were identified by cross-checking the RF values with the standard reference book titled Plant Drug Analysis, 2nd edition by Wagner and Blatt, using toluene and ethyl acetate as solvent system and vanillin and sulfuric acid reagent as detecting reagent. Here are the results of extraction procedure. By hydrodistillation, inadequate quantity of essential oil was extracted, and by solvent extraction method, 4.6 ml of absolute oil yielded, which was 1.6% of the total plant weight. In continuation to that, here are the results of characterization. The color was noted under the visible light, UV short and UV long, and it came out to be dark yellow, yellowish green, violet blue, respectively. The density and specific gravity was 1.86. Refractive index at 15 degrees Celsius was 1.38. Acid value was 4.488 mgkh per gram of sample. Saponification value was 420.75 mgkh per gram sample. Ester value was 79.662 mgkh per gram sample. Peroxide value was 16.01 milliequivalent peroxide per kg sample. Continue to that, the thin layer chromatographic analysis showed the 12 bands of different components, out of which blue band was of benzyl benzoate, violet blue was of terpene alcohol, purple color band was of 1,8-cineol. Lastly, the methyl eugenol showed the presence as greenish was heavier than water. It is must that the extracted essential oil should not become rancid. The low acid value and peroxide value, the larger the saponification and ester value, the better the soap making ability will be. The saponification value and ester value were near to the standard range. Benzyl benzoate, terpene alcohol, 1,8-cineol, and methyl eugenol are some key components of any essential oil, and we were able to identify the same. Moving towards the end of my presentation, conclusion can be drawn as the tuberose oil seems to be thermolabile in nature, hence hydrodistillation produced inadequate quantity of oil. As no preservative were added to the essential oil, the oil may become rancid while opening and closing of the lid of a storage bottle, which can reduce the life, quality, and effectiveness of essential oil. The TLC analysis showed the presence of several key components, but for complete screening, one should prefer GCMS. Lastly, I would like to highlight some points on managerial implications of my project. As we know that the rotary evaporator can recover the solvent used, which can be recycled for the next setup. Hence, it can be a cost effective if extracted at laboratory scale. In India, tons of tuberose flowers are used in garland and headdress making, in temples and shrine, and also get wasted, which can be utilized to move towards the zero waste management. Without applying any drug topically and without consuming, one can improve health by aromatherapy. With this reason, the effect of oil can be studied on air microflora. Here are some of the references which have been used to carry out this project. Thank you.
Thank you, technical team. Here we have done with all the 37 presentations. Now I invite our chairperson to give their views and remark on presentations.
Well, thank you, Shital. Yes, there are quite a few presentations. It's a very long session, I must say. And altogether, there are, I think, 42 presentations were there. And then ultimately, some of the presentations are very good. I'm very pleased to listen to those presentations. Some are simply outstanding, and some are very young students. So they have always there is scope to improve. And I must thank the Khalsa College for conducting such a wonderful seminar and symposium for the last 2 days. And still one more session, I think, is there. And regarding the technical issues, these things does happen, so it's nothing to do. But I would like to tell you that you can download the presentation from the YouTube, and a little bit you can edit it and you can then upload it again. So in the YouTube, there will be uninterrupted presentation will be there. So these things, and I would now like to request my co-chair Sanjeev Roy, Dr. Professor Sanjeev Roy, to sum up, if you have something to say regarding some specific presentation, because I know that there are some very good presentations, and Sanjeev may would like to comment on these presentations. So over to Sanjeev.
Thank you, sir. So, long session. I agree with you that session was very fruitful, whatever the condition we have faced. So, as I told earlier also that this session, this specifically medicinal botany area, lots of paper has come from all over the India, Nagaland, lots of paper I have seen, Assam, West Bengal, all the districts. From South Indian paper also is there. So they have covered almost whatever area we need to cover, almost diverse field they have touched. It is good that people are working in different directions. In near future, what is our ultimate aim of doing these natural products? To get some ingredients or active components which may be ultimately may be used for treatment. We can increase the list of medicines. This is our ultimate aim. Whatever may be that young scholars and some senior scientists also do work here, and their presentation is good. So I know that limited few minutes cannot describe properly whatever their mind, as time constraint is there. So I convey our good wishes that in the future seminar also we will interact, and maybe the more suitable way we can complete our main intention of this interaction. So not wasting time, I congratulate all the speakers, all the presenters, and whoever present in this session. Thank you for your all cooperation as well as patience bearing with us. Thank you.
Yes, thank you, Sanjeev.
Thank you so much. Thank you so much.
Thank you. And I would like to say that, yes, Sanjeev was rightly pointed out that from northeastern region, a lot of papers have come and they are doing it very well. So this is rightly said that this is the effort of the central government, state government is really taking shape nowadays, because northeast is coming up in a big way. And I'm also worked here at the northeast, so I feel proud for that. And now I think if anybody else would like to comment on this, there are a lot of audience. I found that, and not still there are 72 people are present there. If you would like to comment a few words, you can do so now. Yes, Anshuman.
But the next session also they have to start in the right
Yes,
time
yes.
of lunch.
I think yes, we should
We should
now
not
stop.
prolong this session. But
So
this
anyway,
is
I would like to thank
very,
all
very
of you, particularly the Khalsa College and the students of Khalsa College. They are very young, but they are very efficient and energetic. And of course, last but not the least, our beloved Gopa. Thank you, Gopa.
Thank you so much, Arnavda, Anshumanda, then
yes,
Sanjeev.
I must say that the whole team under the leadership of Gopa, they are doing excellent job from yesterday.
Excellent.
Thank you, sir. We will move ahead with the formal vote of thanks. On behalf of organizing committee of GN Khalsa College, I take this opportunity to express my sincere gratitude towards our chairperson, Professor Arnab Sen and Professor Sanjeev Ray, for taking out time from their busy schedule. I know our session was quite lengthy, and for giving their valuable feedback for the participants. Thank you to all participants for their enormous response and for making this session successful. I also thank all the staff members and technical team to run the session smoothly. Thank you, Anshuman sir, to join us in this session also. Thank you, all of you. Now I request Pallavi to take over and continue with the further announcement. Over to you, Pallavi. Thanks.
Thank you, Sheetal ma'am. Now we are moving to the next and the last session of this day. That is technical session 4, biodiversity and environmental quality. I would like to request Dr. Babita Rana to kindly introduce our chairperson for this session.
Thank
to Pallavi. Am I audible? A very good afternoon to all and warm welcome to the last session of the conference Novel Approaches in Life Sciences. Dear participants, the session will begin with a talk of two renowned speakers, Dr. Kusum Arunachalam and Dr. Devashir Datta, followed by the presentation videos of the participants. It's our immense pleasure to have eminent personalities Dr. S.R. Joshi and Dr. Kusum Arunachalam as the chairperson for the last technical session in Biodiversity and Environmental Botany among us. On behalf of Botany Department, GN Khalsa College, Mumbai, I, Babita Rana, extend my sincere gratitude to Dr. S.R. Joshi and Dr. Kusum Arunachalam for accepting our invitation to chair this session. It's my privilege to introduce the chairperson, Dr. S.R. Joshi. Dr. S.R. Joshi is the head of Department of Biotechnology and Bioinformatics, Northeastern Hill University, with areas of specialization in microbiology. Sir pursued PhD from Nehru University. Dr. S.R. Joshi has 33 years of research and academic experience with considerable research credentials. So, in listing a few Sir has published 144 papers at the international and the national level, authored 8 books, 32 book chapters, completed 14 research projects with 2 patents filed, submitted 1542 accession to NCBI database. Under his keen guidance, 23 students have been awarded PhD degree. Sir has received travel grant from ICMR, DBT and DST on research and academic assignments. Dr. Joshi is the recipient of National Merit Scholarship of Ministry of Human Resource Development, Government of India, New Delhi. Sir has been awarded the Best Speaker Trophy by ISRO for the presentation at the Science Communicators Meet during the 97th Indian Science Congress at the University of Kerala, Trivandrum. Sir has been awarded Dr. Ambedkar Fellowship National Award 2009 by Bharatiya Dalit Sahitya Academy. So, thank you, sir, for joining us to chair the session. Our second chairperson, Dr. Kusum Arunachalam, has also connected with us online. Briefing the formal introduction of the Kusum Arunachalam. Dr. Kusum Arunachalam is presently working as professor and head School of Environment and Natural Resources, Doon University. There is a long list of her academic and research achievements. Briefing of you. Madam has completed 24 years of teaching and research experience in forestry, ecology and natural resources management with special reference to Indian Himalayan region. Madam has guided more than 15 PhD students and successfully completed over 20 research projects. She has published more than 100 papers in the national and international journal and several book chapters. Madam has been awarded DST Boycott Better Opportunities for Young Scientists in the chosen area of science and technology postdoc fellowship, and also a DBT Young Women Bioscientist Award. Dr. Kusum is a fellow of the International Society of Tropical Ecology, International Society of Environmental Botanists, the Society for the Science of Climate Change and Environmental Sustainability, and National Environmental Science Academy. She has served as Dean Academics, Dean Environment Science, and is serving Vice Chancellor at Doon University. She has traveled to Germany, Thailand, France, Austria, and Sri Lanka on various academic assignments. Thank you, Madam. Thank you so much for joining us as the resource person as well as the chairperson for the session. So, with warm welcome, I request Madam to share her vast research and academic skill with our attendees. I hand over the session to Madam Kusum. Kusum, Madam, are you there?
Yeah, very good afternoon, Professor Kusum Arunachalam. So nice to see you after a long time. I'm in Shillong and it's been a very bad weather today. You can see it's being dark, it's raining, cats and dogs, very heavy. So sometimes my network may also get broken, so I'll request Professor Arunachalam to take it forward. I'll be there all through, but I think we can proceed. I hand over to Professor Arunachalam to say a few words, and we can move on, I believe.
कुसुम मैडम आप म्यूट पर हो. You have to unmute yourself.
Okay, is it now? Am I audible to you?
Yes, ma'am.
Okay.
Yeah.
Thank you. Thank you so much, Professor Joshi, and it's really nice to see you also. And I could see Dr. Arnab Sain. He's also a colleague from NEHU, our colleague, and some other people also from NEHU have joined. So I'm really happy to be here. And I really extend my thankfulness to the organizing committee, the convener of the program, Dr. Gopal Sharma. And they were Professor Tandon was also there yesterday. I could join for some time, but I was traveling, so I was not there for the entire session. But I was really happy to listen to Professor Tandon almost after 2 decades. And I think in valedictory session we'll be having Professor Veena Tandon also. So it was really a good opportunity that Dr. Gopal Sharma could bring so many people together, and so I'll be really happy to share some of my experiences. So I'm trying to load my presentation. Excuse me just a minute. I think there's some problem. I'm trying to figure it out. Why it's not coming? Pressure, can you please come? Why it's not even showing this. Share this thing. It's not coming. Here it is. Screen share it has not. No.
If you want, you can send it to me.
Can you see the screen? No, just a minute. Just a minute.
Yeah, it's coming. It's coming.
This way.
Good afternoon, Professor Joshi. How are you?
Good afternoon, sir. You've reached home. So nice.
This is Arnav.
Yeah, I can see you. We just made day yesterday, so safe home. Something, sir.
Yeah, I have reached home.
I'll come tomorrow.
It was late and I was very tired, so I didn't call you at the time.
No, sir. So nice.
But otherwise, it is very good.
Thank you, sir.
You and other fellow.
So, madam, you want us to share your screen?
Okay.
Yes, ma'am.
We can see it, Kusum, Dr. Kusum. Dr. Kusum, your screen is visible.
So is it visible now? Sorry for some technical
Yes.
I thought it was on Zoom, but maybe it was on Meet, so that was the problem. So anyway, I hope it's visible now.
Ma'am,
Can I start?
Ma'am, one request. Can you put it on the slideshow?
Yeah, just a minute. I'm doing that. Is it fine now?
No, ma'am. It's not on slide mode.
I think it must be on the slide mode. No.
No, ma'am.
Just a minute. Now?
No, ma'am. You stop presenting. You stop presenting, ma'am.
Just a minute. Now also? Sharing for
I can see Dr. Debashish Datta also has joined. Good afternoon, Debashish.
So is it fine? Gopa, can you see the slide now?
Now you can see, but it is not on the slideshow.
I don't know, but here in my system it is showing on the slideshow only.
Okay, it's okay, ma'am. You can continue.
Okay, so I will just start it. So the topic which I am going to speak today is on ecosystem restoration for biodiversity conservation. And as we all know that nearly 1 million plant and animal species are facing the risk of extinction. And the latest report from the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, which says that we have been able to document only 1.9 million species so far, and there could be almost 18 million species in total which are yet to be documented. And everybody is aware about the biodiversity, what is its role and how it is important for everyone. Biodiversity is providing a lot of ecosystem services, goods, and it's also playing a very important role in the bioeconomy of our different nations. So there's a pressing need for the cooperation action to halt the biodiversity decline. And biodiversity is being lost at a very precedential rate, and we are heading for the last mass extinction, as people are now worried about that. So a decade ago, the countries they united to create a 10-year plan called and that was subdivided into 20 targets for protecting and conserving the natural resource systems of our global planet Earth. And the plan, also known as the Aichi biodiversity targets, they have expired already, and most of the targets we have not been able to reach. So this is the irony that for the last one decade, which was dedicated to the decade of biodiversity, most of the targets are not being achieved. And most measures of the biodiversity suggest that things are going very bad on the ground, and some 1 million plants and the animal species are facing the threat of the extinction. And this is the report by the Intergovernmental Science Policy Platform on the Biodiversity and Ecosystem Services, which we call in short as IPBES. So these achieved biodiversity targets, they were the part of an international agreement, which we call as the United Nations Convention on Biological Diversity. And the member states are now finalizing the replacement for these targets, Aichi biodiversity targets. And these are now being referred as the post-2020 Global Biodiversity Framework. And this post-2020 Global Biodiversity Framework, the new targets are expected to be agreed in the next COP party of 15, which will be held in China in Kunming in very short time period. So the draft of the GBF was published last July, and it is aiming to slow down the rate of biodiversity loss by 2030. And by 2050, it says that we have to value, conserve, and restore and widely use, maintaining the ecosystem services, sustaining healthy planet, and delivering benefits which are essential for all the people. So this next plan, the Global Biodiversity Framework Plan, which is now being which will be launched very shortly, and this comprises again of 4 broad goals and 21 associated targets. Just like in Aichi biodiversity target, we had around 20 targets and 4 goals. Here it is emphasizing on 4 broad goals and 21 associated targets. And the headline target is the main focus of this post-2020 Global Biodiversity Framework is that we have to, include conserving 30% of the land and the sea areas by 2030. And another important challenge which is there is to reduce the government subsidies that human biodiversity, which is harming the biodiversity by almost around $500 billion US per year. So overall, the goals and targets are designed to tackle each of the main contributors to biodiversity loss. And the main contributors of this biodiversity loss, we all are aware, is agriculture and food system, climate change, the invasive species, pollution, and the unsustainable production.
Just a minute, Dr. Kusum, your slide is not moving.
So can you see now? I have just come to the next slide.
No, it is not moving. The slide is in the first slide. We are still in the first slide.
Are you sure? Now?
Dr. Gopa Sharma.
Yeah, tell me.
Yeah, I think Madam has to come out of this presentation mode and keep in the normal mode. Otherwise, it will not be. It will not.
Yes, I think so.
Is it not moving still?
No, now it's moving.
You need to come out from.
I think your presentation mode makes.
Yeah, it is a problem with presentation mode. So you just bring it to normal mode and then scroll, then it will move.
Okay, is it fine now? Can you see the slide has moved to the next?
Yes, yes, yes.
Okay, so that was the picture about how the biodiversity is moving in the global front and how we could not even achieve those targets which were to be achieved by 2020. So the UN Decade on Ecosystem Restoration, which has now been framed on March 2019, the United Nations issued a massive call to action for political and financial supports for restoring the degraded lands. So it has been that we have now understood that about the stake of 3.2 billion people is now being affected by the biodiversity loss, and we have almost 2 billion acres of the area which has been degraded and which can be brought back to its natural state to a certain extent, and that gives a lot of opportunity for restoring this 2 billion acres of land. And this will also enhance in the carbon sequestration potential by removing about 26 gigatons of the greenhouse gases from the atmosphere. So we had this now the United Nations Decade on Ecosystem Restoration, which is from 2021 to 2030. And the entire story started from the Bone Challenge. The Bone Challenge is a global goal which was initiated by the government of Germany along with the IUCN in 2011. And the main focus is to bring about 150 million hectares of degraded and deforested landscape into restoration by 2020. And later on in the New York Declaration in 2014, it was again extended that we have to bring about 350 million hectares of degraded land under the restoration project by 2030. And India is also part to this Bone Challenge, and India has also set up a very massive, ambitious target. And we know that about 96.4 million hectares of the land in India is under the degraded category. So the Indian government has set up a target of the pledge which Indian government has taken in 2015 was to restore about 21 million hectares of the degraded land, and now that was extended in 2019 to restore more land, that was about 26 million hectares of degraded land. So the Bone Challenge will serve as an implementation vehicle for the existing global commitments. Like we have the global commitments of CBD biodiversity target, we have United Nations Framework Convention on Climate Change, UNFCCC, then we also have UNCCD, United Nations Convention to Combat Desertification, and then we have these global goals, the Sustainable Development Goals, which we call them. So this Bone Challenge is trying to actually it is a kind of a vehicle implementation for all this kind of international global commitments. So the global strategy, the 10 years period to restore our planet Earth, which we have degraded over a period of so many years. So how can we do that? So the global strategy is that we have to understand that this is a decade and it is a global movement where we have to bring all the people, like everybody has to be involved in this entire ecological restoration program. So the stakeholders, every individual at own level, they have to do their own actions are very important. So this is very important. So we have to realize that it is a global movement. And another important thing is leadership. The decade will celebrate the restoration leaders and encourage new leaders to step up. So people who are really doing some work on ground on the restoration, they have to be encouraged and they have to be brought on the forefront so that many more people are inspired to take up this kind of activities. Then we have finance restoration. Finance is very important part because all the goals, whatever we try to emphasize, they fail because of the lack of the finances. So the finance restoration is a very important part of this entire ecological decade restoration program. Then we have to shift the behaviors, like the behavior of the people have to be shifted from the degradation towards restoration. So we have been degrading our natural resources with an alarming rate over a period of so many years, but now we have to think about, we have to recreate, we have to reimagine, and we have to restore all those degraded ecosystems. And then we have to celebrate a culture of restoration. So everybody has to realize that how important are these natural systems for our well-being of the planet Earth, and everybody has to participate in this. So the next generation is going to be the generation of the ecological restoration, and many youth, they have to really come up on a large scale to take up this restoration initiative so that all the goals which have been envisaged under this important decade has to be met out. So now let us try to understand what is ecological restoration. Now ecological restoration in a very simple definition which has given by the Society for Ecological Restoration, and it is widely accepted definition. It says that it is a process of assisting the recovery of an ecosystem that has been degraded, damaged, or destroyed. So very simple definition, and this is being followed worldwide. Now, how to differentiate between conservation and restoration? Because people are already doing conservation for a very long period of time. So, but how it is different from restoration? So, restoration ecology, which is actually addresses the subject of restoration, is comparatively new discipline, and it is emphasizing, or it is dedicating to leverage the human intervention which to produce habitat and biodiversity recovery. So here we have to focus in restoration for ecology. The focus is on the restoration of degraded land. So apart from halting the degradation, we also have to do something so that all those degraded lands can be brought back to certain level of their natural conditions. So that is, we call that this discipline as restoration ecology. Then we have conservation biology. Conservation biology is also slightly related to your restoration ecology, but here the focus is to preserve and maintain existing habitats and biodiversity. So here we are trying to just focus on maintaining the habitats and biodiversity, but where in restoration ecology we are going one step further. We are also trying to bring back the degraded ecosystem into their natural status. So that is, then we also have a restoration biology, and the restoration biology is to restore populations which are to be able to persist and evolve within larger communities over a period of time. So this is how the conservation versus restoration we can understand how they differ and how they are similar on the larger landscape. So this is the disturbances. We know that ecological disturbances are there. So many type of disturbances we have seen over a period of time. Natural disturbances, the fires are increasing number, droughts, floods. So for last 2 decades, as the climate change problem has come up, so all these natural disturbances and they are increasing, and the frequency, the intensity is increasing at an alarming rate, and that is a very great challenge for us in this decade of ecological restoration, how to tackle with these problems. So we have developmental projects which are going up. We have invasive species threat. All our protected areas are affected by the invasive species. Then we have air pollution. The air pollution has also come up a big major challenge where we have to really tackle this in this decade of ecological restoration. So if we can revegetate, if we can regenerate our lost vegetation, the air pollution problem can also be tackled to a certain extent. Then we have habitat fragmentation, that is the largest cause of biodiversity loss, and this is the greatest challenge which is lying ahead in this decade of ecosystem restoration. So we know that 80% of the biodiversity loss is because of the human destruction, and we know that how the IUCN has categorized the different species in different categories. We have extinct species, we have threatened species, we have least concern species. So the goal is that how to keep this endangered or this critical endangered species and all the species which are in danger of extinction in their through the ecological restoration, how can we just stop this kind of this extinction of the species which we are facing. So when we talk about the restoration, then the people talk about different types of ecological restoration, whether it is at site level. When you are doing restoration at a very small setting or small area, then we call it a site level ecological restoration. Then we have ecosystem level. But now people are trying to say that researchers have understood that we have to go for large scale landscape level ecosystem restoration, and that is why the forest landscape restoration approach has been very widely appreciated over the globe. And here we are trying to restore the entire forest landscape. So if you restore the entire forest landscape, then you also take care of the other ecosystem, the other degraded sites which are lying within that entire forest landscape. So forest landscape restoration, when we talk about that, it is not simple planting of a single species of tree. So generally when we are going for ecological restoration project, generally what people do, they just go for plantation of some exotic species, but that is not ecological restoration. So we have to understand that it is about restoring the landscape with mix of different native species. So the emphasis should be on the native species, because if you plant the native species, that will give a lot of opportunity for the natural landscapes to come back to the original status. So if you plant the exotic species, that we cannot call as a restoration. So this, because if you have the biodiversity, if you have multiple species, then they also deliver a broad range of forest goods and services to the humankind. We have different kind of people. We have different stakeholders. So we also have to understand their problem, how their dependency is there on the natural systems, and what kinds of goods and services the local communities, the traditional communities which are residing in the vicinity of the forest, they will derive from this kind of landscape ecological restoration process. So the forest landscape restoration is an approach which delivers ecological integrity and the human well-being through multifunctional landscapes. So here it's not that only we are targeting on the goods, but we are also targeting on the human well-being. Along with the human well-being, the main focus is on ecological integrity. So we are also trying to enhance the ecological integrity of the damaged, destroyed, and degraded ecosystem. So in this figure, we can understand like what are different forms of forest degradation and what are different kind of approaches. So here we can see that an undisturbed forest, which is now represented here by the position A. So this undisturbed forest has got certain level of biodiversity, it has got certain level of biomass, structure, and production. And when this undisturbed forest is subjected to extensive clearing, it can lead it to the point B. So point B, where it will have lost a large proportion of its biodiversity, it has also lost much of its biomass, structure, and productivity. So it may or may not recover unaided to C. So it may become secondary regrowth forest, the secondary forest which we see in the northeastern states, where a lot of your jhum cultivation has led to the primary forest being converted into secondary regrowth forest. And then we have some forms of intensive logging. If there is an intensive logging, then undisturbed forest from A can also go down to point B. So point D. So in point D, you have some. You can see that we can have still. It has retained some of its biodiversity. Most of its biodiversity is retained because there will be a lot of seedlings and saplings present in that system. But it has lost most of its biomass structure and production. Now, this ecological restoration, rehabilitation, and reclamation, these are three different approaches when we talk about restoring a degraded ecosystem. So here we can see that if left untreated, the degraded forest at B1. So this degraded forest at B1, if you just leave it untreated, it can go down further in terms of its biodiversity, biomass structure, and production. And it will become B2 because of we are not doing anything. We have just left it unaided. And there may be so many fires, there may be grazing. So the production as well as the biodiversity will be lost further. And then if you seek ecological restoration, if can also seek to return the forest to position A. So if you're trying to, through ecological restoration, we are aiming to bring back this degraded forest to point A. So that is the highest level of restoration which we can achieve at point A, where it has lot of its it has regained most of its biodiversity in comparison to the natural system, and also has gained back its biomass structure and production. Then there's another situation where the point is E1 or E2, which we call as reclamation. So reclamation will lead it to, if you are trying to attempt for the reclamation, then it is trying to, it will regain a lot of its biomass structure and production, which will be much higher than even the restoration. But in terms of the biodiversity, you can see that biodiversity will be very, in terms of biodiversity, it will be very, very poor in comparison to your A and F. And the F where it has this F point, it is a kind of rehabilitation. So rehabilitation means it is a kind of a middle approach, where through our approach, we are trying to regain the lost biodiversity to a certain extent, and we are also trying to regain the lost biomass structure and production. So these are all the different approaches which are there, which we call them as reclamation, rehabilitation, and ecological restoration. Now here we can see the highest quality of landscape restoration. Here we can see at point 1, which is actually where ecosystem integrity and the human well-being both are being improved. So here at point 1, which is the highest quality of landscape restoration, is through improving the ecosystem integrity. So we have to address the biodiversity, we have to address the structure, functional aspects, and then we are also trying to see that it provides some kind of goods and services to the people who are living in the nearby, on those vicinity of this kind of disturbed ecosystem. If you see at position 2 represent a less successful outcome in comparison to position A. So it is a kind of medium approach, and whereas at position 3 and 4 are less likely to be sustained in the long term. So position 3 is where you have a lot of biodiversity, but we don't have any kind of human well-being. Whereas in position 4 we have, it is addressing the human well-being to a very great extent, but it is not talking about the ecosystem integrity. So both this point 3 and point 4 are unsustainable on a longer time period. So the best approach is to go for point 1 and point 2, where we are trying to at least address both ecological integrity as well as the human well-being. So rate of recovery of biodiversity, the rate of recovery depends on the resource devoted to the restoration. And here we can see that when large number of species are planted and substantial resources are applied as represented by position A, the rate of recovery towards the original degree of biodiversity will be very rapid as more species will be colonizing from the outside. Now at point B, we can see that with fewer species and more limited resources, there is much more time before the system crosses a safety threshold where the canopy starts closing and leaving the site exposed to risk of further disturbance such as fires or grazing. So this is a medium kind of approach. And then this third stage we can see, which is represented by C, where we have left the site to recover unaided, means we are not doing any external, our intervention is not there. So in that case, there is an even higher degree of risk and it may or may not recover at all. So these are 3 different categories of recovery of biodiversity, which is depending upon our intervention as well as how we are treating that degraded sites. So this is how we have understood the different approaches. So we have already understood this, that the original ecosystem, if it is neglected, it will be damaged over a period of time. Then if we do some intervention, then we can bring it back to reclamation stage. We can restore it to original ecosystem, and we can also do rehabilitation. So depending upon the situation, what is the goal? The ecological restoration program should aim at that. So that are different approaches to that. So what are the benefits and cost of restoring degraded lands? Now, why we are talking about restoring those degraded lands? So we have to understood that the kind of ecosystem services, the economic value, the total economic value which we are getting from this. So we have to take into consideration the total economic value, which includes the use value, like all the ecosystem services which the particular land is providing to us, and also the non-use value. The non-use value includes your existence values like cultural services and the historical importance of that particular site. So these non-use values, they are very less likely to be dealt with the market, whereas the use values, they are very tangible and they are dealt by the market forces. So that is, and what are the cost of restoration? Now, what are different costs of restoration? So if you are doing restoration, then you also have to pay certain costs. So the opportunity cost is there, which represent the tangible goods and services that were given up to make restoration possible. So if you are trying to restore agriculture field, which were providing you crop yields, or some area which was giving you timber revenue, so we have to lose those. We have to compromise with those kind of opportunity costs. Then we have transaction costs, which represent the cost for landowner and implementing agencies to identify the viable land and negotiate over terms that ensure restoration meets both local and national priorities. Then we also have implementation cost. The implementation costs represent investment in the land, labor, and materials. Because if you are trying to restore a degraded land, then you also have a lot of funds are required, a lot of investment is required in terms of the money. So these are some of the cost-benefit analysis. Then how can land be restored? What are different approaches for restoring the degraded land? So with my large experience of working in the Himalayan region, both in Eastern Himalaya and now currently in the Western Himalaya, we see that there are different approaches can be applied in restoring the degraded land. For example, if you have agriculture in the Himalayan region, in the Himalayan states, this is not very beneficial as the land holding is very fragmented and the soil is very poor in terms of the nutrient organic carbon. So we can bring this agriculture into the agroforestry if the provisions are existing there. And then we also can poorly managed woodlands through well-managed woodland, which is REDD strategy, reducing emission from deforestation and degradation under the UNFCCC framework on mitigating climate change. So that can be another approach for restoring the degraded lands. Now the soil degradation. So the most important aspect when we are going for ecosystem decade on restoration of degraded ecosystem, the soil degradation, which is a very challenging problem, and the soil organic carbon. So the researchers are now trying to say that we have to include the soil degradation, especially the soil organic carbon, into this planetary limit concept, which includes other parameters like climate change, novel entities, ozone, aerosol leaching, etc., loading. So in this, the scientists are now also advocating that the soil degradation can also be taken as one of the parameter for understanding these global problems at a larger scale. So when we talk about this United Nations this Sustainable Development Goal, so the target 15.3 of this United Nations 2030 Agenda for Sustainable Development, it is also aiming to halt the world's net land degradation. So they are trying to see that land degradation neutrality. We have to totally stop the land degradation, and every country has to try for land degradation neutrality. So healthy soils are both are very important for this, because they are the most important natural resource, and they are also, I mean, they will be in the center stage for meeting most of this global biodiversity goals, whether it is the food or the water, energy security, biodiversity conservation, climate change mitigation, and the healthy soil. So the entire this gamut of problems are now being addressed through this. If we can address the soil degradation or soil organic carbon, soil organic matter problem in the degraded sites. So the researcher has estimated that restoring only 12% of degraded agricultural land by 2030 could also boost smallholders' income by about 35 to 40 billion per year, and also help to feed an additional 200 million people annually. So if we can restore only 12% of degraded agricultural land, we will be able to cater to meet the food security requirement of the growing population. So that is very important agenda. So in India, we can see that almost 30% of the India is facing the threat of land degradation, which is very rampant here. And we have lost about 1.87 million hectares of area have been further degraded within the last 1 decade. And this is almost equal to the size of Nagaland, that this much of land area which has been degraded in India. And what are the process of soil degradation? What are the drivers, threat, and consequences? The most important driver are population growth, urban expansion, the dietary changes, industrialization, unsustainable agricultural practices, waste disposal, deforestation, and climate change. And the threats are soil organic carbon loss, biodiversity loss. Then we also have problem of nutrient imbalance, acidification, erosion, pollution, and the consequences finally are ecosystem landscape deterioration. And then food insecurity, quantity and quality, both climate change, violence, poverty, migration, all kinds of social inequality also has to be addressed if we are trying to address this land degradation. So the important strategy should be to avoid land degradation, to reduce land degradation, and to reverse the land which has been already degraded. And this ecosystem restoration decade is going to play a very vital role in doing, trying to achieve all these targets of avoiding, reducing, and reversing land degradation. So soil health restoration, the soil health restoration becomes very important tool if we are trying to recover or restore our degraded land. And the soil health restoration, actually, it is trying to improve the maintaining soil ecology. We have to improve as well as maintain the soil ecology, improve soil organic matter. We have to control the soil erosion. And then all kind of problems are there, so that also has to be addressed through the soil health restoration thing. So once disturbed, succession begins. So this is a natural process, that disturbance, degradation, that is a natural processes. But we have to see that how this because when any kind of disturbance takes place, the natural succession takes over and it tries to reclaim or restore the degraded landscapes. So here the ecosystem, when we are trying to understand the microbial component, the fungal bacterial ratio plays a very dominant role. So as we see that during the mid section of the ecological succession process, this fungal to biomass, bacterial biomass ratio becomes maximum. So we have to address in this restoration program, we have to see that how the mature forest system, we have to shift the trajectory by means of establishing fungal dominance at an early stage. So if we are trying to establish the fungal dominance at the early stage, then we can at least hasten the process of ecosystem restoration to a certain extent. So this is one strategy which is being tried by the people. So we know that soil biodiversity, which is very important, and it plays very important role in this ecological restoration, entire movement of ecological restoration, because that is also driving the carbon, nitrogen, water cycle upon which all the life on the planet Earth is depending. The productivity of land is therefore very important, which is determined to a large extent by its soil biodiversity. So the soil biodiversity, when we are talking about the biodiversity conservation, it's not only the plant biodiversity or the animal biodiversity, but the soil biodiversity, which has to be really understood, and it has to be its role in how it can play a very critical role in ecological restoration program, that has to be understood and applied at a larger extent in the restoration projects. So we were trying to understand this, how this glomalin, we have taken up a study here. One of the PhD students, she was trying to understand how this glomalin, which is a type of glycoprotein produced by the arbuscular mycorrhizal fungi, which belongs to the phylum Glomeromycota, contributes to the mitigation of soil degradation. So here we can see that this is a colony of this arbuscular mycorrhizal fungus glomus species, and its hyphae and vesicles, arbuscules, we can see from this. And so this particular fungus is classified as a member of the Mucormycota subkingdom and the phylum Glomeromycota, which includes 3 classes: Glomeromycetes, Archaeosporomycetes, and Teglogromomycetes. And this AMFF belongs to 11 families, 25 genera, and it has about nearly 300 species. So vast majority of the member of the phylum Glomeromycota fungi are obligate symbionts, that is, they depend on the carbon substrates which are provided by the host plant to survive. So here we can see the structure and function of the glomalin, how its structure looks like under this in the soil, and what is this glomalin related soil protein. This is a kind of a glucoprotein, and its chemical structure. So the most important function which it plays in the soil is it improves the soil health and fertility, which is then plant productivity, rhizomes, sheet formation, seed germination, root growth, and nutrient uplift. So it is playing very critical role in soil aggregation. So soil aggregate formation, and when the soil aggregate formation improves, then soil structuring also improves, and when soil structure improves, the carbon sequestration is also enhanced. So people are trying to understand how this glomalin can play a very critical role in sequestering the soil carbon, because this the hyphae of this fungus, which is not degraded over a period of time, it remains there in the soil and is able to sequester, to retain carbon for about 30 to 50 years in its hyphae. So this is a strategy that how we can take the glomalin for restoring the degraded area. So we were also trying to understand this. And here we also try to see how this aggregate stability in different land use system in the Uttarakhand Himalaya, and how this was related to the glomalin content, the total soil, the total glomalin content and the extractable glomalin content. We tried to correlate this, and we found that this forest, in the different land use system, how this glomalin and its aggregate stability were interrelated with each other. So this was another approach which we are trying to see is how we can restore the degraded land is through bamboo. How bamboo can be used for restoring the degraded land, and we know that bamboo is it has a very effective root system which brings the soil, binds the soil together, and it's very fast growing. Then it also gives a lot of ecosystem services, like water regulating services are very important. And then bamboo is very important because it is playing very critical role in the poverty alleviation. So if you talk about northeastern states or even the Uttarakhand, most of the people, they are dependent on this bamboo for a very large extent, and it can be used for as a livelihood enhancement option also. So this bamboo restoration in both Himalayan states, whether it is northeast or Uttarakhand, can play a very critical role. So we were trying, in the large, when I was doing my PhD work, and later on when I was in Arunachal in Northeastern Regional Institute of Science and Technology, so we were trying to understand how this microbial processes in the soil, like you have soil microbial biomass, how they play a very critical role in the rehabilitation of dune lands. So that a lot of work was done there, and we tried to evaluate. Then later on in Itanagar, Arunachal Pradesh, we also tried to understand how the evaluation, we try to screen the bamboos which could be used for the eco restoration in the dune fellows, and depending on their performance in the soil, how much nutrient they are able to provide, how much microbes, microbial component, microbial biomass they were releasing, they were contributing. So we found that this Bambusa nuten, which was a very important bamboo which could be used, and we found that it was very important and it could be used for rehabilitating the degraded dune fallows, especially with reference to the soil nutrient enrichment. So that was another study. So now one more approach we can look forward for the ecosystem restoration during this dune and decay ecosystem restoration is agroforestry. So agroforestry, again, in the hilly terrain in Himalayan state, it can be very beneficial, because we know that agroforestry, whether it is the commercial plantation or it is the traditional agroforestry systems, They are very important because they can generate multiple livelihood options and also provide simultaneously the multiple environmental benefits. And because of that, only the IPCC, they have also said that agroforestry could be one of the most viable mitigation option to combat this climate change problem. So we can use this agroforestry also. So why agroforestry? Because it promotes resilient cropping and farming environments. It can sequester carbon to a very larger extent. And beside that, it is also providing some other this livelihood option requirement, livelihood opportunities for the people who are living in the vicinity of the forest or in the degraded mountain slopes. So this agroforestry can be very beneficial and viable option for restoring the degraded lands. So here, this figure shows that how much total potential area in our country can be there for bringing that area under the total geographical under this agroforestry. So based on that, we also tried this. This is that we all now India is the first country which has a national agroforestry policy also in place in 2014. So it gives a lot of emphasis that agroforestry has to be a very key driver for addressing the problem of ecosystem degradation, especially the soil degradation, and also providing the other beneficial requirements which are for the local people. So to address the problem of livelihood and other things. Soil erosion, in soil erosion reduction also the agroforestry system can play a very vital role. So we were trying to understand this, and we found that how much soil reduction potential the agroforestry systems have. So this is another study which we are trying. And also we have a lot of traditional agroforestry systems which people have been raising for their fodder and fuel bill requirement, and this figure shows how much percentage of geographical area is available which is under the agroforestry in different 13 districts of Uttarakhand. And this is a figure which shows that the current fallow, so we were trying to see how much current fallow land is there in Uttarakhand which can be brought under the agroforestry system. Similarly, this is here we can see that the area under erosion, so a lot of area under erosion is also there which could be brought under the agroforestry system. So we were trying to understand through this land use land cover mapping and using the remote sensing and modeling aspect that how much land suitability, how much land is suitable for bringing the degraded area under the agroforestry cover. So we were trying to see that, and here we can see that the highly suitable land, almost about 294 square kilometer area is there, which is possible to be brought under the agroforestry system. Then we have this yellow point is representing the moderately suitable area which can be brought under the agroforestry system, and then we have the red dots, they are representing the least suitable area under the agroforestry system. So this is the study which is undergoing, which says that in Uttarakhand also, that Uttarakhand, because there's a lot of problem of migration, the people have migrated over the period of time, and most of the villages have been left. They have left their villages, have been migrating to the cities, and they have been leaving their abundant land. So in this abundant land, there's a lot of scope for the agroforestry, because if we can reclaim or restore this degraded abundant land through the agroforestry systems, so that will also help in mitigating the climate change, carbon sequestration, and also if we can bring some horticulture systems into this agroforestry system approach, then there's a lot of scope for the horticulture in the Uttarakhand states. So that is also we are trying to see how we can do that. So with that, we were also trying to, so based on our whatever research we have been doing on the research, the applied and the basic research, we also try to see that how we can do this in practice on the ground. So we have taken up some activities which have been initiated by the School of Environment and Natural Resources, and this particular project has been funded by the Hans Foundation. And here we are trying to restore the degraded land. So we have taken up some area in 4 districts: Tehri, Garhwal, Almora, Champawat, and Pithoragarh. And based on the 8 principles which underpins the ecological restoration, which have been given by the Society for Ecological Restoration, which says that we have to engage the stakeholders. That is the principle 1. The principle 2 says that you have to draw on many types of knowledge. So you have to take into consideration the ecological knowledge of the local people, the traditional people who are living there, the scientific knowledge. So the knowledge of different types of knowledge has to be integrated, and then we can really do the good ecosystem projects. And third principle, it says that ecological restoration program has to be informed by a reference ecosystem. So you have to have a reference ecosystem, that system which was there earlier, in what stage it was there. The ecosystem restoration also supports ecosystem recovery process, because if you are doing the ecological restoration, then you are also supporting the recovery process. Then you have clear goals and objectives. That is the first principle. Sixth principle says that you have to seek for the highest level of recovery. So we have to really aim for the highest level of recovery. And then the seventh principle says that ecosystem restoration gains cumulative value. So if you are doing the ecosystem ecosystem restoration, then it will become successful only if you do it as a larger scale, that is a landscape level, instead of the small site level or the ecosystem level. And the eighth principle of the ecosystem ecological restoration, which has been given by SER, Society for Ecological Restoration, it says that ecosystem restoration is a part of continuum of restorative activities. So you have to have, it is a kind of a continuum process. So if you are trying to treat a site, then if you are trying to treat the ecosystem, and you are trying to treat a larger landscape level, so it will be a kind of a continuum. So with this, this is another figure which says that social benefit wheel. So when you are doing some ecological restoration project, then you have to also have to address the social benefit wheel, where you will be having the community well-being that has to be addressed. Stakeholder engagement is very important, and how much benefits are being you are driving the benefits, how equally they are distributed among the community, then knowledge enrichment. So this is a kind of a wheel which can track the progress of your ecological restoration program. So with this in keeping that in background, we are trying to collect some seeds of the natural tree species which are growing in the Himalayan region in the upper landscape, and we are trying to establish the nursery of these seedlings. Because what happens whenever we go to the forest department, then they are not having any of this seedlings, saplings to give to the local people. So what we have said, but we are trying that we are just creating the nursery. The people are being involved in collecting the seeds from the wild, and then creating the nursery in their villages. So with this project we have just started after the pandemic, because the fund, although it was there in 2021, but because of pandemic we could not start it. But now we have started, so we are having a lot of community engagement, or people are being engaged, and the staff which is working in this project, like we have about 14 to 15 people who are working in this project. So out of 15 people, about 10 staff is from the local community only. So we have involve the local community totally in this restoration activities, and this is how we are trying to reclaim the springs and natural water bodies which are there in the Uttarakhand, because in Uttarakhand also, again, because of climate change, we are facing a lot of problem in the water resources. And when you do any kind of restoration project, the water is very important critical factor. So this one panchayat, there's a lot of one panchayat, which is a community conservation program, which is very unique to the state of the Uttarakhand, where every village has got its own one panchayat. With the local people are custodian of that one panchayat. So we are trying to trying to see how this one panchayat, which have been degraded over a period of time, can be again restored back to their natural status by transplanting the natural seedlings which were growing there over a period of time. So this is how we are also involving some kind of scientific knowledge, how the nursery beds are being prepared by the people, the local people there, and this is how, yeah. So the monitoring and evaluation is very important part. So we also have periodical monitoring and evaluation of this, how our project is working on the field. And this is, you can see here in picture, we are also trying to introduce some kind of income generating activities apart from restoring the forest and the water bodies. Like we are seeing how the vegetables, because the Uttarakhand at the higher altitudes, the vegetable which grow there, they are very they are very rich in the nutritional constituents. So if we have this spices, the ginger and turmeric. So all these spices which are growing in the high altitude, they fetch a lot of money, and then they are organic in nature. So we have also integrated some income generating activities by introducing the spices, especially the ginger and chili, and because again in Uttarakhand, if you see that there's a lot of problem of this wild animal and human wildlife conflict, because a lot of animal are there as the forest has been degraded. So we have to see like which crops, the crops which are not being damaged by the animals, especially the monkey and the wild boar, that can be introduced in the restoration program. So our objective is that apart from just restoring the degraded forest landscape, especially the one panchayats and the water bodies, we should also bring the people, stakeholders together so that the income generation activities are also addressed. So if there is no incentive for the local people, then they will not be, I mean, their momentum in this eco restoration project will not be kept for long. So that has also to be addressed. So this is the figure which says that nature positive by 2030. So there are a lot of goals which have to be achieved by 2030. So everybody has to work together and everyone has to do their bit. And this is, I would like to show this. So this is ecology is permanent economy. So the late Sundarlal Bahuguna, Padma Vibhushan Sundarlal Bahuguna ji, who we lost in last year in the third COVID wave. So he used to say that ecology is permanent economy, and now we have realized that how important what he used to say about 40 to 50 years back now, how how pertaining it has become in the present context. And one of the district which we have chosen for this ecological restoration program is also Tehri, where his Sundarlal Bahuguna ji's ashram in Siliara is located. So we are also trying to see if we can do to that area where his ashram and the entire ecology program was being launched by the Sundarlal Bahuguna ji in late 70s and 80s. His life was dedicated for preserving the ecology of the Himalaya. So with this, I would like to end my presentation, and I'm really sorry for the initial glitches, technical glitches, which delayed this presentation, and I might have extended my time also. So I'm extremely sorry. So I would like to just finish by saying that let's nurture the nature so that we can have a better future. And I would like to thank all my team of the researchers who are there with me at School of Environment and Natural Resources, the students and some of the faculty members. So this is the combined effort of each one of them. Thank you so much.
Thank you, ma'am.
Thank you. Yeah.
Yes, sir, you can continue.
Okay. No, I just wanted to congratulate Professor Arunachalam for a wonderful talk, so informative, because we see a lot of students who are there, and this was basically, from science, deep science that he has done to classroom information. It is very educative. Congratulations, and wonderful, Professor Arunachalam. Yeah. Please, may see carry on.
Yes, sir. Any query from the participants? Any query from the participants? Dr. Kusum, madam, I have one query what steps can be taken where mining operations are high? How to restore the land in that particular area?
Pardon, can you be a bit loud? I'm not able to listen. Hello?
Hello, ma'am.
Yeah, what is the question? Can you please repeat it?
Ma'am, my query was that how we can restore the land, what steps we can take where the mining operations are high?
Yes, mining, because at mining, like if you are addressing the mining area, that will be the site-specific approach. And mostly I have seen that generally in the ecological restoration program, they do the site-specific approach only. So what you have to address will be the most important will be your soil. So if until and unless you restore the soil, then your entire efforts will not be successful. So you have to see that how the local soil biota can be enhanced. So if you can enhance the soil, local native soil biota, which can add to the soil organic carbon, soil organic matter, so that will also provide you all the beneficial nutrients. So that will be the most important approach, and that can lead to the improvement in the site condition, which can then support the above ground vegetation of plants and other communities. So the soil biota, I would emphasize on that. So, and that is what the people are now understood, that we have to address the soil biodiversity aspect to a greater extent if you are going for any kind of conservation or ecological restoration program.
Yes, ma'am. Thank you so much, ma'am. Any other query from the participant side?
Yes.
No, so we can conclude the session. Thank you, ma'am, for your excellent presentation on the topic ecosystem restoration for biodiversity conservation. A very informative and eye-opening session. As Madam said that 80% loss is due to human destruction, so we have to come together to initiate the collective efforts to restore our ecosystem for a better survival. Ma'am, thank you once again for this enlightening session. Thank you, ma'am.
Thank you so much for the opportunity. Thank you.
Yeah. Now I hand over the session to Dr. Shweta.
Thank you so much, Babita ma'am. A very good afternoon to everyone. Now I would like to introduce our next plenary speaker, Dr. Debashish Datta sir, for this last session, technical session of today. The sir is a senior academic staff in Toi Ohomoi Institute of Technology, New Zealand. Sir is a faculty member in environmental management and has a strong interest in plant ecology along with biodiversity. Dr. Datta also has a keen interest in the impact of invasive species on the native ecosystem. Sir has born in and educated in Shillong in northeast India, and he has taught at the tertiary level of several years in India and now teaching in New Zealand since 2003. Sir strongly believes in the applied aspect to teaching and research, whereby knowledge gain can be useful for the betterment of the environment and its related problems. Sir has also had secondments with Auckland University of Technology and University of Waikato, Hamilton, and worked in biosecurity for the New Zealand Ministry for Primary Industries. Sir has also delivered a talk on the impact of introduced herbivorous mammals on the regeneration of native plant species in New Zealand. For this brief introduction, I am welcoming Datta sir for their lecture. Welcome, sir.
Thank you for the introduction, and greetings to everyone from Tauranga and the Bay of Plenty in the east coast of New Zealand. So yeah, good afternoon to all of you who are tuned in from India, and probably good morning and good evening to others who might be in other parts of the world. So I'll hopefully the technology works. If it doesn't, can't see a thing, let me know. And just make sure, just give me a second over here. So can you, don't know, can you see?
Yes, sir.
Can you see my slide? Okay.
Yes, sir.
So I'll give you a glimpse and a quick summary of a little bit of the work that we are doing here at the moment. And it's got quite a New Zealand flavor to it, which I thought would be useful because the New Zealand ecosystems, as most of you are probably aware, are quite different to ecosystems on continental landmasses. Not just because it's an island ecosystem, but also because it's an island which has evolved in relative isolation for prolonged periods of time. So just to give you a bit of a background, can you see the change in slides?
Yes, sir.
Okay. So just let me know if we have any glitch over here, and I can try and reboot.
Sure.
So just to give you a bit of a background, you know, the New Zealand's terrestrial ecosystems, being, as I said, an isolated archipelago of islands, they have evolved for more than 65 million years since they broke off from Gondwana land in isolation, and therefore these ecosystems have had very little impact and mixing about us from other continental landmasses. And therefore, not surprisingly, you've got a very high degree of endemism when it comes to both the plant life as well as the animal life. So if you look at the botany and you look at the plant life, then you've got around 2,300 species of vascular plants, which, yes, does seem, you know, in comparison to continental land masses, relatively confined and small. But what we need to remember is two things. One is that in island ecosystems, you'll obviously get lower diversity than on continental land masses. Plus, New Zealand happens to be in the temperate part of the globe. So that obviously also influences the biodiversity to some extent. But what might be worthy mentioning here, just for comparisons, is if you compare the New Zealand biodiversity with biodiversity of, say, for example, Japan or the United Kingdom, which are also island ecosystems of more or less similar sizes, then you find that the diversity here in New Zealand is much, much higher compared to those comparable land masses. And what you have is, like I said, obviously a very high percentage of endemism. So around overall an 80% species of plants are endemic. And then if you break it up into angiosperms and gymnosperms, then you find 85% of native angiosperms are endemic. And when it comes to gymnosperms, it's 100%, which obviously is, a number which is which cannot be exceeded. So when it comes to gymnosperms, there are two main groups over here, which are very, very dominant. So one is the podocarps, so Podocarpaceae family, which is obviously Gondwanic in origin. But the other one, which is this image at the bottom right of, sorry, the bottom left rather of the screen, which is the Araucariaceae family, which again, as you probably know, in the Western Pacific, has a very strong presence. So you've got these ecosystems where you've got high endemism of plants, but the other interesting thing is that prior to human settlement, there were no terrestrial mammals here in New Zealand except for a few species of bats. So you've got a relatively large ecosystem with no large mammals at all, which again is very, very unique, but has shaped ecology a lot, especially when you think of what has happened in the last 200 years. So in the last 200 years, when European settlement has been very rapid, a range of exotic mammals have been introduced from different parts of the world for a variety of reasons. So some of them could have been introduced for farming, some, for pets, which have subsequently escaped from domestication and become feral. But a lot of them have also been introduced just because there were all these empty niches in the ecosystem, which could be filled. And when Europeans settled here in New Zealand, they realized that they were missing a lot of these species which they're familiar with from back home. So a lot of these larger mammals, like deer, like multiple species of deer from different parts of the globe, were introduced, along with other herbivores like goat and chamois and tar as well from the Himalayas. So once these larger mammals were introduced, as you can imagine, they had no natural predators. Plus, they had a food source, that is native plants, which had no chemical defenses in them, right? Because they had not evolved in close proximity to browsers. And therefore, the population of these introduced mammals exploded and today have reached pest proportions in many, many places. And what we find is therefore that hooved mammals like ungulates, for example, like deer and goat, they are having a severe impact in forest ecosystems because of the pressure that they create on browsing of plant species, especially on the forest floor. And what you can find is that you could have a situation like the image on the left, which is how it should be in the absence of browsing pressure or minimal browsing pressure, where on the forest floor you've got a healthy diversity of native plant species, which are germinated from the seeds and fruits which have fallen from the trees or the canopy above. But because of this browsing pressure, which is intense, it can sometimes get to a stage where from that kind of a situation, you can get a forest floor which is almost totally barren and bare. And the first question that we'd want to answer is, okay, what could be responsible for this kind of browsing? And we suspect, that it could be ungulates, but this is where we can take the aid of technology and install some trail cameras, because most of this browsing usually takes place at night. And the reason is because most of these deer species which have been introduced have become very, very wary of humans because they hunted, not just for sport, but also to control their populations. So what happens is during the day, they don't want to expose themselves to danger, and will only come out and browse in many places only at night. So if you put up trail cameras, then you could very well confirm what is responsible for causing this kind of damage, right? So now you've got a situation that we need to rectify, and you've got a culprit, right? So what are some of these implications of this very intensive browse which is happening in most parts of the country? What we do find is that when they browse through the understory, it's very much like, picking up for you and me picking up food at a buffet. So like we have our preferences, so these animals also would obviously have their preferences. And what we find is that they have, at one end, a very, very strong preference for some plant species, which they find highly palatable. So those are the ones which they will browse first. And then once those are gone, they move down the hierarchy and looking at those which are probably intermediate in their preferences, right down to the lower extreme, where they're the ones which are of low palatability, the ones that they don't really like to browse at the first instance. But if the high and medium palatable or preference ones are gone, then they might browse on the low palatability ones. So what we find is if ungulate densities are low, that is the browsing pressure is low, these browsers will selectively browse their preferred species. That means the ones which have high palatability. And therefore, what you'd find is there's still seedlings and saplings on the forest floor, at least to some extent, but the species composition starts to change. And the ones which we are able to identify are the ones which are the low palatable species, right? So what we might find is if we take a snapshot in time, we might find that the numbers of these saplings do not appear to be significantly impacted, right? But like I just mentioned, if you start identifying the species that are left over, then you find that the species composition of these saplings have been skewed, and skewed very, very strongly towards low palatable species, right? And what this will do, as you can imagine, is this will significantly impact species composition and therefore long-term forest structure. So once these larger trees, the established trees, start to collapse, what we find is that for many of them, they don't have juveniles to replace them because the juveniles, if they're highly palatable species, have already been removed because of the browsers, right? So what do we do to try and protect these forest ecosystems? So what is happening is when ungulate populations increase, that is the browsing pressure increases, control measures are implemented. So the government organization here in New Zealand, which is responsible for taking care of conservation land and of forests, is the Department of Conservation. So what they do is they contract out hunters who will basically go out either in helicopters and shoot deer which are above the tree line in hilly country and therefore can be viewed easily from a helicopter. So they will shoot them or cull them in large numbers to keep their populations down. If it is within forest ecosystems where a helicopter is pretty much useless because you can't see them through the canopy, then they will have to hunt them and cull them on foot right? And as you can imagine, this is easier said than done, because not only is it time consuming and difficult, but you will never be able to eradicate these pests. Or the best you can do is bring numbers down and keep numbers down for a relatively, probably a small portion of time before they breed and the numbers start to increase again. And you have to do the whole thing all over again. Whereas the other problem associated with this kind of an operation, as you can imagine, which involves helicopters and hunters and specialized permits required, etc., and closing of large areas to the public, if you've got people out culling deer, is that it can become extremely expensive. But one of the first things that we need to do is to determine if there is actually an increase in the density of these pests, right? Is it worthwhile? You need to do a cost benefit analysis. I said, okay, if this is what is going to cost us to bring in hunters to cull deer populations and decrease their numbers to try and protect forest regeneration, we first need to justify that actually the deer numbers are high enough, right? And how do we do this? So a quick way of doing this is by looking for signs, so indirect signs of their densities. So using things like fecal pellets as an index of density of deer, or signs on trunks and branches of trees of deer where they rub their antlers, the male deer that is, and they leave these scars on the trunks of trees. Or, like we just mentioned, you could look at sapling abundance and diversity, right? The problem is that all of the above, especially the first two, can be misrepresentative of actual density and impact of pests. Because as you can imagine, both fecal pellet counts and signs of their activities can be influenced by a large number of factors which are there in the environment. So this is where they came up. So two of these researchers, Sweetapple and Nugent, who work for Landcare Research here in New Zealand, they came up with this method called a seedling ratio index, which looks at not only considering the number and diversity of saplings, but looking both at seedlings and saplings. And to try and answer this question on how abundant these pests are in the environment and when would be a good time to go out and actually deploy these control measures. And why is it considering both seedlings and saplings? Why can't we just look at saplings? So the reason is because studies here in New Zealand have shown that these browsers, because they're larger mammals, they're taller, when they bend their necks and they're browsing, they obviously find it a lot easier to browse the saplings, which are taller, right? Whereas the smaller seedlings, which are less than 30 centimeters in height, they just leave them aside because it's not worth their while. Not only because they're much shorter, so it requires more effort to bend down and browse them, but also because they're much smaller. So it's not worth their effort because the amount of biomass that they can get from the seedlings is not significant. So what we did is we adapted this approach by Sweetapple and Nugent to try and answer a few questions in the local forest ecosystems here in the Bay of Plenty. And like we say, no work, as you all know, no work is done by one person. And I'd like to acknowledge two of my students here who have done a bulk of this work and continue to do so. And so Rio and Nicholas, Rio Miyanami and Nicholas Sharp deserve special mention here because a lot of this data which has been collected has been done by them. So what we did find is we looked at two forests. I won't bother about pronouncing those names. Those Maori names can be a bit of tongue twisters. But these two forests, which are in the Bay of Plenty, and we selected these two forests because there was anecdotal evidence, right, without specific surveys being done on deer densities. So anecdotal evidence which says that both those areas had more or less equal number of deer. So they thought, okay, they're low to moderate deer densities, so we don't need to worry about them. So we said, all right, let's try out this proposal that we had and look at it from the saplings and seedlings and see what the saplings and seedlings are telling us about this anecdotal evidence that you're providing, right? And as you can see, you have a very, very different picture in the two forests in the area. In the first one, as you can see, almost 60% of the tall seedlings here refer to the saplings, by the way. So almost 60% of the saplings were the high preference saplings, right? So which means that the browsing pressure must be very low in that first habitat. And if you compare it with the second, only around 20% of the saplings in the same category are high preference, right? So it gives a clear indication that when you compare it with seedlings, the saplings are being browsed at a much, much higher level at the second forest, not just for high, but also for moderate, right? And if you flip the coin, that just confirms our suspicion. So if you look at the low preference, which is the darker green, then you see in the first forest, only 17% of the saplings are low preference, right? And that's simply because there are so many high preference saplings still around, so they take up the vast majority of the saplings, 60%. Whereas in the second one, more than 50% of the saplings are low preference, right? And please remember, I just wanted to clarify, this is not reflective of what the seedlings are telling us, right? So when it comes to seedling diversity, they are quite uniformly distributed in both forests. But when it comes to saplings, it's quite a different picture, simply because the browsers come into the picture, right? So from a situation like this, a snapshot in time, we can go back to the authorities and say, hang on, you know, your anecdotal evidence is not what the saplings and seedlings are telling us. So we have clear evidence here that your browsing pressure is much higher in the second forest as opposed to the first. And therefore, you need to implement ungulate control in the second forest ASAP. Otherwise, things are going to go downhill very, very rapidly, right? Taking this further, the same two forests, we looked at, again, the ratio of seedlings to saplings. And again, as you can see, you've got, again, when you look at high preference, the first forest, much higher ratio of saplings to seedlings, right? Whereas in the second forest, the ratio is much lower. If you look at low preference, the picture is just reversed, right? So again, it is confirming what we suspected in the first case by using a slightly different approach and not just looking at saplings, but taking a ratio over here, right? And we ran a couple of regressions. And again, if you can see in the first forest on the left, very, very obvious that you've got a very strong correlation between the tall seedlings, which is basically the saplings, and their species richness in relation to the short seedling species richness. In other words, if you've got higher diversity of seedlings in the first forest, you're also getting a higher diversity of saplings in that first forest, right? So you can see the p-value obviously is very less, right? 0.0006, so it's very significant, right? The R-square may not be that high, but the P is really good, which is what just confirms our suspicion. And the R, you're getting your R of 0.7, which isn't too low. Whereas if you look at the second forest, there is no correlation at all. In other words, even with increasing short seedling richness, you do not get an increase in tall seedling richness. And we can guess why. If you go back to that first two images, it's because the browsing pressure is obviously a lot higher over there than in the first forest, right? So therefore, to conclude, how could we use this kind of existing approach? So just to clarify, Sweet Apple and Nugent's approach, the seedling ratio index, that's not new. It's been around in New Zealand for a few years, devised here and used here widely. But what we are trying to do is we are trying to adapt it to try and tell us the same story, but from a different perspective. And more importantly, to confirm things which anecdotal evidence of deer densities or other measures of deer densities would not be able to confirm without this kind of a data. So this kind of an approach would enable us to do what? Would enable us to do three things. Let me just sum this up very quickly for you. So first is to determine browsing pressure, right? And that is instead of angular density. So the reason why I wanted to stress this very clearly is, please remember again, if you go back to the initial slides, we can get an indication of angular density without looking at browsing pressure, and therefore call in the forces and bring in control. But like I said, there are two problems with that. The first is those indices of browsing densities or the browser's densities are not really very accurate. Okay. And the second thing is, like I said, it doesn't really matter if your angular density is high or low, because at the end of the day, what you're trying to protect, the resource that you're trying to protect are the seedlings and saplings. So you could have low, relatively low angular density, but if your seedlings and saplings are still getting hammered, then it doesn't matter that the angular density is low. You still need to bring in the reinforcement and still need to tackle those angulates at that stage. Right? I hope that makes sense. Right? So this is what it can help us. So at a site, at any site, it can help us determine browsing pressure and therefore determine whether we need to do something immediately, or we can afford to use the resources that we have got at another site which might warrant more action sooner than this particular site. The second is it enables us to compare different sites. So the example I showed you was of 2 sites, but you could have multiple sites, because this kind of an approach is quite time efficient. So you could collect a large amount of data within a relatively short period of time without having to worry about trying to find out what the density of angulates is, right? And like the past example, you could then prioritize which site or which habitat needs control to be brought in sooner rather than later, right? And the third thing, which we've found through our studies is quite interesting, and again, some of you have probably guessed this while I'm speaking, but we wanted to confirm our suspicions, is it can confirm whether ungulate browse is in fact the problem, right? So you could have trail cameras out at night. You could catch them red-handed browsing on seedlings or saplings, but it is only when you compare the species richness of seedlings with the saplings that you know for sure that it is indeed browsing pressure which is removing the saplings from the habitat. Because you could get a situation, which I've summarized in point 3, that there are no saplings of high palatable species, but there are also no seedlings. So if there are no seedlings, then you know that's only naturally you're not going to get saplings, right? But the question that you then need to go back and ask is, why are there no seedlings, right? You've got mature trees in the forest, by what? Why are there no seedlings? And the answer, as you can guess, is that the problem or the lack of plant regeneration is likely originating elsewhere. So yes, you might have some browsing pressure, but that probably is not your priority. So for example, the problem might be in the canopy, right? So for example, along with the deer and goats and all these other animals which have been introduced, some of you might be aware that in New Zealand, in the last 200 years, another group of animals which have been introduced are possums from Australia. So possums are marsupials, they're arboreal marsupials, and they are herbivores. So they do extensive damage, unfortunately, to the canopy of trees in areas where they're not controlled, right? And when there is extensive herbivory in the canopy, as you know, photosynthesis rates will drop, and what that will affect is flowering and fruiting. So you could get a situation where a canopy may not look extremely damaged from the ground, but it is damaged enough to reduce photosynthesis to a level where it cannot produce enough fruit. And therefore, it's not shedding enough seeds on the forest floor, and therefore you do not have enough seedlings. Okay? And I think that's the third point is really key to identify, which our studies have helped clarify as well, because sometimes you're backing up the wrong tree and you're blaming it all on deer and goats and other herbivores on the ground, whereas you're totally missing the fact that the problem is starting somewhere else. Okay, so that's all I had to say. Hope that made sense. And if there are any questions, yeah, let me know. But otherwise, yeah.
Any queries from the participants?
Madam, may I ask one question? I am actually
Yes,
responding
sure,
from
ma'am.
Ufsc College.
Yes, ma'am. Over to you, sir.
Sir, there are two questions. First question is that how the pasture management is done by local people, because there will be shepherds and all, how they must be doing some pasture management for their cattle and all. So how that is being practiced in New Zealand?
Are you talking about managing
Pasture.
pastures
Yeah, sir. Yeah.
from what?
Sir, means surrounding to forest, there must be some native people who must be doing this grazing activity. So for
Yep.
them, are there any awareness programs that they should do the pasture management like that? The efforts are done there.
Are you talking about pests in pastures
No,
or?
pasture. Yeah, pasture management. The cattle they are browsing and then like that.
Yeah, the pastures
Yeah,
management of pastures is
yeah,
not
yeah.
much of a problem because even if it's adjacent to a forest,
Okay.
you do not. The only thing that they need to worry about is invasive plant species, right? So weed species.
Okay.
If I get your question right,
Fine. Sir,
right?
one more question is that phenology also with alphabetic ecology, floral calendar,
Sorry, you're breaking up a little
floral
bit.
calendar, floral calendar, phenology.
Yes.
Therein we are going to see the different stages of plant life and we constantly monitor it. So in that case, there will be a large impact if suppose the grazing activities, browsing activities are going on. So in that case, how that is monitored?
Can you? You're breaking up. The line is not clear. Sorry. Can you email me your question?
Yeah, yeah, fine, fine, sir. May I
Okay.
get your mail ID, sir?
Yeah, it should be there in the brochure
Okay, fine,
or on the
fine, sir.
website. Okay, it should be over there.
Otherwise, I will
Just
take
email it to
from,
me.
yeah, I will take
Yeah,
from
yeah, yeah. Just send it to me. Yeah,
organizers. Yeah, yeah,
yeah.
fine.
It's breaking
Okay,
up a
sir.
little bit. Yep.
Thank you so much, sir.
Any other questions or comments?
I have one.
Dr.
Hello.
Joshi. Yeah.
Hi, good afternoon, sir. Good afternoon. It's really a pleasure to listen to your beautiful work. I just wanted to comment. It's so really nice to get back and reminds me of the days when we were doing research together. And it's just wonderful and beautiful piece of work that you, dwelt on. And I was just wondering if, if you look at forest from that perspective, there could be so much of work anybody could do, especially in this part of the world. I have not read any of the papers on the line. My many congratulations to you, sir. It's just my comment. Thank you.
Yeah, I think. Yeah, thanks, Santaram. I think the issue is because it's quite a unique problem faced by island ecosystems like New Zealand. Because you wouldn't have this kind of an issue on a mainland ecosystem, right? When you think of deer, I remember when the first time, in New Zealand 20 years back, and I encountered deer in the forest and was told that deer is a pest. It was obviously something which is really strange because, say, back in India, deer is a protected species. And here you've got 7 species of deer and they're all running rampant around the forest and their populations are increasing all the time and need to be culled just to try and keep their numbers down and protect the forest ecosystem, which is very unique. But yeah, it's interesting that you mentioned because Hawaii is having the same problem because Hawaiian Islands are, the same issues. So they didn't have native mammals over there, and deer and other species have been introduced. So a lot of these studies, you find the authors or common authors or groups from Hawaii and New Zealand working together to try and find, better ways to try and manage these, I guess.
Yeah, that's the other thing I believe could be, our country has the problem of population, and there are a lot of meat eaters, and deer hunting is one major issue the forest department has to handle. I think New Zealand also got to do with the population density, and I don't know what is the extent and the percentage of meat eaters. If deer hunting, if Indians are available there, I believe we would have handled the entire population. I think the number and
Yes.
the meat eaters also would be.
Yeah. Interestingly, deer is also farmed here. So they also have a lot of farms, large farms where deer are farmed for the meat. And the venison, New Zealand venison is in fact quite in demand. So a lot of it is exported to Europe and the US, but also locally available here. But it's the wild populations that are really difficult to control just because of the lack of predators, natural predators in the ecosystem, I guess.
Thank you. Thank you, Devi. It's wonderful
Okay.
listening to you after many years. Hope to see you soon.
Same here. Thank you. Thanks, everyone.
Okay, I just want to have one query. So,
Thank you so much for this informative session, Dr. Gupta, sir. So as a saying by Pythagoras, that leave the roads and take the trails. But you bring
Shweta ma'am, Kusum madam.
Okay, so just want to say hi to Dr. I think you remember me. I was your junior in the lab.
Yes, I
So,
remember you very well.
Yeah,
Good to see
yes.
you all. Yep.
Yeah, good to
I
see
can
you
see a
too.
few
So,
other familiar names in the list. Yeah.
really impressed with the kind of work you're doing. So I just have a question, like as you say that in New Zealand, this deer is being farmed. So in India, we have a problem of this monkey. So what kind of suggestion you will have? Like this monkey menace is too much in Indian context, and people are trying to find out the solution for that also. But still, it is a bit. So do you have anything which can, from your experience of working there, that you could like to say that this problem can be, I mean, it can be addressed, this monkey menace here?
A good question. I don't know if I'm the right person to answer that because, like you, I'm a botanist. So obviously, I look more at the plants. For me, deer is, just a means to get to the plants. I know nothing about deer except that they're a problem and they're having an impact on a plant. So the only reason that I brought deer into the equation over here is because it's the plants that we are trying to protect and the forests that we are trying to protect from collapse. But I'm sure there'll be zoologists. I can see Anshuman over there in the panel. I think maybe Anshuman can.
I think in India there are many people who are working on primates. They are called primatologists, and I think they are thinking about these problems which are arising due to the global change and the climate change issues. So they must be they must be thinking to find out some ways to protect the environmental balance and to protect the animals and the species and the biodiversity.
Yeah.
And one more question. Like in Australia, we have seen that there have been forest fire into rise for the last few years. So what is the situation in New Zealand? Because that is also, I mean, closely connected nearby. So is it the same?
No, not really. I mean,
Parts or
here the ecosystems are very different. So Australia is obviously, very dry. Well, most of it is very dry. Whereas in New Zealand, it's pretty much like the northeast of India. So you've got relatively high rainfall, you have cooler climates, and so the soils and the forests obviously retain a lot more moisture. So you don't have forest fires over here. It's extremely rare, and even if you do have an instance of forest fires, then the forest fires usually don't spread very fast or don't spread far because there's a lot of moisture content in the soil and the forest itself. So no, it's not like Australia. It's in fact quite diametrically opposite.
Thank you. Thank
Okay,
you, Dr.
is there any query for the person? Okay, thank you so much, sir. I was saying that there is a saying by Pythagoras that leave the roads and take the trails. But you bring us to the notice that there is a need of assessment to control the measure to enable plant regeneration. You told us about the New Zealand condition. So thank you so much, sir, for this informative session. And now I am handing over for further proceeding to this session to Babita ma'am. Over to you, ma'am.
Thank you, Shweta. So moving ahead with the next part of the session, paper presentation in biodiversity and environmental conservation, environmental sciences. I invite Honorable Chairperson Dr. S.R. Joshi and Dr. Kusam Arunachalam to join the session. Before we begin with the paper presentation, Dr. Joshi and Dr. Kusam, with your kind permission, I'd like to brief the paper presentation session in biodiversity and environmental botany.
Yes,
Can
you
I?
can. Yeah, no problem.
Yes, ma'am. Yes, ma'am. We have received 28 pre-recorded paper presentation videos from the presenter. Our technical team, Ms. Kaveri, Ms. Rutuja, Ms. Rinkal, and Ms. Pratibha, our PG students, they have compiled all the videos in 2 files with 14 videos in each. All the videos will be played in the sequence shared with the participants. The participants are requested to be there throughout the session. The score sheet to evaluate the presentation is shared with both the chairperson. Now I request Dr. S.R. Joshi and Dr. Kusam Arunachalam to kindly share their valuable inputs with our participants after the paper presentation session. I request our technical team to start with the paper presentation. Rutuja, Kaveri.
There's just one little query because the paper Professor Kusum is around, I believe. Before we begin, can the technical team hold on for a while? Yeah. How do we go about if there are certain queries that are going to be made on the presentation? How do we go about handling those? Am I audible? Am I audible to the rest?
Yeah, Joshiji, are you talking of the
Sir, you are
scoring?
not clearly audible.
Joshiji,
No,
are you
what I'm
talking
talking about is I'm talking about you have presentations which are video recorded, so they are going to be presented in series, right? If these presentations have certain queries, maybe by the participant or anybody else, how do we handle those because they are recorded now?
At the end of the session, if you have any queries, you can ask it.
So participants
At the end of the session,
At
the end of the session, all the participants will be there, I think. So if there is any query, they could be addressed to them.
They are yes.
That is
Yes,
fine.
ma'am. Yes, all the participants are there throughout the session, and after the video play will be over, then participants and our chairperson both they can interact.
Okay, no problem. That's fine.
So we are expecting this session to be almost 3 hours, 5 minutes for.
Yes, sir. Yes, sir. Almost 3 hours. Maybe more than 3 hours. I think 3 hours 10 minutes. What our team has calculated, it's going to be 3 hours 10 minutes.
A very good day to all. Myself, Abhijit Simon.
I'm going to present a research paper on study of physicochemical parameters before and after sewage treatment by using novel method from an urban town. Let me introduce you. Sewage is 99% water carrying domestic waste originating in kitchen, bathroom, laundry, urine and night soil. Discharge of the domestic sewage as such in the form of untreated, treated or partially treated is left into nearby water bodies is a common practice in many countries, including in India, which is a major point source of water pollution. There is a large gap between generation and treatment of sewage in India. Current and future freshwater demand could be met by enhancing water use efficiency through treating the sewage. The various conventional methods for wastewater treatment are present since ancient times, but they are very costly and not economical. The advanced new technical methods need to be introduced to overcome the conventional method of wastewater treatment. Objective of the present study, an attempt has been made to develop an cost-effective novel sewage treatment method and evaluate the efficiency of the developed novel method by analyzing the different physicochemical properties of untreated and treated sewage water at Bhiwandi district, Thane, Maharashtra, which is a textile hub. Materials and method. Study area description. The study was conducted in Bhiwandi city in the Thane district of Maharashtra state, in Konkan division, located 20 kilometers to the northeast of Mumbai and 15 kilometers to the northeast of Thane city. Sample collection. The raw and treated sewage samples were collected in 1 liter plastic bottles, which were carefully rinsed with the respective waste water samples before collection and labeled properly. On the same day of collecting, samples were transported in ice chest in the laboratory. Physicochemical analysis. The physicochemical parameters such as water color, temperature, pH, TDS, transparency, total hardness, alkalinity, chlorides, calcium, silica, iron, nitrate, phosphates, ammonia, carbonates, COD and BOD of the untreated sewage and treated effluent were measured in this study. Results. All parameters in the treated effluent were within the permissible limits. The result indicates that the treated effluent is non-polluted and can be used for different purposes. The developed novel sewage treatment technique. In this, you can see that the sewage is entering into the first tank in which a sieve is stepped. This sieve helps the solid particle and the plastic carry bags, vegetables, all the big particles to be separated from the sewage. The B diagram is first sedimentation tank in which the sewage is made to flow through the tank and it is made to obstruct in different levels by walls, and because of which the sludge settles at the bottom, and at the bottom of the tank at different wall level there are pipes which are kept. Through these pipes the sludge is removed out. These pipes are a result of different trials and error during the course of this research. While opening this pipe, a vacuum is created, and because of that vacuum high pressure above the pipe, only the sludge is sucked out while opening the cork or wall of the pipe. C diagram is the second sedimentation tank, which is similar to the first sedimentation tank. The difference is only that it is having a deeper space by which what happens, even the small suspended part is made suspend at the bottom. The last diagram is D diagram, which is the final product water tank, in which the water is made to fall from a great height, because of which the oxygen depleted water takes the oxygen which is present in the atmosphere and gives away the foul smell and the carbon dioxide and other gases which is present in excess in the water to the atmosphere, thereby enhancing the quality of the water. These photos are of the actual site of the sewage treatment plant, in which daily around 15 lakhs liters of water is treated and supplied to the nearby textile industry. The important thing to note in this physicochemical result is that the color of the water, which is blackish, and now after treatment it is colorless. That is a very important thing to be noted. The important aspect is that you have to note that the foul smell has been removed and there is no smell now in this treated water, and the water is clear. Neither saying colorless, it is clear than clear blackish water. Results of this study show that all parameters were within the acceptable limits, indicating that the quality of water had been improved. So the novel technique of sewage treatment is efficient, and the treated water can be used for secondary purposes such as floor washing, irrigation, fish farming, textile processing and gardening instead of fresh groundwater and surface water. This treated sewage can also replace the groundwater used in the textile industry of Bhiwandi, which is popularly known as Manchester of India. Thank you.
one and all. I am Dr. Ufsc. My topic is differential response of growth level for oxidation to two strains of Azolla, that is Azolla microphylla and Azolla peculatus. Because of anthropogenic or man-made activity, there is a gradual decrease in ozone layer. And because of the decrease of ozone layer, gradual decrease of ozone layer, UV radiation is directly hitting the earth. There are three types of UV radiation. UV radiation that is not very harmful, UVC radiation that is most harmful, and UVB radiation. UVC radiation is not hitting the earth because it is being absorbed by ozone layer. UVB, that is very harmful, it is reaching the earth. And it is a kind of abiotic stress, and because of this stress, that is a lower wavelength radiation, so its energy is very high. So directly it damages the proteins, nucleic acids, and biomolecules. And they produce reactive oxygen species like superoxide radical, peroxide radical, hydroxyl radical, etc., etc., etc. I have chosen Azolla, two strains of Azolla, that is Azolla microphylla and Azolla peculatus. Azolla is used as biofertilizer, and not only biofertilizer, it is also used as animal food, human food, medicine, hydrogen fuel, production of biogas, water purifier, weed control, etc., etc. Basically, in tropical and subtropical countries, it is used as biofertilizer in rice paddy fields. To compare the UVB, the impact of UVB strains of Azolla, that is Azolla microphylla and Azolla peculatus, with the following objectives: to evaluate the impact of UVB on growth of Azolla plants and to illustrate the antioxidant defense mechanism of Azolla microphylla and Azolla peculatus exposed to UVB. Third sign adopted during the research work. Now, all I did growth, and I did growth in the form of dry wave. So I gave 0 minutes with 0 minute UVB to 60 minutes 1 hour UVB, like 15 minutes UVB, 30 minutes UVB, 45 minutes UVB, and 60 minutes UVB. And after 4 days, 72 hours, after 3 days, I took the reading. And I observed at 15 minutes of UVB radiation in Azolla microphylla, the growth was decreased by around 6%. And Azolla peculatus, that was 15%. And gradually it is decreasing, and around at 60 unit of UVB exposure, Azolla microphylla was reduced. The rate was reduced by 30%, and in case of Azolla peculatus, that is 52%. This is because of the low synthetic activity. Directly the UVB effects on protein synthesis. It's the DNA and RNA also. So because of all these things, there is a decrease in the dry weight of these. And there is a significant increase in the MDA and the malate dialdehyde. So when I did malate dialdehyde for oxidation, so I observed that there is a gradual increase in malate dialdehyde. So at 15 minutes of UV, that is around, it is increased by 6%, and in case of Azolla peculatus, that was 11%, increased by 11%. And at 60 minutes of UVB, this was increased, MD was increased by 25%, and in case of Azolla peculatus, this was increased by 36%. There's a defense mechanism also. Every action has equal and opposite reaction. So there is a reaction, or there is a defense mechanism from the plant cells also. So antioxidant metabolites and antioxidant enzymes. They scavenge such type of free radicals like reactive oxygen species like superoxide radical, peroxide radical, etc., etc. So I try to observe the impact of UVB and the response of the plants to strains of Azolla against this. So first of all, I did proline, and after giving 15 minutes of exposure, proline was increased. This mechanism. Next, I did two antioxidant enzymes such as superoxide disc and ascorbate peroxidase. So these two enzymes are they act as defense mechanism. Oxide radical. So it scavenges, but it converts the superoxide radical into H2O2 reactive oxygen species, and it is equally harmful. So for that, there is another defense mechanism, another enzyme on H2O2, that is ascorbate peroxidase or peroxidase enzyme. So I do peroxidase enzyme. Peroxidase enzyme converts H2O2 into H2O and O2. So first I observed superoxide dismutase enzyme, and I observed after giving 15 minutes of UVB, this was increased by 1% in case of microphylla, and this was increased by 7% only in case of folliculitis, and in case of 60-minute 1 hour exposure of UVB, it was increased by 40% in case of microphylla, and this was increased by 19% only in case of Azolla folliculitis. Similar, I got in the case. So at 60 minutes of UVB exposure, in case of microphylla, this was increased by 25%, and this was increased by 16% only in case of Azolla folliculitis. So after completing this, I got the Azolla microphylla strain is better tolerant to the strain. It is tolerant to UVB better than Azolla folliculitis when we compare it. And I concluded my result in these sentences. I'll read it out for you. The result shows the differential response of two strains, Azolla microphylla and Azolla folliculitis, in response to UVB. The observed varietal diversity in response of UVB is probably because of Azolla microphylla appears to be better tolerant to efficient scavenging of radicals by increasing antioxidant activity. In contrast, Azolla folliculitis showed poor antioxidant activity and detoxification of free radicals, making them sensitive to UVB. These observations serve as a basic idea for analyzing Azolla genotypes towards increasing the Anamina association, as it is used as biofertilizer for rice crop, despite the threat of increasing levels of UVB. These are the references I mentioned here, and thank you so much.
Hello and warm greetings to one and all present there. I am Dr. Ajita Kumar, Assistant Professor in the Department of Botany, Wilson College, Mumbai. I am presenting my paper titled Improvement of Physiochemical Characteristics of Soil Using Biodegradable Kitchen Waste, Trigonella Fena, under the category Biodiversity and Environmental Science. The physiochemical properties of the soil affect plant growth and soil management. The nature of soil with respect to quality and quantity of its content defines the future of the produce. It is well known that soil quality helps in retaining water and supporting agriculture. Also, biowaste might help in reforming the soil structural makeover effectively and retain the nutritional aspects. The soil carbon and nutrient availability play a crucial role in ecosystem sustainability, and they are controlled by the interaction of climatic, biotic, and social physiochemical variables. Various parameters to study the physiochemical nature of soil are the soil texture, structure, color, depth, bulk density, porosity, the mineral content, organic matter content of the soil, moisture, carbon to carbon ratio, carbon to nitrogen ratio, cation exchange capacity, fertility, soil pH, etc. The interference of biotic components lead to variations in the soil composition. Hence, soil physiochemical properties have been recognized as variables for predicting soil organic carbon and the nutrients. As we know, the present status in agriculture sector has created a problematic situation due to the improper management and chemical fertilizers in soil. There's a wide range of research focusing on improving soil quality characteristics through effective utilization of naturally available resources becomes the priority. This piece of work was undertaken to study the effectiveness of biodegradable waste, Trigonella phenum on water holding capacity and organic content of the soil. The experimental work consists of treating 3 different soil samples with the kitchen waste Trigonella phenum and study the comparative account against the control soil samples. Fresh bio-waste of Trigonella phenum was sun-dried for 48 hours. And after that, the semi-dried waste was chopped finely and used for the experiment. The soil types selected for the studies were garden soil, sandy soil, and clay soil. All the soil samples were oven-dried. The untreated soil samples were the control, while the treated soil samples were prepared by mixing 100 gram of bio-waste in 100 gram each of the soil type. The proportion of 1:1 was maintained for all the treated samples. And accordingly, post-treatment, the soil samples were recognized as untreated and treated soil sample types. For estimating the water holding capacity, 100 gram each from the untreated soil samples and treated soil samples were taken in separate beakers, to which 100 ml of water was added and stirred vigorously. The soil solution was allowed to stand still for about 1 hour, and after 1 hour, the soil solution was filtered through Whatman filter paper number 41, and the observations were recorded. Now, for calculating the organic content of untreated soil samples and the treated soil samples, Walkley and Black's method was used. This method is widely used experimental methodology for testing of organic content in the form of percentage of carbon and organic matter present in the soil. This method is also known as wet test, in which quantitative organic carbon is determined in terms of percentage. Here, the potassium dichromate used acts as an oxidizing agent in presence of concentrated sulfuric acid, and the volumetric titration is carried out against 1 normal ferrous ammonium sulfate. Diphenylamine was used as an indicator, and orthophosphoric acid helps in preventing the interference of oxides of nitrogen and other minerals. The percentage of carbon was calculated using the formula given, and the organic matter of the samples, the treated and the untreated, was calculated by multiplying the percent of organic carbon with the Allison factor, which is 1.72. The results were tabulated, and it was observed that the water holding capacity of untreated garden soil, sandy soil, and clay soil samples was found to be 35%, 20% and 60% respectively, while that of the treated garden soil, sandy soil, and clay soil samples were observed to be 95%, 57%, and 94% respectively, as expressed here graphically. The water holding capacity of the treated garden soil and sandy soil samples was found to be twice the amount of their untreated counterparts. On the other hand, the water holding capacity of clay soil samples were found to be increased by 32%. The organic content, carbon content in untreated garden soil, sandy soil, and clay soil was observed to be 1.85, 1.2, 2.1 respectively, while in the treated soil samples of the three types was found to be 8.3, 3.3, and 6.3% respectively. So there was a clear increase in the percent organic carbon in the treated soil samples of garden soil, sandy soil, and clay soil. The amount of organic matter in the three soil samples when untreated was found to be 3.18, 2.07, and 3.61 respectively, whereas the treated garden soil, sandy soil, and clay soil showed 14.3, 5.67, and 10.87 grams of organic content. These results proved to be really encouraging and hence confirming that the kitchen bio-waste of Trigonella phenum is an effective source in the field of agriculture for improving and organic matter necessary for a better produce. With that, I conclude my work and presentation. These are the references used for the same. Thank you.
I'm going to present my research work entitled Chronic Exposure to Environmental Relevant Concentration of Fluoride causes fluorosis and alters DNA reproduce expression by epigenetic modification in mice. Fluoride is an essential trace element. It can be present in both surface and groundwater. Anthropogenic activities can also increase fluoride level to some extent. As per the guideline of WHO, permissible limit of fluoride in drinking water is 1.5 ppm. When the concentration is below the permissible limit, it prevents dental caries maintenance and helps in growth of skeleton. But chronic exposure at higher level causes severe form of dental and skeletal fluorosis. And recently, it has been found that low IQ level of children in fluoride endemic region. This is the global scenario of endemic fluorosis. In India, 62 million individuals are suffering from fluoride toxicity. This is the state-wise fluoride level here you see. In almost all states of India, high level of fluoride has been reported. Although 200 million people all over the world are suffering, but there is effective treatment because the molecular mechanism is yet to discover. In this context, to discover the mechanism of fluoride action, we first made a survey in Rampurhat 1 block, Bihun district, West Bengal, and found that the villagers are still getting exposed to much higher concentration of fluoride. As permissible limit is 1.5 ppm, but we found that their drinking water contains highest 11 ppm of fluoride, and their urinary fluoride concentration was also found to be very high. And as a result of this, they are suffering from severe form of dental and skeletal fluorosis. These are the mild dental fluorosis, these are the moderate form of dental fluorosis, and these are the severe dental fluorosis. That teeth became deeply brown along with mottling or pitting appear. These are the victims of skeletal fluorosis, and they are unable to stretch or bend their hand and legs, and some of them got crippling deformities. From this survey, we selected 15 ppm sodium fluoride as an environmental relevant concentration because it readily dissolved in water and yields 7.5 ppm fluoride. After treatment period, we measured the deposition of fluoride content, which was found to be significantly higher in all fluoride treated group. This is the organosomatic index, which was found to be lower in treatment group. And biochemical analysis of stress parameters showed significant downregulation of glutathione content in liver and brain, whereas MDA, that is end product of lipid peroxidation, was found to be higher in liver, brain, and skin tissue. Catalase activity was also found to be higher in treated group. Histological studies show drastic structural alteration in fluoride treated group in case of liver, inner central vein, loss of its epithelial lining, integrity, hepatocellular hypertrophy, tubular sinusoidal dilation was noticed. In case of kidney, distorted Bowman scaffold, reactivation of glomerulus, tubular widened movement are noticed. In case of brain, loss of disintegration of meninges lining the cerebral cortex, pyknotic nuclei, and neuropil hyperpolarization are distinctly evident. In case of spleen, we did not find any significant changes, although in treated group, white bulb region was found to be diffused to some extent. The studies so far, along with my study, confirmed fluoride induces RS-mediated oxidative damage and causes chromosomal breaks, chromatic aberration. And among the bases, guanine is highly susceptible to RS-mediated oxidative damage, which transform it into 8-oxoguanine, a mutagenic byproduct. And one enzyme called oxogenic glycosylase 1 repairs this. And RAT52 is crucial because it helps in DNA double-strand break repair. Thus, our next objective was to find out the status of DNA repair gene due to fluoride treatment. After 8 months of fluoride treatment, we performed real-time PCR study which showed significant downregulation of ODG1 in liver and brain, whereas RAT51 was found to be downregulated only in brain. As gene, and we then also tried to find out the cause of gene downregulation. For that, we performed methylation-specific PCR study. MSP study showed significant promoter hypermethylation in RAT51 brain, but we did not find any association in ODG1 liver or brain. As DNA repair machinery was getting compromised, so there is a chance of triggering cell death or apoptosis. That's why we then performed apoptotic Western blot, and Western blot results showed significant upregulation of P53 bags, cytochrome C, APAF1, KIP9-3 protein in treatment group, whereas downregulation of BCL2 in fluoride treated group, which is anti-apoptotic protein. Thus, we say that we can conclude that fluoride induces RS-mediated oxidative damage, alters DNA repair gene, and causes promoter hypermethylation RAT51, and ultimately triggers cell death by apoptosis. We also tried to find out the status, find out the role of calcium and vitamin D supplementation, whether it has any role in alleviation of sclerosis. This is our experimental design. Here we divided the laboratory mice into 6 groups. Group 1, that is untreated control, received fresh drinking water. Group 2 received 15 ppm of sodium chloride for 4 months. Group 3 received sodium chloride for 8 months. Group 4 received sodium chloride for first 4 months. And next 4 months, their sodium chloride or chloride treated water was substituted with fresh drinking water. Group 5 received 15 ppm of sodium chloride for first 4 months, and after that, they received fresh drinking water along with calcium and vitamin D supplementation. And group 6 is co-exposed group, that is they received both fluoride and calcium vitamin D supplementation. After the end of the treatment period, we measured the deposition of fluoride content in femur bone, and it was found to be higher in fluoride treated group, that is group 2 and group 3. But in co-exposed group, the fluoride level was found to be much lower than the fluoride treated group. And in case of calcium vitamin D supplemented group and group 4, it was like near normal. And we also monitored or measured, the status of dental fluorosis. This is the incisor of control mice. It is noteworthy mentioning that unlike our teeth, yellow coloration of control mice teeth doesn't indicate a bad oral health. Rather, yellow coloration is due to the deposition of iron containing fluid. And after onset of fluoride treatment, we found teeth gradually turned into whitish, and at the end of the treatment period, they became completely deep white. Although calcium and vitamin D supplemented group, severity of dental fluorosis was alleviated. We also performed X-ray imaging study for monitoring dental fluorosis, and we found that in group 2 and group 3, that is fluoride treated group, a distinct sign of osteosclerosis or abnormal hardening of bone due to fluoride deposition. But in case of group 4, that is first 4 month fluoride water, next 4 month fresh water, the sign of osteosclerosis was found to be abolished. But in case of calcium and vitamin D supplemented group, we didn't find any significant sign of osteosclerosis. We also performed metal profiling by EDXRF study and found that in fluoride treated group, the content of calcium, manganese, potassium, sulfur, nickel was found to be much lower. But in case of calcium vitamin D supplemented group, it was found to be like near normal and retained elemental homeostasis. Thus, in a nutshell, we can conclude that chronic exposure to fluoride accumulates in teeth and bone, causes dental and skeletal fluorosis, alters metal profile. But supplementation of calcium and vitamin D effectively alleviates fluorosis and retained elemental homeostasis. Thus, we can, from our study, we can conclude that calcium and vitamin D acts as a redeemer of fluorosis. Thank you.
Hello everyone. The topic that I'm going to present is on seasonal variation of soil fungi in tea garden, Mugokchung district, Nagaland. Tea plant is one of the major cash crop in northeast India, and tea leaves is cherished as a popular beverage in a lot of healthy human diet. In the state of Nagaland, Naka people are giving due importance to tea cultivation in areas of some districts, including the district of Mugokchung. Despite on a small scale, the state has started tea leaves export, and tea factories are also being set up in many places. The distribution and occurrence of fungal species is found to be influenced by seasonal fluctuations, surface vegetation, and endogonistic activity of the fungal species. For many years, decultivated soil has been a hot topic for researchers, as soil fungi are key drivers of plant productivity, and understanding the fungal diversity is a necessity for the growth and yield of an economic important crop like tea. Therefore, the present study was undertaken as an attempt with the objective to record a data on native soil fungi occurring in different seasons in tea garden of Mukokchung district, Nagaland. The present study was undertaken with these two objectives. Methodology. Seasonal collection of soil sample was done from the tea garden during January 2017 to December 2017. Isolation of mixed fungal growth was done on raw spinkel agar media from which colony forming units were observed. Pure fungal isolates were maintained in 4 different culture media from which each fungal isolates were identified by studying their morphological and microscopical characteristics along with the help of available literature. Important value index was calculated to find out the most dominant fungal species in the present study. Figure 1 shows the study map. Figure 2 shows the study site, tea garden. Results. Figure 3 shows the mixed fungal growth isolated on raw spinkel agar. Figure 4 and Figure 5 shows the pure fungal isolates with the culture plate and microscopic images. Table 1 shows the total fungal population occurring during winter, spring, summer, and autumn season. From the table, it is observed that the highest colony forming unit was recorded in spring season, whereas lowest colony forming unit was recorded in winter season. A total of 60 fungal species and 27 genera was isolated during the present study. Table 2 shows the variation in fungal species during different seasons. The highest fungal species was recorded during spring season with 52 species, whereas the lowest was recorded in winter season with 25 species. Figure 7 shows the seasonal wise distribution of 27 soil fungal genus. This figure shows us which fungal genus had the highest species and in which season. The 27 fungal genus are represented by different colors and is shown on the top of the figure. So the figure shows that the highest fungal species was recorded during spring season in genus Aspergillus with 8 species, followed by genus Penicillium and Trichotoma with 7 and 6 species, whereas the winter season recorded the lowest fungal species in genus like Epigocum, Acremonium, Cleoclatium, and Pasteludiopsis. Seasonal variation in fungal distribution in tea soil were observed during the present study, which may be due to the fluctuations in environmental factors during seasonal changes, which has also been reported by other workers. Genera of Aspergillus, Penicillium, and Trichotoma were found to occur in maximum in all the seasons, and the highest species was recorded under this genus during spring season. The reason for this may be due to the better spore-forming structures of this fungal genera, as fungi with higher spore-relating features are easier to discover, and increased moisture and rainfall during spring season might have favored the abundance of the fungal spores. Highest IVI was recorded in Aspergillus niger, which may be due to the antagonistic behavior, as Aspergillus species are reported to show strong antagonistic activity towards other fungal genera. Therefore, dominancy of some fungal species during the present study period can be attributed to the antagonistic nature, seasonal fluctuations, and environmental conditions of the present study site. In conclusion, the present investigation provides information about the seasonal fungal variation in tea soil of Mukokchung district, Nagaland. For a sustainable utilization, the local stakeholders should be made aware of the potential and rich biodiversity in the region. Inventory of bioresources and its sustainable utilization is recommended. Modern technology for obtaining a detailed overview of microbial diversity can be intensified in extension of this investigation. References. Thank you.
Hello everyone, myself Ashwini Deshpande from SIS College, Mumbai, wish to thank the organizers for giving me an opportunity to present my research work titled Diversity of AMP Spores from Industrial Beds of MIDC Dombivli for Winter Season. I always say that we have to think globally but act locally, and that is why I have chosen the local area of MIDC to study ant fungi. Generally, fungi, they play a villainous role in the plant kingdom because they are known for their pathogenicity. But arbuscular mycorrhiza, they show symbiotic associationship with the higher plants. The cortical cells are invaded by mycorrhiza. In turn, mycorrhiza is getting the food from the plant, and the mycorrhiza is taking the uptake of phosphorus for the plants. So this is a symbiotic beneficial relationship which they exhibit, and that's why they play a heroic role in the ecosystem. So let us see that what is my study area. I have chosen MIDC area, which is a GPS map of this area under study. Here you can see that industries located in this area are chosen for survey of ant fungi diversity. We can see the actual map of industries, how they are located. The weeds were collected, which are growing around this area, and the diversity of ant spores was checked by using weight seeding method, which was described by Gardaman and Nicholson. You can see the steps involved in the weight seeding. Here the soil is collected from the rhizosphere of the weeds, and later on it is seeded. All the spores which are being accumulated on the seed, those are collected, they are washed, they are eluted, collected, they are filtered, and then analyzed for their presence of ant spores and morphology. They are authenticated by Dr. V. Mohan, who is a scientist in IFGTV. We have seen the observation that there are three basic genera of ant spores which are dominant. Out of that, Glomus was the most dominant genera. This table shows that we can see Glomus, Acaulospora, and Gigaspora. These are the three genera which were giving the highest percentages. Out of that, Glomus, with its many species, was the most dominant genera in the study area. We can see this diversity in the form of images of spores which I have collected. So we can see that Glomus is the dominant genera with its species Glomus occultum, Glomus geosporum, Glomus fulvus, Glomus interradicis, Glomus fasciculatum, Glomus deserticola, Glomus multiculae, Glomus australe, Glomus alvidium. Along with Glomus, we have also found the three species of Acaulospora, which was Acaulospora scrobulata, Acaulospora melia, and Acaulospora foveata. Along with that, Gigaspora margarita also was found. These are the pictures of root colonization. For this, we have used two stains. First is trypan blue stain, and the another one is zinc in a dark stain. So these were described by scientists Philipson and Berling et al. So here we can see the images wherein the root cells or the cortical cells of the roots are invaded by fungi. You can see here the globular structures which are called as vesicles. These are sent inside, and then the fungi will ramify inside. The second image shows the dissected hyphal structures called as arbuscles, which are also exhibited as a form of mycorrhiza. In this image, we can see that how the vesicles are sent inside and they are growing inside as a part of root cortex. In the another image, you can see the groups of vesicles which are growing in the bunches, and this is how the mycorrhiza will spread throughout the root. In yet another picture, we can see that vesicles are fully established, and you also can see over here the hyphae are ramifying, ectomycorrhizal also and endomycorrhizal also. In another yet picture, we can see that the vesicles are established themselves, and that's why they are staining very dark in trypan blue. So from this, we can conclude that ant fungi, they have a capacity to grow in the challenging ecosystems such as polluted ecosystems. They can uptake the metals, heavy metals which are being there as a pollutants. So this is a perfect solution for the problem of pollution. We also can see that the dominant genera like Acaulospora, Gigaspora, Glomus can be planted over there. They can be trap cultured. They can be planted along with the weeds going over there so that they can take up the heavy metal and do the micro-remediation. So sandbinder species such as Paspalum and Cynodon can be used as a weeds which will grow very much enormously over there in that area, and they can take up the heavy metal. So this way, this model can be implemented for the entire world, and of course the metropolis can be have an eco-friendly cleanup strategy with the help of this diversity of ant spores. Thank you so much.
Greetings to one and all. Myself, Mrs. Dipti Deshpande, Assistant Professor in Zoology from GN Khalsa College, Matunga, Mumbai 19. I'm here to present a PPT on our paper in the category Biodiversity. The title of our paper is Effect of Physiochemical Parameters of Water on marine microalgal biodiversity of Bhuigaon Beach, Palghar district, Maharashtra. India is a tropical country having about 8,000 kilometer coastline spanning across 9 coastal states, 2 union territories and 2 island territories. Maharashtra is one of the states contributing to about 720 kilometers of coastline, and due to this huge coastline, there are a lot of algal species that have been reported. Maharashtra alone records for about 159 species from 79 genera. These algal species or seaweeds, these are grouped into 3 categories or called as chlorophyta, that is green algae, phaeophyta, that is brown algae, and rhodophyta, which is red algae. Algal biodiversity is found to be varying from region to region, coast to coast, and from time to time. Entire west coast of Maharashtra is shown 73 algal species. Only the coastline of Rajgarh, Konkan has shown 44 algal species. In case of other states like Karnataka and Orissa, 78 and 8 species have been recorded respectively. The site that we have selected is Bhuigaon Beach in Palghar district of Maharashtra. This is one of the virgin beaches, untouched beaches, and that is why collecting algal samples from these beaches is pretty easy. There have been 23 species of microalgae reported from freshwater bodies in this region. So our aim was to study macroalgae from this beach, and the time duration that we selected was 2016 to 2019. The time period that we collected the algal species was in the late winters, so beginning of summers, that is February and March. This we did for 4 years, and at the time we selected the algal samples, we've also collected the water samples to check the physiochemical parameters. And we've tried to establish any kind of correlation if they have. So at the time of sample collection, before we went for the sample collection, we've checked for the low tide timings. We've collected the algal samples. We washed them and transferred them to Ziploc bags, got them to the lab, removed any kind of epiphytes or any other unwanted material which was entangled in these samples, preserved them in 4% formalin, and then prepared herbarium. And later, these algal species were identified by making use of common seaweeds and seagrasses of India, volume 1 and 2, published by CMFRI. As I said, we also collected the water samples at the same time. So following physiochemical properties of these water were checked. There is pH, dissolved oxygen, free CO2, alkalinity, salinity, phosphates, nitrates. These are the results that we obtained. So there were about 8 species that were recorded from 2016 to 2019. Under chlorophyce, there were 2 species, Enteromorpha and Alva. Under phaeophyce, we had Sargassum and Padina. And rhodophyce, we had 4 species, Gracilaria, Halimania, Hypnea, and Gelidium species. Physiochemical parameters of seawater, these were the results that we obtained. pH, dissolved oxygen, free CO2, and alkalinity didn't show much of a change. It remained more or less constant in a particular range, but some changes were seen in case of salinity, phosphates, as well as nitrates. So coming to the correlation between these parameters and the algal species, Halimania species, these are found to be affected by some important physical and chemical properties of water like pH, salinity, dissolved oxygen, nutrients, and turbidity. This has already been shown in 2018. And in our results, we found Halimania to be present only in 2016. In the remaining 3 years, this was completely absent. So probably this could be one of the indications that the water quality is deteriorating, because of which this particular species vanished. Hypnea, on the other hand, its growth is affected by salinity and temperature. Its growth declines with increase in salinity. It grows very well at optimum salinity of 25 milligram per liter and temperature of 25 degrees, which is again shown in 2013. But in case of 2016, when the salinity was found to be high, Hypnea species was absent. So high salinity could be one of the reasons why Hypnea species was not found in 2016. On the other hand, the third species which we found showed some change in its appearance was Sargassum. Biomass of sargassum is found to be influenced by changes in phosphates and nitrate concentration in water. In 2019, sargassum species was not found by us. So the reason for this would be decrease in the phosphates and nitrates in the water sample. I would also like to add here, although this is not included by us in the papers, but after a gap of 2 years, right now in 2022, when we went and we checked for presence of valuable species again, so sargassum has been found again in lots of amounts. So probably nitrates and phosphates have come back to the required concentrations. So in the preliminary checklist of marine microalgae of Buigao Beach, a maximum species of Rhodophyceae were found. Our study also has shown a maximum of Rhodophyceae species, and Rhodophyceae species are seen to be affected more, followed by Phyophyceae species with respect to changes in these physiochemical properties of water. These marine algae, they are important because they are a great source of compounds like carotenoids, dietary fibers, proteins, essential fatty acids, vitamins, minerals, and important source of medicines as well as fertilizers. Sargassum and alva is used as animal feed because it's rich in high protein and nutritional content. Gracilaria is used for making compounds like agar and alginates. Hypnea is used as food, raw material for paint as well as agar. And there are quite a lot of other economic importance as far as marine microalgae are concerned. So these marine algae can prove to be a good source of raw material for various industries. Plus, their presence or absence, according to change in the physiochemical properties of water, indicates that these can be also used as good bioindicators for water quality. These are our references.
Good morning, everyone. It's my pleasure to present the paper in international conference organized by Guru Nanak Khalsa College. I, Dr. A. Bremina, Associate Professor and HOD of Botany, Andhra Mena Sabha Arts and Science College for Women, Usman University campus, and my co-author M. Maitri, University College of Science, Usman University, Hyderabad. Today we are presenting a paper on the topic, Germination Study of the plants Trigonella Phenum Graecum and the Vigna radiata on contamination with heavy metals in the soil. These are the contents. The soil pollution is caused by many direct or indirect anthropogenic activities. With advancements in human livelihood, the damage to nature is also rising rapidly, soil pollution being among them. The soil pollution affects the food we eat, the water we drink, the air we breathe, and the health of our ecosystems. Heavy metal toxicity is one such consequence of the soil pollution that leads to bioaccumulation of heavy metals and is of concern in agricultural production. Heavy metals naturally occurring in various concentrations in the bedrock at different places. Due to these anthropogenic activities, they pave their way into the biosphere, causing many hazardous consequences of plant growth and development. The accumulation of heavy metals in the plant body results in the alteration of physiological and metabolic processes, thereby showing the adverse effects, like its metal, its abundance, probability of its occurrence, the pH of the soil, the transport proteins, the chelating ability of the metal in the form in which the metal is present in the soil, the biotic factors such as light, temperature, etc. The toxic effects of heavy metals in the plants vary from metal to metal and also from one plant to plant. Our present study is aimed to observe the consequences of heavy metal contaminations with lead, mercury, chromium, and cadmium on the germination of plants Trigonella Phenum Graecum and the Vigna radiata discerning from normal conditions. Now, my co-author M. Maitri will continue.
Trigonella Phenum Graecum and Vigna radiata are annual, branched, dicotyledonous plants belonging to the family Fabaceae with nutraceutical importance. Coming to the methods and materials of our study, the heavy metals chosen for our study are chromium, cadmium, lead, and mercury. These heavy metals were added in the form of their salts, which are potassium chromate, cadmium acetate, lead nitrate, and mercury chloride. 1 gram of each salt was taken in respective clean test tubes and dissolved by adding 10 ml of water. The respective heavy metal salt solution was mixed thoroughly in 4 respective pots and 1 pot was left for control. The seeds were grown in 5 pots and given adequate amount of water. Coming to the observations made in Trigonella Phenum Graecum, after day 2 there was no much difference, but after day 4 there was luxuriant growth in the control plant and no conspicuous observation in lead, cadmium, and chromium, and mercury showed no sprouting. After day 6, lead showed considerably good sprouting, whereas there was less sprouting in cadmium and chromium.
After day 8, control showed copious growth and many seeds could not viably germinate in the lead, whereas very few could reach sprouting in cadmium, and there was phase germination in chromium. After day 10, the control showed heavy plantlets and less biomass was observed in lead. Very few could reach plantlet stage in cadmium and negligible germination was observed in chromium. Coming to the observations in Vigna radiata, after day 2, there was normal germination and the seeds of cadmium and chromium turned darker. There was no much difference observed in lead and mercury. After day 4, there was no conspicuous observation observed in lead, cadmium and chromium, but there was no sprouting in mercury. After day 6, the control showed plentiful growth and good sprouting was seen in lead. Early stages of germination were marked in chromium and seeds turned more dark in cadmium. Initiation of a fungal growth was seen in mercury. After day 8, lead spotted fair growth and curled and wrinkled leaves were observed in chromium. There was negligible germination in cadmium and deformed seedlings with shrunken seed coat were observed in mercury. After day 10, the control showed healthy plantlets and there was reduction in plant length and leaf size in lead. Burnt spots and cotyledonary leaves, curled and deformed leaves were observed in chromium and meager viability was spotted in cadmium. Deformed cotyledonary leaves were seen in mercury. Coming to the results and discussion of our topic, the contamination of heavy metals in the soil and their accumulation in the soil influenced the normal plant growth in many ways. Most of the seeds could not germinate in the soils contaminated with heavy metals. The comparative germination and the order of sprouting in different soils is as follows. In Trigonella fernumbracum, control was more than lead, more than cadmium, more than chromium, more than mercury. And for Vigna radiata, it was control, more than lead, more than chromium, more than mercury, and the least was spotted in the cadmium. The burnt cotyledonary leaves imply that foliar uptake of chromium is comparatively more than other heavy metals. Mercury was potentially toxic for seed germination in Trigonella, and cadmium was potentially toxic for seed germination in Vigna radiata compared to other heavy metals because the rate of germination was drastically low. Coming to the overall effect shown by heavy metals, there was overall reduction in growth and development, there was reduction in biomass, burnt appearance, deformed leaves, seeds turned unviable, and there was rate of reduction in germination. Let us have an overview of our project in an animation video. These are our references, and thank you for giving us an opportunity to present our paper in an international conference. Thank you.
Good morning, everyone. I am highly honored and obliged to Dr. Govind Kumar Barupal and International Conference Organizing Committee for giving me an opportunity. I am extremely delighted to attend the conference. Myself, Anandra Singh from Phycology Lab, Botany Department, Government Dunga College, Bikaner, Rajasthan. My research title is the Datum Profile of Cordem Jhunjhunu, Rajasthan. Abstract. Phytoplankton and other algae play an important role in carbon sequencing by forming the largest producer of biotic system. With their varied and vast habit and habitats, they have sprawled every niche of the ecosystem with diverse modifications to adapt with their changing environmental conditions. These adaptations are necessitated for their perpetuations and survival in varied conditions. Basidiophyci are highly successful and distinctive group of algae and found in almost all freshwater habits. The cell wall of diatoms made up from silica and that refer as the frustules and organic material the coat, the valve and girdle. The festoons can vary greatly in shape, which can be symmetrical or asymmetrical. Diatoms are further divided into centrioles and basilarioles order based on their morphology. The present study, coat dam of Jhunjhunu district, was selected. The district Jhunjhunu is the part of Shekhawati region, which located in the northeast part of Rajasthan. A systematic study of diatom flora of coat dam has been undertaken for the first time samples from different types of habit of coat dam were collected for a period of 12 months from July to Simbella, Amphora, Gomphonema, Cyclotella, Vacilleria, Dietma, Didesmis, Gyrosma, Helmphora, Sinendra, Calonic, and Fragile. From the coat dam Jhunjhunu, the most specific genera is the Gomphonema, Gestia, and Navicola. Introduction. Biodiversity is the measure of the variety of organisms present in various ecosystems. The abundance and richness of biodiversity depend on the climatic conditions and area of the region. Algae and other phytoplanktons have an important position in aquatic life and serve as a direct source of food to the other aquatic plants and animals. Rajasthan, situated at the northwestern part of India, is the biggest state of India, which covers the 10.41% area of the country. Maximum part of Rajasthan is the arid and semi-arid. The climate of Rajasthan desert is characterized by the high solar radiations, seasonally and diurnally fluctuating temperature, and precious and erratic low annual rainfall that acutely varies from year to year. Fort Dam Jhunjhunu, also known as Sardusaga Dam, that's present across the Sakamri Hills. It lies in between 27 degrees 39 minutes north latitude and 75 degrees 25 minutes east longitude. The height of Fort Dam is approximately 25 feet and length and width of the dam is 260 feet and 75 feet respectively. Legal analysis, a very useful for the assessment of water quality in any type water body and also contribute to understanding of the basic nature and economy of aquatic system. Change in meteorological parameters such as temperature, wind, precipitation pattern, and nutrient cycle affect the diversity of algae. Algae is a non-vascular haloid cryptogams that show huge diversity with largest source of photosynthetic activity in our planet, but it is more ignored to in terms of
Connected.
diatoms are photosynthesizing unicellular algae that form the basic bulk of planktonic population in the fresh water, characterized by having a cell wall with silica. The cell wall consists of two walls that have or less flat surface held together by a girdle. Plates of coated at different angles or material and methods. Sample collection were made from the different sides of them by the help of phytoplankton nets and sample bottle from July 2019 to June 2020. For detailed study of diatom, wall of diatom were filled with concentrated hydrogen peroxide to eliminate organic matter and later with the HCl to dissolve the calcium carbonate. After washing with distilled water, sample was mounted on slides with naprox. Trailing thresholds were examined for their morphological investigations. Micrometric measurement. The help of state and ocular micrometer has been taken and chemical history diagrams were made for the morphotaxonomical identification with the help of standard monograph. Identification of diatom up to species level were made following the key characters suggested by Gonzales and Gandhi. Gandhi, Prescott, Thoreau, and Kamath, Jessica Theory, Brown, Taylor, and Karthik et al. Result and discussion. Total 51 diatom species identified first time from coat dam genome that belong from 14 different genera such as Navicula, Nigestia, Timbella, Amphora, Gomphonema, Cyclotella, Basilaria, Dietma, Didesmis, Gyrosigma, Alphora, Sinendra, Kelenia, and Fragillaria. The table shows different species of Amphora, Basilaria, Cyclotella, Timbella, Keloni, Didesmis, Dietma, Fragillaria, Gomphonema, Gyrosigma, Amphora, Navicula, Nigestia, and Sinendra. Out of these, most richest Gomphonema and Nigestia and Navicula. Thank you so much.
Power off.
Good evening. Today I am going to present on the topic orchid diversity in community managed subtropical forest in Khasi Hills of Meghalaya. Now the family Orchidaceae is the second largest family of flowering plants in the world and is represented by 800 genera and 26,000 species worldwide. The family accounts for up to 10% of all flowering plants in the world. Orchids are distributed all across the world except
Connected.
hot deserts and Antarctica. Now, greatest diversity of orchids occur in tropical and subtropical regions of the world. In India, the orchid diversity is represented by 1,378 species and under 186 genera. The northeastern states sustain highest number, about 876 species belonging to more than 150 genera. Meghalaya is being in a northeastern state is rich in orchid flora, having 363 species under 107 genera. Meghalaya is a hilly state showing wide variation in rainfall, temperature, altitude, and soil conditions. These climatic conditions and topographic conditions support different types of vegetation, namely tropical evergreen, tropical semi-evergreen, tropical moist and dry deciduous, subtropical broadleaf hill forest, subtropical pine forest, grassland, and temperate forests. Most of the forests in the area are conserved wild and managed by local communities due to socio-cultural and religious beliefs. These forests are divided into three categories: sacred groves, number 2, protected forests, and number 3, village forests. Maintenance of these community forests has been in practice since time immemorial and is viewed as a traditional and cultural identity of Khasi and Jantia tribes. Some of these community forests have been studied for the purpose of vascular plants diversity and for plant population regeneration of some rare species. But little information is available about the orchid diversity in these community managed forests. Unregulated collection due to their commercial importance and habitat destruction have led to the gradual decline of the orchid population in the wild. Due to overexploitation, many orchid species are listed in Appendix 2 of CITES and are protected by this international convention. Therefore, the current study was carried out to assess the orchid diversity in community managed forests of Khasi Hills, and to study, the study was aimed at providing baseline data for the species present in this forest and would be helpful to the planners and conservationists in future. Now, materials and methods. The study sites, as you know, it is Khasi Hills of Meghalaya, and we know the area of Meghalaya. Covers around 22,429 square kilometers and altitude ranges from 50 to 1990 meters above sea level. The climate of the area is monsoonal. The wet season is relatively warm and extends from May to October. The dry season is comparatively cold, extends from November to March. And the average rainfall is varies from region to region. Southeast, it is greater than 4,000 mm. East of Meghalaya, 2,698 mm. And in north, between 2,500 to 3,000 mm. The climate of the central and east Meghalaya, that is Khasi and Jaiti areas, is characterized by low temperatures. And in the Shillong Plateau, the average temperature is about 5.8 degrees Celsius during winter months. At the foothills in the north and east, the climate is warm and humid. The state is mountainous and primarily consists of Archean rocks containing different types of minerals like coal, limestone, uranium, etc. Now, data collection and analysis. Extensive field surveys were conducted in 87 community forests in Khasi Hills during 2016 to 2019 during different seasons. The study site included 3 types of community forests, namely selected, protected, and village forest. You can see in the diagram the state of Meghalaya, and the encircled part in the middle represents the study area. Now, identification of species was done using published literature, mainly taking help from Kotaki 1984, Kotaki 1986, and by comparing the archaeologists housed at the Botanical Survey of India, Eastern Region, Circle Shillong. The collection, tracing, and mounting of specimen was done according to standard procedure of Jain and Brown 1977. Species whose distribution is restricted to Meghalaya or to northeastern region, including Indo-Burma hotspot and Eastern Himalaya, were considered as endemic. Species that were considered as having low population in the state of Meghalaya by earlier authors like Jain and Rao, 1983, Nair and Sastry, 1990, were classified as rare. And threatened category includes those plants that are classified as per IUCN list 2015. Now, the result of the study. The present study showed that community forests are rich in orchid diversity. The study revealed the presence of 118 species belonging to 55 genera. The majority of the recorded species, about 83, were epiphytic and 35 were terrestrial. Of all the species studied, that is 118 species, 28 were recorded as endemic, representing 23.73%, and 12 were recorded as rare, representing 10.17%. The highest number of species, 66, were recorded in sacred forest, followed by reserve forest, 51 species, and village, 32 species. Dendrobium with 19 species was the dominant genera, followed by Sironi with 8 species, and 32 different genus was monospecific. Now, conclusion and conservation strategies proposed from the study. Now, higher species richness in sacred forest is due to lesser disturbance and higher level of protection as compared to other forest types. Because the sacred forest where no collection activities, no disturbances are allowed by the local bodies. That's why the species richness is high. It has been widely accepted that anthropogenic disturbances cause an immediate decline in species diversity, ultimately leading to disappearance and extinction of the local species and their subsequent replacement by immediate species. Various anthropogenic disturbances followed by habitat loss have subjected these species or this species to continuous threat. Hence, these species warrant immediate conservation actions. Several strategies could be adopted, like total ban on the export of wild collected plants, orchid species, protection of the habitat of these orchids growing. Okay. Then salvation of orchids from damaged and threatened habitats and their maintenance in rescue centers, mass propagation of threatened species and their reintroduction into well-protected habitats are needed to be undertaken. Consequently, mass propagation through conventional and tissue culture techniques should be carried out to save the natural dwindling orchid diversity of the state from commercial collection pressure. Now, here we have some picture of the orchids that have been studied, and some of these have been maintained in the main house of the university. And some of these orchids are highly valuable, like dendrobium, and pandan, etc. They have very high ornamental values, and they are extensively exploited for this for their flowers. Now, the authors would like to acknowledge the grant received from DBT and Government of India for the sanction. The authors are thankful to the traditional heads and the local people for allowing the team to work at the study sites and for helping navigating and recording information in the forest. The help received from Botanical Survey of India, Eastern Regional Circles, in species identification is also acknowledged. Thank you.
My name is Simku Yawar. I am a research scholar from the Department of Environmental Sciences, National University of Mumbai. The topic that I'll be presenting is on the preparation of ornithines using TPPP and the evaluation as potential anti-diabetic agents via a computational approach. My aims and objectives are environmentally benign synthetic routes for preparation of ornithine compounds, characterization of the synthesized compounds in silico evaluation of the ornithine compounds as potential agents for anti-diabetic activity. The quaternary organic phosphonium trichomides have developed as alternative chemical reagents that have the potential to perform multiple chemical transformations with proven superiority over many known reaction-specific reagents. Physicochemical properties inherent of these reagents such as phase transfer property, chemical stability, thermal stability, ease and maintenance of their stereochemistry, recyclability of the spent reagent, etc. make them reagents of choice for organic transformation under clean conditions. On the other hand, in one of our other works, we have accidentally found that trichomides can catalyze the formation of one-time compounds from coupling of bacteria, and therefore we want to further develop these findings into a proper methodology for synthesis of quantities. In this slide, I would like to explain how the quantities were prepared. Through a solution of phenyl isothiocyanate in ethyl acetate was heated clockwise, separating at room temperature. Formation of diarrhea was observed within 5 minutes as judged from DLC. Studies worsened aided TPPP pinch twice over a period of 10 minutes. The reaction was kept steering at room temperature in complete conclusion to quantitine compound was observed within 40 minutes as seen by TLC and from the decidurant of the only color of TPPP. This is the mechanism proposed by the conversion. The mechanism essentially involves the initial attack of the soft nucleophile sulfur on the bromide molecules released from the TPPP. Thereafter, formation of the disulfide intermediate, followed by the attack of one of the helium nitrogen onto the adjacent helium carbon with the concurrent expulsion of elemental sulfur to give the desired product. These are some of the representative examples of quantum compounds prepared by the present method. All prepared compounds are characterized by product NR, complex protein NR, FTIR, as well as analysis, mass spectroscopy, etc. And a list of such prepared compounds has been assessed for advanced properties. As mentioned, having successfully synthesized the quantitine compound, we went on to study their biological properties. The predicted AD and AT properties of the compound under study are shown here. The results are that all compounds under study were predicted to be low or non-reactant and non-mutagenic. The blood-brain barrier (BTB) and human intestinal permeability (HIA) predicted has shown good permeability and absorption for the entire range of compounds under study. The acute toxicity of the compound was reasonably safe as category 3 and were considered to be non-toxic. Thus, the results of the compounds were predicted to be promising for further optimization as lead candidates. A good oral viability will be one of the vital pharmacokinetic factors for the development of drugs candidates. The quantitine molecule under study was prioritized to be phytochemical parameters to assess their drug likeness and viability. The result predicted are shown in this slide. Molecules that did not violate the rules of Chem 3, 2, 6, 9, and 12 respectively, hence predicted to have good oral viability properties. Based on the result of the ATMT viability prediction, some compounds were selected to elucidate the interactions and the bonding between the ligand with the target protein or enzyme that may relay the anti-diabetic effects to molecular docking studies. Diabetes mellitus is a severe metabolic disease that causes high mortality rate. There are various complications such as cardiovascular disease, strokes, retinopathy, etc. result from hypoglycemia and hypoglycemia. The most common diabetes is type 2 diabetes mellitus, differentiated by insulin resistance. Therefore, the present study illustrates in silico evaluation using different enzymes and receptors such as glycogen, phosphorylase involved in insulin secretion, and glucose metabolism as potential docking targets. Glycogen phosphorylase plays a central role in glycogenolysis, regulating the hepatic glucose output. Therefore, in a given hypoglycemia, inhibiting GP, that is the glycogen phosphorylase, to reduce hepatic glucose for type to diabetes. Type therapy has been an important target. The molecular talking result indicates that compound strongly interacts with GP through different amino acid residue as like the talking scores of the compound is in the range 136.642 kilocalories per mole to minus 172.765 kilocalories per mole, with compound 9 acidity component with minus 172.76 kilocalories per mole as shown in the table. The mode of interaction of the lead compound and the active side of glycogen phosphorylase is shown in this slide. The active interaction results suggest that the compound may inhibit glycogen phosphorylase by preventing glycogen breakdown, facilitating decrease in blood glucose level. PPARY plays a major role in the metabolism of glucose and lipids. Activating PPARY influences free fatty acids, drugs thereby reducing insulin resistance and lowering blood glucose level, PPAR. Therefore, PPARY was used as a target for studying the effect of the compound agonistics are used in the treatment of type 2 diabetes, PPARY. The molecular interaction studies as shown in the table showed that compounds strongly interact with PPARY through different residues. These interaction studies reveal that compound may activate the PPARY effectively, which may overturn insulin resistance or increase the insulin stimulus. Hence, during stimulus, the compound may reduce the blood glucose level. The molecular interaction of the ligand at the active pockets of the protein is shown here. In type 2 diabetes mellitus, glucose transporters plays a vital role in physiology located in the muscle cells plays a major role. So therefore, targeting GLUT4 may ensure the therapeutic target for DM, thus activating the function of GLUT4 may increase glucose uptake in the small cells, small muscle cells in epithelial agents that will increase in the blood glucose level. In conclusion, a simple and efficient typical catalyst method for the synthesis of guanidines has been proposed. In silico studies of the synthesized compounds of potential anti-diabetic agents. Thank you.
Good morning or good afternoon, whatever the time it is. I'm Irfana Amin from JNK and I teach botany at Srinagar Women's College. Today I am presenting on the title, Effect of Benzyl Aminopurine on Growth, Flowering and Post-Harvest Performance of Antirrhinum majus. Antirrhinum majus is an ornamental flower which is commonly known by the name of snapdragons. This ornamental plant, it belongs to family Plantaginaceae. Previously, it was kept in family Scrofloriaceae. The flowering period of this ornamental starts from May and ends by September in our valley. So it has a very long flowering period here. Inflorescence is racemose, also called as spike. And this type of inflorescence is a valuable inflorescence in the commercial market. Flowers are variable in color and longevity per flower is only 4 days, which is considered to be shorter. Visible signs of senescence in this ornamental is water soaking, and after that, the flowers are shed away. Why we chose Antirrhinum majus? It's one of the most popular and widely grown garden annuals throughout the world, and also in our valley. Besides, it has many types of cultivars. The tall cultivars can be grown among shrubs in mixed borders and also with herbaceous plants. Intermediate cultivars can be grown in beds and in pots. Besides, they are also valuable as cut flowers. And the dwarf cultivars, they are used for edging and bordering formal beds. Besides, they can be grown in the rock gardens also. The flowers have several attractive color tones that emerge and shade in such a way as to provide a very pleasing effect in floral arrangements and bouquets. What was the objective? The objectives were to improve the desirable floricultural characteristics of Antirrhinum majus as bedding or as a pot plant. Now, this was kept in mind that we will be improving upon the lateral bud development, and also we will try to thereby increase the number of spikes per plant. Besides, another objective was to enhance the post-harvest performance of Antirrhinum majus as a cut flower. The experimental procedure that was followed for this work, plants of Antirrhinum majus were raised in pot soil culture in 25 centimeter earthenware pots using soil, sand, and farmyard manure in the ratio of 1:2:1. And this experiment was laid in the University Botanic Garden in the pot house. Plant growth regulator benzyl aminopurine, which is a synthetic cytokinin, was applied as foliar spray at 1, 5, and 25 milligram per liter concentration, dissolved in 0.1% tween 80, which is a surfactant, and sprayed at 4, 12, and 16 weeks after transplantation. Each concentration was represented by 5 pots with 3 plants per pot. Besides a set of 5 pots, each represented the distal water control and the tween control respectively. Plant height, fresh mass, dry mass, number of laterals, and number of inflorescences were recorded at the termination of the experiment. Post-harvest longevity of spikes and blooms was also recorded in days. The results that were achieved through this work was that benzyl aminopurine at 25 milligram per liter concentration was effective in increasing the plant height. Besides, one could easily observe the early emergence of floral buds in the plants sprayed with benzyl aminopurine. Benzyl aminopurine at the higher concentration that we used was also effective in increasing the number of laterals. However, this increase in the number of laterals did not increase the number of spikes per plant, which was expected, as BAP is known to do so. BAP was also found to be effective in enhancing the post-harvest longevity of spikes and blooms. I'll be showing you a couple of histograms here. You can observe this histogram showing how the number of laterals were increased significantly here at 25 milligram per liter. But that significant increase in the number of laterals did not improve upon the number of spikes per plant. Then in this slide, you will see how you can see a marginal increase in the longevity of spikes, as well as the longevity per bloom in the post-harvest. So what could be concluded from the study was that benzyl aminopurine at 25 milligram per liter was effective in increasing plant height, fresh mass, dry mass, and the number of laterals, besides resulting in the early emergence of floral buds than controls. There was no significant effects recorded in the number of inflorescences and flowers per plant at the harvest, presumably due to the dilution effect of the growth regulator. Benzyl aminopurine at the lower concentration we used, 1 milligram per liter, was effective in enhancing the post-harvest longevity of spikes as well as blooms. So the present study suggests that benzyl aminopurine is an effective cytokinin in enhancing the pre- and post-harvest performance of Antirrhinum majus. Thanks for listening to me.
Respected dignitaries, I am K.C. More, here to present the topic on Maturinski-based phylogenetic revision between some species of tribe Bambuaceae and Arundinary based on the theme Biodiversity and Environmental Sciences. The bamboos with about 1450 species are the only grass of family to diversify primarily in forest. Amongst bamboo species, the vegetative phase varies from 1 to 120 years. The traditional way of classification is somehow controversial in some species, and it is also not possible to totally rely on reproductive and vegetative characters. Due to irregular and delayed flowering as well as phenotypic plasticity, the researchers need to shift into molecular markers-based taxonomic identification, species discrimination, and its sustainable utilization. Materials and methods. The 14 different species of bamboos belongs to bamboo nursery, Wadadi, Amravati, were selected for the present study. The DNA extraction, fission amplification, and sequencing was done. Sequences were analyzed and species discrimination also performed. The 14 natural sequences of 14 species of Bambusoideae were analyzed, edited, and coincidental sequences were prepared by using BioEdit version 7.0 software. The coincidental sequences were submitted to NCBI via NCBI Banking Submission Gateway. The coincidental sequences were assembled allied using the software Mega. The maximum likelihood and neighbor joining trees were constructed to elucidate the taxonomic positioning of species under study. These are the species selected for the present investigation, and accession numbers were generated from the NCBI. Result and discussion. The evolutionary history was inferred by using the maximum likelihood method by using the Chimera 2 parameter model. There are three different clusters formed. The cluster first representing Gigantochlorostrata phylogenesis diagram, cluster second representing Bambusa assamica, Gigantochlora acrovolusia, Bambusa cacharensis, and Schizostachum bulua. This particular cluster representing the sister relationship between the species, and also this species evolved from the common sub-ancestor, whereas the Bambusa oliveriana representing the separate branching. Moreover, the Bambusa vulgaris, Bambusa newton, Bambusa polymorpha, and Schizostachum polymorpha representing the sister relationship also found to be evolved from the common sub-ancestor. The neighbor joining method also employed to illustrate the evolutionary history between the species. The maximum likelihood phylogenetic analysis revealed that the close relationship between Gigantochlora rostrata and Phylostasis nigra, which were traditionally placed in different tribes. Phylostasis manae, Phylostasis aurea, and Schizostachum brachylidum showing close association but evolved through separate branching. Moreover, the Bambusa assamica, Gigantochlora acrovolusia, and Bambusa cacharensis, Schizostachum bulua showing the sister relationship respectively, whereas Bambusa oliveriana were separately evolved from sub-ancestor clade. Bambusa vulgaris, Bambusa nutans, and Bambusa polymorpha and Schizostachum polymorpha showing sister relationship and represent separate common ancestral branching. This present investigation reveals that researchers wish to shift into the molecular-based taxonomic identification, because in case of bamboos, we can't rely on morphological characters and reproductive characters due to the phenotypic plasticity and irregular flowering in bamboos. This kind of molecular-based taxonomic identification will be very helpful to restoration of the biodiversity as well as the sustainable utilization of the bamboo resource. These are the references. Thank you very much.
Hello everyone, I am Kitty Kunikuju, a research scholar from the Department of Botany, Nagaland University, and I'll be presenting on the topic agrobiodiversity composition of some temporary freshwater bodies of Kohima district, Nagaland. So what are temporary water bodies? Bloslin and Schwartz in 2001 described temporary water bodies as any standing water that can hold water long enough for some species to complete their life cycle. So basically, there are water bodies which experience a cyclic phase of dry and flooding period. They are mostly fed by rain, floodings, and overflows from rivers and streams. So why do we need to study the temporary water bodies? The biological communities that are present in temporary water bodies, they are mostly different from the organisms that are present in permanent aquatic habitats. They help in recycling of nutrients, toxin removal, groundwater recharge, etc. And the cyclic nature of temporary water bodies makes them sensitive to environmental changes and fluctuating abiotic factors. So they can be used as a primary source to monitor climatic changes and global warming as a whole. In spite of the biological and environmental importance, they remain understudied and overlooked by researchers and environmentalists. Okay, why do we study agro communities in this habitat? The proper functioning of habitat, particularly temporary waters, depends on primary producers, which are mostly algae in case of aquatic habitat. Algae being the base of food chain, the growth and survival of other organisms in this special habitat hugely depends on them. They are also good indicator of environmental conditions, and they also have numerous countless other uses like industries, biofertilizers, human and animal feed, etc. So the scope of the study was taken up to understand the agro communities of this unique and special habitat, and globally there is limited work available on this topic, and this will help us to understand the ecological structure of temporary water habitats, and this can be used as a primary source to raise concerns on the importance and need of protection of this distinctive habitat. This is a map of the study area. Nagaland lies in the northeastern part of India, and the present work was done in a few locations of Kohima district in Nagaland, and this is the cheapest location and the average elevation of the study area. The algal samples were collected from 9 small and shallow rain-fed temporary pools and puddles of Kohima district. The algal samples were collected in a sample bottle, and they were preserved in a 4% formalin and stored at 40 degrees centigrade in the refrigerator. The algal samples were observed under the microscope, under different magnification, and photomicrographs were taken. And identification of algae was done by consulting standard literatures and monographs, and also the internet. Morphological characters were taken into consideration for identification of the samples. Results of discussion. A total of 26 taxa have been reported belonging to 6 classes. Signomatophysci was found to be the dominant class, where 9 algal taxa were reported. This was followed by Bacterophyci and Chlorophyci with 7 algal taxa each, and 1 algal taxa each were reported from class Cyanophyci, Trigonophyci, and Euglenophysci. The dominant genera in the temporary pool ecosystem of Kohima district were Spirogyra with 6 taxa and Odontogonium with 2 taxa. And only 3 algal taxa out of the 26 taxa that were reported were found to be pollution-tolerant algal genera. This was done according to Polar Pollution Index 1969. These can indicate good quality status of water. The dominance of Bacterophyci and Chlorophyci also indicates good quality status of the temporary water bodies of Kohima district. These are the list of the algal taxa that were reported in different classes of algae. And here are some pictures of the algal samples that were collected. Okay, in conclusion, the lack of study and management of temporary water bodies are leading to the degradation of this habitat, and the loss and endangerment of organisms is occurring. Human activities such as urbanization, pollution, etc. These are all the primary cause of the degradation of temporary habitat, and they need to be kept in check to monitor and protect specialized habitat. The present study can be a primary source for understanding the temporary water bodies, and encourage environmentalists and policymakers in taking necessary steps to protect and restore the unique and distinctive biodiversity of temporary water bodies. I'm grateful to the Department of Botany and Nagaland University for providing necessary facilities, and UGC non-lab for their financial assistance. Thank you.
Rutuja, are we going to play the second video slot now?
Hello everyone, my name is Mahila, one a research scholar from the Department of
I think we can hear
The topic of my presentation is uncooperation of 4 dominant bamboo species involved in Western Nagaland, and the topic falls under the category of biodiversity and environmental science. Bamboo is the highest yielding and renewable natural resources, there are over 1,000 species belonging to 19 genera distributed all over the world. 148 bamboo species belonging to 29 genera are found in India. The Northeast region is recognized as one of the reserve of bamboo in India. It harbors nearly 90 species of bamboo. Out of 90 species, 46 species are found in Nagaland. Bamboo and in climate change mitigation. In Nagaland, bamboo serves as an alternate to many forest products. Therefore, keeping in mind the abundance of bamboo resources, the plum growth variation among four dominant species in Mountain District subjected to same environmental regions like temperature situation. Bamboo shoot emerges during the month of May to June, and the plum inundation was observed during the month of June to November. To study the plum condition, a total of 120 microscopic plums were randomly selected, that is, 30 plums from each species. Height measurements were recorded daily for 50 days, alternate days for 3 months, and weekly during the let phase, and the average growth rate was calculated using the following formula. This is the sanitary graph. The growth curve of Ufsc Chimera shows a smooth S-curve. From the growth curve, it is observed that during the first 30 days, the plum elongation increases up to 1 to 5% per day. Contrastingly, over the next 45 to 75 days, the growth rate was rapid, ranging from 5 to 30 centimeters per day. This period is called the peak growth period. The total time period for plum elongation in Bambusa Palita was 121 days. Bambusa Palita also exhibits a smooth S-curve. During the first 30 days, the height increases up to 1 to 5 centimeters, and over the next 45 to 70 days, the growth rate was rapid, ranging from 45 to 73 centimeters per day. The total time period for plum elongation in Bambusa Palita was 110 days. Dendrocalamus S-curve also exhibits a smooth S-curve growth rate. During the first 30 days, the height increases up to 1 to 4 centimeters, and over the next 45 to 80 days, the growth rate is rapid, ranging from 20 to 35 centimeters per day, and the total time period for plum elongation in Dendrocalamus S-curve was 116 days. Dendrocalamus mandorli also exhibits a smooth S-curve. During the first 30 days, the height increases up to 1 to 3 centimeters per day, and during the next 75 to 100 days, the growth rate rapidly increases from 10 to 28 centimeters per day. The total time period for plum elongation in Dendrocalamus mandorli was 128 days. Statistics of plum height of the four dominant species was estimated. One way ANOVA was carried out to gain information about the relationship between the dependent and independent variables, and it was found that the differences among the groups were statistically significant. To compare group means, both Hpl tests was performed to uncover specific difference between the bamboo species. Comparison of the whole group shows that the difference was statistically significant by 5%. With the increase in plum height, the color of the plum also changes. In Bambusa palita, at the early stage, the plum are olive cream color with white water, and as it increases in height, the color changes to dark green with some white powder. Similarly, at initial stage, the plum color are cream covered with white powder, and it changes to dark green at mature stage. In Dendrocalamus S-curve, dull light cream changes to dull dark green. In Dendrocalamus mandorli, dull cream covered with white brown with the center is shining dark green. It can be concluded that the growth curve of each species show a smooth S-curve. The shape of the growth curve depicts the rate of plum growth pattern at different time intervals. The total time period taken for plum elongation in Bambusa palita was 110 days, Bambusa palita 121 days, Dendrocalamus S-curve 116 days, and Dendrocalamus mandorli 128 days. The plum shows slow growth rate first 30 days and start to grow rapidly till September and gradually declines during the next September and falls to zero during November. The study of the lung strategies in the present study put forward a perception towards how the bamboo species will, which grow under semi-warm conditions, shows growth pattern active conditions. These are some of my references. Thank you.
Myself, Rajtam Silvaraj, student of SIS College of Arts and Commerce, would like to present my research topic on study on effect of air pollution on plants by comparing from this site, which was edited by Ashwin Desmani. And I was going to some articles like on 9th August, I came across a paper which was stated by IPCC, which released a report stating that there's a rise in air pollution level across India and other parts of South Asia. And they have stated that the various air pollutants frequency in highest in South Asia. And another recent articles which I came across, which we all of them came across on March 2022, that the I2AR, the I2AR company have reported that reported a list of 100 most polluted places on Earth, and among them 63 were from Indian cities. So much concern about this air pollution is because its harmful effects on human beings. So this made me think about even plants are also living beings and how this air pollution will affect the plant. And that's what I came across this topic. And I researched papers and some books, and I came across that even plants are going to face abiotic stresses, and among them one is the air pollutant. And I found out that that affects the physiological, morphological, and biochemical constituents of plant, and which is going to indirectly affect the plant growth. And it's how it happens is it first enters through the leaves, and it destroys the cells and reduces the constituents of photosynthetic pigment, and which indirectly is going to affect the biochemical process. Various authors have also researched and they found out the effect of air pollution on various biochemical parameters of plants. And I reviewed some of the research papers. I reviewed almost 34 research papers, and I made sure I reviewed in such a way that the research which has been carried out not only in Maharashtra but in other states of India and as well as in different countries. And this is the two interesting findings which I came across. One was from which is recently published on Sarita. That team they have researched in Nepal, and they have found out that there's a biochemical difference between polluted and non-polluted site, and they have stated fibers is tolerant to air pollution. And another paper which was published around 2021, they have did a research in Pune, and they have found out that in all the biochemical parameters, the concentration is low in all the parameters, and they have found out that Dilomyceta is tolerant to air pollution. Fiber species is intermediate, and Diamondis indica is sensitive to air pollution. And these are my aims and objectives. This is the my study area. It's the RIT Road Vadala, which is the polluted area, and Maharashtra Nature Park Mai, this is the non-polluted area. And materials and method, I studied the plant species that is called the longifolia, and Ficus religiosa, Azalea indica, it's a green plant. And the physiological analysis which I performed was chlorophyll estimation, pH of leaf extract, relative water content, estimation of ascorbic acid. So these various parameters was calculated, and from these values, API value was calculated. This is how I performed chlorophyll estimation, and this is how I performed the pH estimation. And this is how I performed the ascorbic acid when it was standard value. And so the other plants, how I perform the ascorbic acid. And this is some other parameters from metal index, protein estimation, reducing sugar estimation, dust capturing capacity. So this is how I perform protein estimation. You can see the difference in the color of the extract for the longifolia from polluted and non-polluted site. So the agent was prepared after incubation. You can see the color variants. And the reading was taken. So I estimated the sugar estimation and performed it. This is the simple extract of Ficus religiosa after keeping it under degree water bath. This is the color various which you can see sample as well as the standard one. And the observance reading was taken. The dust capturing capacity of polluted site. And it's the different discussion. So from this, we can clearly see that from chlorophyll content, relative water content, ascorbic acid, and pH of these structures, we can observe that yes, there's a difference. There's a significant difference in polluted and non-polluted area. And you can see in polluted area, the value is less by comparing non-polluted area in all the plant species and all these parameters. And you can clearly observe that in other indica, so in neem plant, the value, the concentration is high in all the parameters, and in Polyethyl longifolia, the concentration is low in chlorophyll, relative water content, ascorbic acid, and pH. And you can see that the Phytochemical religiosa is having value lower than neem and higher than Polyethyl longifolia. And through those values, we calculated the API value, and this is the standard value. And from this, we can see that comparison that Azada indica is having a value which is tolerant to air pollution, Phytochemical religiosa is intermediate to air pollution, Polyethyl longifolia is sensitive to air pollution. So the other parameters which I performed was carotenoids and stomatal index. So you also can see that the Azada indica is having the high value, and Polyethyl longifolia is having the lower value, even the stomatal index also. And that's the difference in polluted and non-polluted area. Polluted area is having the less value in all these parameters. And this is it. In estimation, sugar estimation, through this standard graph, we were able to estimate the protein value. And in so we are also in Polyethyl longifolia is having the less value in sugar as well as in protein estimation, and Azada indica is having the highest value. This shows that how Polyethyl longifolia is from various parameters. You can see that how Polyethyl longifolia is affected by the air pollution, because you can see in all the parameters the value is low compared to other both parameters. So I have an estimated dust capturing capacity. It was observed that in Phytochemical religiosa, the dust capturing they have good dust capturing capacity because of the surface area. And these are the interesting findings which I came across after analyzing after all partial. Suddenly I came across this this findings. That is, by comparing among the plant species, in polluted and non-polluted side, it was observed that of course there is difference. In polluted side, the value is low in all biochemical parameters in all the plant species, but the rate of reduction, it is the rate of reduction is different in different species. For example, if we take in polluted side, the pH of Azada indica is 7.21, and in non-polluted side, okay, and in polluted side it is 7.15. So the rate of reduction is that, but it is less while comparing with other plant species. For example, the same Polyethyl longifolia in non-polluted side it is 5.57, but if you see in polluted side it is 3.8. That is, the rate of reduction over here is more. Okay, so this is not only observed in only one particular parameter, but in all the parameters, if you see, you can see the rate of difference is less. It's more in Polyethyl longifolia, and it is less in Azada indica plant, which says that how that portion is affected the Polyethyl longifolia plant in the biochemical parameters. And my conclusion is, yes, in all biochemical parameters, the value is low in polluted side compared to non-polluted side. As I said now, the rate of difference, the reduction is different in all cited plant species based on the tolerant level to the air pollution. The dust capturing capacity was more in polluted side. The Azada indica is tolerant to air pollution. The Phytochemical is intermediate, and Polyethyl longifolia is sensitive to air pollution. The sensitivity is reason because their biochemical processes have been largely affected because of the air pollution. And you can say that Azada indica it can be used for mitigation for green plague development, and Polyethyl longifolia can be used for biomonitoring the air pollution. And my policy significance is, from this finding, to reveal the effect of physiological response on the plant in polluted area. We were able to identify the tolerant plant and sensitive plant. We were able to know the reason. We can knowing it now why we should plant tolerant plant species in the polluted areas. And this research we can study, we can relate, comparing more plant species in increasing the geographical area, and in future, by doing more number of plant, that can lead for designing a better green belt in roadside, polluted side, traffic islands, in urban areas. And these are my reference. I mentioned I refer very recent articles. Thank you.
Hello everyone, I'm Neha Sinha, and today I'm here to present my review paper on the title Dermatoglycotoxic Excellent in Milk, Fruit improvement through selection using morphological and physiological characters against biotic stress under the category Biodiversity and Environmental Science. Tomato fruit borers are one of the most common pests which greatly influence the growth and development of tomato plants, which indeed even affect the yield production of the plants, the tomato plant. In this review paper, an attempt is made to study the tomato improvement programs against the fruit borer using morphological characters and chlorophyll estimation, and how molecular studies can help in future breeding programs. So it's we all know that the tomato is an active member of the Solanaceous family, and it is the second most producing vegetable after potato across the world. It is consumed by 25% of the Indian population, yet the productivity is low due to the number of factors, in which insect pests being the most prominent one. And in order to control these pests, chemical methods are widely used by the farmers, and it is efficient, and it is very necessary to produce an insect resistant crops. In order to discourage the use of these chemical methods, tomato is more prone to insect pests and diseases, mainly due to its tenderness and softness, and fruit borer cause high damage to the crop because of its texture. The seedlings are susceptible to the tomato fruit borer, Helicoverpa armigera, also known as H. armigera, which causes necrosis in the chlorophyll tissue of the leaf and renders mature fruits unfit for consumption. They are plagued by a series of pests from seedling to harvest, and there is an urgent need for the development of non-chemical management solutions by biological, mechanical, cultural, and post-plant medicine. Control by insecticides has several deleterious effects, including environmental pollution, health hazards, and pest resurgence, etc. Thus, this becomes the key factor for the future of agriculture. It is necessary to cultivate a resistant and tolerant variety against fruit borer, which becomes fundamental in improving the cultivars. The agronomic traits such as morphological characters and chlorophyll estimation and molecular studies can help in future breeding programs in selection of genotypes with desired traits towards variety development. In order to develop an insect-resistant variety, agronomic traits are being studied. So, agronomic traits can be studied using the morphological characters and chlorophyll estimation. So, morphological characters are studied as a first the selection of different varieties is done by it can be any local or a wild variety, and they are sold in the main fields, and no chemical measures are being carried out in order to study the rate of infestation by Helicoverpa armigera in it, and they are allowed to get infested as no chemical measures are being carried out. And after fruiting, the morphological studies are carried, and the fittest varieties are used to create a transgenic or an insect-resistant variety. So the morphological characters are studied as follows. They are the leaf hair number per unit, plant height, stem diameter, number of branches per plant, number of leaves per plant, second leaf area, number of fruits per plant, and the total chlorophyll content is being studied. As we all know that the molecular studies plays a very efficient role in the today's modern science. So the molecular studies are carried out by creating molecular maps, quantitative trait loci, also known as the QTLs, integration lines, CRISPR-Cas-based targeted mutagenesis, CRISPR-Cas-based precise DNA changes or replacements in which DNA editings are done to create an insect tolerant or antibiotic stress resistant kind of variety, and accelerated allele integration and day domestication are done under the molecular studies to create an insect resistant variety. And with this, I would like to conclude my presentation. Due to the softness and fragility, tomato plants are especially susceptible to pest infestation, and the high infestation rate has an indirect effect on the lycosicum overall yield production. So to get rid of this pest, growers are of course going to get relied on the chemical methods, and which is not only suitable for human consumption, but it even affects the soil. We are all aware of the safe soil that is now being going on by the Asian population, which states that the soil, the microbial content of the soil is dying because of the high chemical use, and because of that, the soil is now turning just into sand, so it is even affecting the sand. So as a result, the greatest alternative for safe consumption and high yield production is to produce a pest resistant crop. We can determine which kinds are more likely to be suitable for generating hybrids and transgenic crops based on the infestation rate using the approach of identifying morphological features and chlorophyll estimation. This method not only provides a better understanding of the suitable variety, but it also aids in estimating pest behavior on each portion of the plant, and it is incredibly simple and inexpensive, and use of this technique can lead to a better crop production, that is a more insect tolerant variety, and it is possible to gain a better understanding of which variety should be chosen for the best results. References, and I would like to thank the Botany Department of Guru Nanak Khalsa College for organizing the international conference and providing me a platform to present my review video. I would also like to thank the audience for patiently listening to me. Thank you so much. A very good morning to the respected chairperson and to all the dignitaries. My name is MG Falguna Monsang, and I will be presenting on the topic changes in soil carbon and nutrients under different forests in Manipur, Northeast India. Soil is a natural resource which is obtained from rock-led rain. It functions as a feedback system of water and nutrients for the crops to grow and reproduce. Also, soil provides terrestrial habitat for all forms of life. Further, vegetation affects the soil formation and structure because of a complex interrelation between soil and forest vegetation. In addition, forests are the major carbon pool in the terrestrial ecosystem, storing 50% in its vegetation and 50% in soil. As such, soil properties are important in determining soil productivity and forest site quality. The objectives of the present study were to determine soil physicochemical properties of natural pine forests, pine plantation, and lithocarpus species dominated forests of Manipur, Northeast India, and to assess soil carbon and nutrient pools in different forest types of Manipur. Materials and methods. Study area. The study was conducted in 3 forest types of Manipur, that is, natural pine forest, pine plantation, and lithocarpus species dominated forest. Here the table represents the forest type and its coordination and district of the study area, as well as the map showing the study areas. Soil collection. Soils were collected randomly from 3 caves at 9 locations, and 3 samples were composited to make it 1 and further sieved through 2 mm nails, which were used for analysis of different soil parameters. Soil analysis. Soil texture was assessed by hydrometer method, and soil moisture content by gravimetric method, soil pH by digital pH meter, water holding capacity using the kin box technique, bulk density using the core method. Wilkie and Black method, gel down method, Gray and Butch method, clamp photometer were used to determine soil organic carbon, total nitrogen, total phosphorus, and exchangeable potassium. Statistical analysis. One day and why and these significant designs were performed to test for significant differences between soil depths and among the site. Results. Figure 1, 2, 3, and 4 shows different soil physical properties of the study sites. In this figure, it can be seen that soil moisture content, soil organic matter, water holding capacity were found significantly high in pine plantation, the natural pine forest, and lithocarpus forest, which may be attributed to higher elevation in this region where temperatures are low and accumulates more soil organic matter, thus making soil porous and rich, thereby lowering the bulk density in the study sites comparing to natural pine forest and lithocarpus forest. Table 1. It shows the soil texture for different forest types. Here, the study sites have all the same textural classes, that is sandy loam. And also it can be seen that the sand percentage is more higher in natural pine forest and lithocarpus forest than pine plantation, which may be the reason for low bulk density and soil organic matter in these sites. Table 2 shows soil pH, carbon, and nutrients of different forest types. Here, the pH values are lower in lithocarpus forest, followed by natural pine forest. As for soil organic carbon, it is higher in soil surface layer, and it decreases with increase in depth in all the forest type, which may be due to the higher input of carbon to the surface soil layer. However, the content of nutrients vary within different soil depths and across different forest types. It can be seen that Lithocarpus forests have high little nitrogen, which may be due to the presence of tree species that accumulates higher little quantity, since little accumulation are directly related to the total nitrogen contained in soil, as per report by Alejandrin Guerrero et al. 2006 and Augusto et al. 2012. Conclusion. The result of the present study indicates that pine plantations have significant stock of soil organic carbon than natural pine forests and lithocarpus forests, which may be attributed to high accumulation of organic matter and slow decomposition of pine leaf. Further, results indicate that pine plantation soil is rich in physical properties which may be associated to higher altitudes that may hinder the decomposition rate and microbial activities because of low temperature, thereby enhancing the accumulation of organic matter and carbon. Therefore, it is concluded that pine plantations serve as a potential site for carbon accumulation and climate change mitigation. Here is the references. Thank you.
Hello everyone, I'm Nidhi Goswami from the Center of Excellence in Astrobiology, Hamilton University, Mumbai, and I will be presenting our work on the culture-dependent microbial diversity studies of thermophiles from potential mass analog Buga Hot Springs, Ladakh, India. Throughout the presentation, I will be covering the following topics. What is astrobiology? The astrobiology seeks to understand the fundamental question of the origins, evolution, and distribution of life on Earth and other planetary bodies. Astrobiological studies are interdisciplinary in nature and focused on discovering the existence and evidence of life on Mars, Europa, which is the moon of Jupiter, and Titan and Enceladus, which are the moons of Saturn. To understand the evidence of life on Mars and other planetary bodies, it is essential to study the terrestrial analog sites. These sites host multiple extreme environments and are home to extremophiles, which are the microbes thriving in extreme environments. Multiple research stations, such as the Flashline Mars Amity Research Station, Mars Amity Research Station in Ladakh, and the Mars Desert Research Station in the US, facilitate the research of these terrestrial Mars analog sites. Studying extremophiles from these analogs may help serve them as potential model organisms for astrobiological studies. Ladakh is a very high altitude cold Himalayan desert which hosts various near pristine extreme environments. These include permafrost mountains, sand dunes, hypersaline lakes, sand rams, and hot springs. The geological settings, saline waters, and the rain-shadowed region of Ladakh has a resemblance with the Noachian-Hesperian transition of ancient Mars, which is proposed to have water bodies and a probable habitable environment. Hence, Ladakh serves as a potential terrestrial Mars analog site. During the Earth and Space Exploration Program, which is a field expedition in Ladakh organized by Amity University, Mumbai, we collected samples from Buga Valley. The astrobiological relevance of Buga Valley includes the hypothesis for the origins of life, which is based on the terrestrial hot springs. The previous astrobiological studies from the region include work done in prebiotic chemistry. The present work is among the first to study the culture-dependent microbial diversity of thermophiles from the region. These microbes tolerate multiple extreme environments, including hot and cold temperatures, higher UV radiation, and lesser atmospheric pressure and oxygen levels. The sample collection was done from hot springs as seen in figure A and D. The region also includes sulfur deposits as seen in figure B and halite crust as seen in figure C. The samples were collected and stored at 4 degrees Celsius. The sediment and water samples were enriched in nutrient broth and incubated at 50 degrees Celsius. The enriched culture was spread onto nutrient agar and restreaking was done till pure colonies were obtained and preserved in glycerol stocks at minus 20 degrees Celsius. These isolates were biochemically characterized for the production of hydrolytic enzymes and tested for their polychemophilic properties and further centered for molecular identification. So we have isolated 3 thermophiles from the water and sediment samples of Buga. These isolates have been tested for qualitative enzyme activity and polychemophils. The thermophiles are moderately halophilic and facultative alkali-phils, and they also test positive for the production of protease, lipase, amylase, and urease enzymes, as seen in figure 4. The molecular identification was done by PCR amplification of the 16 srRNA gene and its subsequent sequencing. In the present study, we have 3 thermophilic isolates which are identified as Deprimonas ignava, Bacillus anisi, and Anoxibacillus flavitolens. These isolates show positive enzyme activity in production of exopolysaccharides and polyextremophilic properties. Our future work includes the screening of these isolates for the potential industrial applications. To conclude, exploring such mass analog sites from a microbiology perspective will provide insights into the microbial diversity in extreme environments and their potential role as model organisms for, but not limited to, future astrobiological studies. Thank you.
Respected judges, organizing committee members, and all the participants present over here, greetings of the day. My name is Dr. Nikhil Devi, Assistant Professor from Viva College of Arts, Commerce and Science, and I'm here to present a research topic entitled Application of Nitrilotriacetic Acid Modified Pods of Phytochemical for the removal of hexavalent chromium from aqueous solution, a sustainable approach. So basically, in this research work, we have used pods of phytocarbon as a green adsorbent, and in order to modify their bioabsorption potential for the removal of hexavalent chromium, we have used nitrilotriacetic acid, which is readily biodegradable chelating agent, and thus the approach becomes sustainable one. Let's start with the introduction part. As we all know that water, a precious resource, will sustain all form of life on the earth. But due to rapid expansion in the field of agriculture, commerce, industry, and healthcare sector, tremendous amount of toxic heavy metals are released into the environment. Thus, the release of heavy metals into aquatic ecosystem has become a major problem of concern in India over the last few decades. There are so many different conventional methods which are available in order to remove heavy metals. But if you consider all these techniques, most of the techniques are costly, most of the time inefficient and time consuming. And thus, there is a need to find an alternate technique which is cost effective, eco-friendly as well as efficient. And thus bioabsorption comes into the picture. So bioabsorption can be defined as removal or binding of desired contaminant from aqueous solution by biological material. Hence, this research is aiming to solve the problem by using a low-cost plant-based waste material as a clean adsorbent and their modification by using biodegradable chelating agent to increase the sorption potential for the removal of hexavalent chromium ions from aqueous solution. Next section, that is methodology. In this, the first step is preparation of green adsorbent PPNA, that is nothing but nitrilotriacetic acid modified pods of phytochemical, which was carried out by similar method described by Marshall et al. In 1999. After that, batch bioabsorption studies were systematically carried out by considering effect of pH, contact time, temperature, adsorbent dose, agitation rate, and initial chromium ion concentration. Adsorption isotome studies were also carried out by considering 4 different adsorption isotome models such as Langmuir, Freundlich, DKR, and Temkin. Further adsorption kinetic studies were carried out by considering pseudo first order, pseudo second order, Lovich, and Weber and Morris intraparticular diffusion kinetic model. Thermodynamic studies were carried out by considering gas-free energy, change in enthalpy, and change in entropy. After that, desorption studies were carried out by 4 different elements such as 0.1 molar NaOH, 0.1 molar H2SO4, 0.1 molar HNO3, and 0.1 molar HCl. And finally, determination of calorific value were carried out by using bomb colorimeter before and after chromium adsorption by ppNPL. Next is result and discussion. As you can see, the table 1 gives you an idea about the optimized parameter with optimum chromium removal value. Optimum pH were found to be pH 2 with 94.417% of chromium removal. Optimum contact time were found to be 90 minutes with 93.803% of chromium removal. Optimum temperature were found to be 60 degrees Celsius with 95.935% of chromium removal. Optimum adsorbent dose were found to be 0.6 gram with 94.262% of chromium removal. Optimum aggregation rate were found to be 120 rpm with 94.791% of chromium removal. And last parameter is effect of initial heavy metal ion concentration. The reach was 20 to 100 ppm that we have selected. 20 ppm were found to be optimized parameter with 96.005% of chromium removal. In figure 1, it gives you an idea about graphical representation of different adsorption isotome model that we have selected for the study. That is Langmuir, Freundlich, DKR, and Temkin. Table 2 gives you an idea about adsorption isotome constant for chromium bioabsorption by ppNPL. As you can see, the R square, that is regression coefficient value, is maximum for the Freundlich adsorption isotome as compared to the other 3 isotome model. Therefore, the chromium bioabsorption by ppNPA clearly follows Freundlich adsorption isotome model, which indicates multi-layer adsorption mechanism between adsorbent and adsorbate. Next. Figure 3 gives you an idea about graphical representation of different adsorption kinetic models such as pseudo first order, pseudo second order, Lobich and Weber and Morris intraparticular diffusion for chromium bioabsorption by PP and TA. Table 3 indicates the adsorption kinetic data for chromium bioabsorption by PP and TA. As you can see, the R square, that is regression coefficient value, is maximum for pseudo second order rate kinetic model as compared to the other three kinetic models. Therefore, the chromium bioabsorption by PP and T clearly follows pseudo second order rate kinetic model, which clearly indicates the bioabsorption mechanism is chemisorption, which means there is a chemical bond formation take place between adsorbent and adsorbent. Next. In figure 4, it indicates the plot of LFKC against 1 upon T for the determination of thermodynamic parameter for chromium bioabsorption by PP and T. As you can see in the figure, in the table 4, the T value is negative, which indicates the bioabsorption process is spontaneous. The enthalpy change value is positive, which indicates the bioabsorption process is endothermic, and the change in entropy value is also a positive, which indicating bioabsorption process proceeded with increased randomness. As you can see, the figure 5 indicates the result of desorption studies. 0.1 molar HNO3 showed good desorption potential, that is 62.447%, which is a maximum as compared to the other three eluents for chromium desorption. As you can see in the table 5, the calorific value has been increased after chromium bioabsorption as compared to the before chromium adsorption. The calorific value after chromium bioabsorption were found to be 4,274.63 calories per gram, which is equivalent to E-grade charcoal. Thus, whatever waste is generated after performing bioabsorption in terms of biosorbent, that can be easily channelized in order to obtain the thermal energy. Next, in conclusion, the chromium removal efficiency of PP and T was found to be dependent on all the process parameters. The equilibrium data best fitted to frontline adsorption isotherm model and pseudo second order rate kinetic model. Thermodynamic study revealed that the bioabsorption process was endothermic spontaneous with increased randomness. 0.1 molar HNO3 responsible for maximum chromium desorption, and it was observed that the calorific value of green adsorbent has significantly increased after metal ion adsorption. Hence, the approach is sustainable in nature, owing to the fact that the green adsorbent as well as chelating agent used for the modification of green adsorbent is biodegradable. These are the references that we have considered for this particular research work. Thank you for attending this session. Thank you.
Good morning, everyone. I am Pallavi from Wph University. Today, I am here to present the research paper presentation on the title Seasonal Variations in Diversity of Aquatic Macrophytes of Tadikatte Lake, Chitradurga District, Karnataka, India, under the guidance of Dr. Parmeshwar Nayak P. Let's move on to the introduction. The term aquatic macrophytes refers to a diverse group of aquatic photosynthetic organisms, all large enough to see with naked eye. It includes macroalgae of the division Chlorophyta, Xanthophyta, Rhodophyta, and blue-green algae, Bryophyta, Pteridophyta, and Spermatophyta. The vegetative parts of which actively grow either permanently or periodically submerged below, floating on or growing up through water surface. Aquatic macrophytes are classified into 4 types, such as submerged, floating lived, emergent, and free-floating. These plants are capable of colonizing several kinds of aquatic environment, for example, lakes, lagoons, wetlands, rivers, reservoirs, waterfalls, and even bromeliad tanks, with a wide range of limnological features presenting high plasticity and adaptation ability. Aquatic macrophytes are known as highly productive and with an important structuring role in aquatic environments. Macrophytes, though providing food and shelter for aquatic animals. Let's move on to the materials and methods. Study area. Tadikatte Lake is situated in Valkere Taluk, Chitradurga district of Karnataka, India. The area lies between the latitude 13 degree 9 angstrom 19 second north, 76 degree 14 angstrom 6 minute 6 second east, at a mean altitude 727 meters above the sea level. Tadikatte Lake covers a geographical area of 71.22 hectares. About 35% of lake is occupied by aquatic macrophyte. This is the location map of Tadikatte Lake. Collection of macrophytes and their analysis. Macrophytes in shallow water were collected by hand, while those from deeper water were collected with the help of long-handled hook and wherever possible using the local fisherman community Collection, the specimen was thoroughly washed, excess water soaked with the filter paper, kept in a polythene bags lined with the filter paper, preserved according to herbarium techniques. Collected materials were brought to the laboratory and identified using with the help of standard literature and confirmed in the Herbarium of Botanical Survey of India. Move on to the result and discussion. The aquatic macrophytes in Tadipata Lake were observed, classified, and presented in Table 1. All 40 aquatic macrophyte species were recorded belonging to 4 different groups such as free-floating, submerged, emergent, and marginal macrophytes. Out of 40 macrophytes, among 24 marginal, 1 free-floating, 8 submerged, and 7 emergent macrophytes were present in the lake basin. Aquatic plant serves as a good food source for humans and animals, thus forming a palatable food for water birds and best for aquatic wildlife conservation practices. Aquatic vesicular plants are essential indicators of water pollution. Many workers contributed to recent aspects of production studies of macrophytes. In this present study, Ipomia pistulosa was enormously distributed in Talikata Lake, and the other species were also found. In this lake, entry of domestic wastewater is the standard way that various nutrients enter the aquatic ecosystem, resulting in the death of those systems. And Ipomia pistulosa is one of the most productive plant on the earth. They are emergent freshwater bodies. This is the list of aquatic macrophytes in Talikate Lake. This is the photographs I have taken during the study period. They are Ipomia pistulosa, Polygonum glabrum, Cyprus longus, Elatica palustris, Potamochitan species, Najas marina, and other species also. Conclusion. The aquatic macrophytes vary in different habitats based on their nutritional component present in the water bodies across the globe. The Tadipata Lake in Chindradurga district is a serene water body harboring different macrophytes. The rich biodiversity of macrophytes supports a large number of Ipomia pistulosa. These two in all seasons. However, during the present study, it has been found that due to natural and anthropogenic activity, along with the negligence of concerned government authorities, the process of gradual degradation of wetland has been started. Therefore, proper conservation measures should be taken for the conservation and existence of this important wetland. These are the references. Thank you.
Myself, Dr. Pallavi Singh, and my presentation topic is analysis of molecular data of some species of genus Junonia butterfly using RFPD technique. It comes under theme biodiversity and environmental science. The butterfly taken under investigation comes in the family Nymphalidae, which is the largest family with 12 subfamilies and around 6,000 species. They are also termed as brush-footed butterflies or four-footed, as their first pair of legs it is much reduced. Butterflies, they are very susceptible to the changing environment, and therefore they act as a key taxa for monitoring biodiversity. If there is loss of biodiversity, this means it poses a threat to the species which are restricted geographically. Many phylogenetic studies have been done on various species, and that gives an idea about the relationship within the genera, taxa, or the evolutionary history among the population. This evolutionary data it can be supported by principal component analysis, and in which this component analysis it selects very selective characters as a principal component and gives a clear idea in a form of simple dot map. For the investigation, first the butterflies they were collected on the ground of primary characters, that is, the first pair of legs are reduced, so they could be identified as inthalids. When they are collected, the legs they were preserved in 70% alcohol. The rest of the body it was oven dried for correct identification of the taxa. In the laboratory, the segments that were preserved in 70% alcohol, they were subjected for extraction of the DNA according to the protocol mentioned in the kit.
After obtaining the DNA, quantification and visualization of the extracted DNA took place just for confirmation of the DNA. Later, the extracted material was amplified by means of polymerase chain reactions. When the amplicons were obtained, the data it was converted into binary data as 0, 1 for presence or absence of the character, and that was converted into statistical form for generating the phylogenetic tree. And later, this was supported by generation of principal component, and this was supported by the dot map provided by principal component analysis. The result that was observed was the dendrogram. You can see the dendrogram was obtained, and that shows two different taxas. Of this, one clade, it shows Junonia hyata and Junonia lemonias as sister taxa, and the another clade, it shows Junonia alevana and Junonia audithia as sister taxa. That is showing the closeness between the two taxa. Further, similarity coefficient, this reveals that there is maximum genetic similarity between alevana and audithia, and whereas the Junonia lemonias and audithia, they are distantly located. This data it could be supported by means of principal component analysis, where we can see that Junonia audithia, it is completely diverging out, and it is distantly located from Junonia lemonias. This reveals that there is closeness within two species and in their genetic makeup, whereas some genetic characteristics are expressed in a specific species. For example, the primer OPP18, this showed highly distinct species-specific bands, and that was ranging below 200 base pairs. So these could be considered as diagnostic bands. The conclusion from the study, it is amplified fragments, they were repeated in the genome and in varying degrees. So this RAPD PCR technique, it could provide a powerful tool to improve the species identification, whereas they are also helpful to understand better genetic variability and points where divergence is occurring within the genus. Also, the presence of species-specific bands among some species, it suggests that interspecific genetic relatedness does occur between them. This was my presentation. Thank you.
Good day to everyone. I am Dr. Payal Ajrekar from the Department of Botany, Jai Hind College, Autonomous, Mumbai, and I will be speaking on stomatal index, leaf thickness, and noise absorption studies of some plants. I have done my work under the guidance of Dr. Ambika Joshi. The talk will be subdivided into the following headings, such as basic introduction, materials and methodology, results and discussion, conclusion, future prospects, and references. To start off with, why this topic? As you must be aware, with the increase in urbanization and population explosion, there is also an increase in the number of industries and vehicles that lead to release of harmful chemicals, further leading to air pollution. But at the same time, these also manage to generate and release excessive noise. For a very long time, noise was not even considered worthy of a pollutant, and nobody gave it any significance or importance. It was later discovered, thanks to the WHO report, that a lot of health ailments were a result of the direct impact of noise. Now, it has been proven time and time again that trees play a major role in reducing air pollution, and there is also ample evidence that there is effect of music on the growth of plants. So why not use these trees in abatement of noise? But will planting random trees help? Another study has already shown us that the shape of the tree canopy plays a major role in abatement, and that the conical canopy shape absorbs more noise in comparison to the other shapes. We, on the other hand, are trying to emphasize on the sound absorption capacity of the leaves, the stomatal index, and the leaf thickness. As observed in this image, here we come across various types of leaves collected from a survey: simple, compound, big, small, linear, overt, chordate, and so on. Now let's proceed to see how they can contribute in noise abatement. To start off with, the major aim of this research was to make use of inexpensive and effective methods for the purpose of noise studies and at the same time noise abatement. There could have been advanced expensive methods for the same, but we chose the simpler one. The procedure is fairly simple and yet a little time consuming due to the consideration of multiple parameters. We need to do a survey of all the tree species that are commonly found in and around the study area. I have done a survey in Greater Mumbai. Then from the results of these tree surveys, we need to bring the leaves of those tree species for the sound absorption capacities and for stomatal index as well as leaf thickness. As seen in the image on the bottom left hand side, this is the experimental setup. Now an interesting fact about this setup is that it is a simple physics experiment which is used at the third year. We have tweaked the procedure a little bit to apply it in biophysics. We have replaced the material with our leaf material in the path of sound, and it is thus recorded by a microphone which is read on the CRO or oscilloscope in the form of wave pattern, as you see in the image in the center. This wave pattern is further used for calculations in Sabine's ratio. Lower the ratio or lower the value, higher is the sound absorption capacity and vice versa. The second step is to use these selected leaf samples and measure their stomatal index. The method followed is the standard procedure. And thirdly, we measure the leaf thickness using a screw gauge. Now let's see what the observations have to tell us about this experiment. The results of sound absorption capacities were correlated with stomatal index and leaf thickness, and it is clearly seen that there is no direct relation between leaf thickness and stomatal index, but it definitely has a relation with the sound absorption capacity. It is also seen that the absorption capacities are better at lower frequencies than at higher frequencies. It was also concluded that Caesium pumini and Ficus hispida showed the best absorption capacities. At the same time, their leaf thickness was fairly large and they had a bigger or larger stomatal index too. Whereas compound leaves showed poor absorption capacity. It can also be concluded that denser the canopy of such leaves, better is the sound abatement. We wish to future prospects. We wish to recommend appropriate tree species which can not only help us in town planning, but at the same time help us in producing more oxygen, reduce air pollution, and abate noise. And we also aim to continue research in the field, in this field, with the help of the current study to produce better suggestions and recommendations. These are my few references. Thank you very much.
Hello everyone, I am Shweta Deshmukh, research scholar from Department of Botany of the Pancreatic of University of Turkey. Here I am delivering the morphological evaluation of William Hplc under the climatic conditions of Maharashtra. William is a genus which belongs to the family Rubiaceae and it comprises near about 100 species and over the cultivars. It is distributed throughout the world, especially the northern hemisphere, Asia, North America, and Europe. But the China, Nepal, Korea, and Japan are the gene centers of this genus. William is a very important ornamental plant because it is the fourth important flower in the ranking of flower position and it offers the second position in the flower bulbs. Lilian flowers are the sign of love, beauty, and tranquility as they are pure. In horticulture, the Lilian genera is very important because for the pot plantation and for the blood flower production, which gives economical importance and the commercialization importance to the Lilian. Lilium is a superior flower because it having the longevity, fragrance, beauty, and its size. It is gaining more popularity because it has the attractive flower, wide ranges of color, and it possesses the rehydrated ability because the horticulture is appreciated living and nowadays Lilium breeding is the main important topic in the world. Lilium is mainly classified into the three groups: globicular, ascending, and vertical hybrid. This belongs to the different taxonomical sections. Here I am using the globicular ascending, which shows a large variation in the flower color. The present investigation is the array hybrids Lilium morphological studies, which gives the variation in the morphology as well as in the especially to the flower characters. In morphological analysis, I use the three excisions of the hybrid, and I have collected this sample from the CSIR ISBT Palanpur, Himachal Pradesh in the winter season and in winter as a stock in our department. I used thin hybrid, bright diamond, Erimo and natural, which shows the variation in the qualitative as well as the qualitative traits of morphology. Here you can observe the bright diamond, Erimo and natural, which gives the three different colors of flower. One is the white color, orange color and the yellow color. And here you can see the arrangement of leaves, how compact, how they, how they are arranged and the facing of flower, the numbers of flowers, the position of flower, they gives the variation in the morphological characters. As you can see here, the outer temple, inner temple of the flower shows a differentiation. Here you can see there is no variance in the writer. But in case of the Erimo, it shows the more diameter, brightly colored, the anthocytic stigma is different, and the outer temple, inner temple is very, and it shows as a furrow in the middle. Whereas naturally, it is also belong to the same group, but in the variance or the diameter is totally different among the cultivars. Here you can see the maximum plant height was shown by the Erimo, whereas the length of leaf and breadth of leaf is more observed in the natural cultivar. The number of leaves and the number of leaves at the flowering condition is also maximum in the Erimo cultivar. The tail width is near about more in the Erimo cultivar. The length of fruit is maximum shown by the bright diamond, whereas the bulb length and bulb width is maximum in Erimo. Also, number of scale was observed in maximum shown in the Erimo as compared to the other two cultivars. In case of the floral characters, the number of flower per plant was maximum in Erimo. The flower length was maximum shown by the plant. The pedicel length is maximum, pedicel outer tiple length is maximum in the Erimo, whereas the inner tiple length and inner tiple width is maximum in Erimo, but near about the inner tiple width is near about same in these two cultivar right diamond and the Erimo. The stamen length is maximum shown by the right diamond. But as compared to the all floral characters, the maximum ratio was shown by the Erimo as compared to the bright diamond and natural. Hence, it was shown by the three cultivars, though there are several morphological characters was observed like their height, bud diameter, pedicel length, flower length, leaf shape, etc. But the morphological traits are maximum shown in the Erimo as well as in the reproductive traits of the lily, flower diameter, flower number, attractive flower color, flower fragrance, inner taper length, outer taper length, and their width was maximum shown by the Erimo. Hence, it was concluded that though the cultivars are origin is same in the three cultivars, namely bright diamond, Erimo, and naturally, but the best response was shown by Erimo because it gives the maximum number of flowers. Lately, reproductive traits of the plant is good, and the maximum plant height was shown by the Erimo. And early growth in the response to the climatic condition of Maharashtra, though the early cultivars is three, namely bright diamond, Erimo, and naturally, but the Erimo shows the best response in the climatic condition of Maharashtra. These are my references.
requested to go to respective candidates. The name of my presentation is changes in the floral damage to the livelihood process because of the zone of long-term wildlife sanctuary in the whole district of Meghalaya. The going person in everyone is a beautiful fact. Last 5 decades, fossil more simply incomparable time in history. Demand of the non-forest product is one of the prime causes of forest degradation, species loss, and progressively since altered ecosystem today now 40% of the country are living in forest challenges, and proportionally these each basic cutting this is occurring at an alarming rate. Monoculture plantation are a major threat to the survival of several species. Nowadays, contribute to 59% of the forest cover. In Meghalaya, there's forest cover out of its 5%. Only 5% is under the state control, and the remaining is under the district council, community, and the private name. And the NRF comes with biodiversity hotspot and manages the support for conservation and protection of the area. This is also just reported about 1036 Taxa of the vegetation of the area, even after the shifting cultivation and encroachment exploitation of the area for commercial purposes of Pacific land. Some of them are threat and with the areas. Many endangered mammals species have been reported from this area, including the whole of given. As for the working plan, it is still relying on it because it's still the clear people rely solely on the changes in the situations as per the transition of the earth. Sanctuary, as per the definition, the area of the ecological and foreign geographical, natural or geological significance, etc. The objective is to encourage the vegetation after the changes into the neighborhood forest area and district conservation within the region of the leaders to suggest appropriate measures for the forest district council area. The state area covers per kilometer, both 125 per kilometer. Government and for district council private land, 300 meters to 845 meters extent of the PHA, elevation 965, temperature 8.6, 34 degrees. The river is market natural evacuation the central river places followed surrounding river temples. We have the use of pockets for the trees, 5 to 5 shrubs, 1 to 1 for the herbs, for the seasonal, was collected seasonally. These are the formulas for and the IPA which has been calculated for the collected species from all the direction of the sanctuary. Bamboo is a very prominent in the area. Forest area, plantation area. Collected all the 4 years. You get the simulated index of the index of the forest type is observed during the sampling scarcity. Sal forest, the tropical forest, moist mix, the Himalayan moist mix. Also, later in discussion, overall, there have been 31 number species which have been protected area, whereas the total numbers in 4 areas surrounding the PHA. So, IBI rendered from 56% to 60% with a dominant specifics for your robustness, Navalicha, etc. Diversity is maximum the ladder to this, minimum of the this, stability as well. There's a reduced flora in the district council area and the base size of flora in the village area.
various large diversity has been found protected area. The secondary growth of antibacterial interaction which is implicated in whereas products don't validate illegal in because of the person because both are supplies the continuous disturbance natural forest species in the Shinon's area. Natural regional species have been observed with various auxiliary crop house shrubs and plants like etc. The poor economic conditions, lack of development farms within classic villages, and slow the villages for the trade with further small timber and to copy the gene collections, contribute reduction of the plant diversity in the Shinon's area. The livelihood option like this common patients less preferred by the in conclusion, to ensure the communication the community to ensure proper enforcement of the basic rules, to promote the regulated to provide conservation friendly sustainable livelihood standards, to preserve of green species, scientific, technical approach for agricultural purpose, social awareness, higher education institution and developing hospitals, and to provide agricultural skill job training. Thank you.
Hello, good morning to all of you. Myself Vinod Kumar. Today I'm going to present my research paper entitled as Isolation, Morphology, Characterization and Screening of Interphytic Fungi for Enzyme Cellulase isolated from the different parts of mangrove plant Avicennia marina in the category of biodiversity. Now, as we all know that mangrove forest play an important role in subtropical and tropical coastal ecosystem. The structural complexities of mangrove vegetation create an unique environment which provide ecological niches for a wide variety of macro and microorganisms. Fungi is a large group of eukaryotic organisms, heterotropics, associated with humans. So these interphytic fungi basically living within the plant organ without any apparent harm. These interphytic fungi are promising source of bioactive compound. Now, these mangrove-derived fungi are the resource of novel metabolites comprising novel structure classes with a diverse biological activities. But these mangrove forests drastically decline due to the urban development, aquaculture, mining, and overexploitation. And so there is an urgent need of studies aiming to mangrove-associated interphytic fungi. So in the study, we investigated the fungal communities associated with the various parts of mangrove trees, Avicennia marina, from protected woodland mangrove forest. Now, material methods. So this study was basically carried out in the Godrej mangrove forest, Pikroli, India. Now, the healthy leaves, twigs, and nematophores of Avicennia marina were sampled from the Godrej mangrove forest, Pikroli, India. In total, 40 samples were randomly collected from the different places. Now, for isolation, the collected sample were washed with the tap water to remove the dust particles, followed by washed with the sterile water, and then immerse the plant parts into 70% null for 1 or 2 minutes to sterilize microorganisms on the surface, followed by 2% sodium hydrochloride solution for 30 seconds, and then last again 70% null for 1 or 2 minutes to complete sterilizations. On each and every stage, washed with the sterile water, and after final wash, all these samples were cut into small pieces around 5 mm long, stay and lives in the square tissues, and then placed on different media including PDA, PCA, and EA, and then kept in 30 degrees Celsius for 7 to 8 days for incubation period. Now here, after 7 to 8 days, we found that there are two types of isolates. Sporulated isolates were identified based on morphological characteristics with the help of standard literature and monographs. So basically, coelomycetes by Stone, then numidaceous hypomycetes and related reference. And those who are non-sporulated culture were designated as mycelia sterilia, which were divided into different morphotypes according to the culture characteristics such as colony, color, texture, and Data analysis, so basically authentication of mangrove plant Avicennia marina was done from the plant science division, Alvaro Research Institute, Pune, India, and the relative abundance, relative frequency, saline diversity index, Soren's coefficient were calculated. These are the observations that the list of isolates from the various plant parts. So these are the leaves associated mangrove endophytic fungi. These are the twig associated endophytic fungi, and these are the nematophore associated fungi. Now, followed by the biotechnological uplink potential, so screening of isolates for their cell activity. So isolated fungus was grown on the basal salt media supplemented with 2% CNC, and the pure culture were inoculated in the center with almost equal amount and incubated at 30 degrees Celsius until the substantial growth was recorded. The petri plate were flooded with the congruent solutions, and after 5 minutes in the congruent solution was discarded, and the plate were washed with the one normal initial solution, allowed to stand for 15 to 20 minutes. The clear zone was observed around the colony when the enzyme had utilized the cellulose. Cellulitic fungi were screened on the basis of their ability to hydrolyze cellulose by forming a diameter zone of clearance around the fungal colony. So these are the results of screening of isolate for their cellulitic activities. And resultant discussions, so total 40 sample segments were analyzed, 32 segmented colonies by fungi. So overall colonizing frequency was near about 65.67. So total 21 fungal isolates recovered, 55.8% from the leaves and 21 from the leaves from the stem and 23 from the nematophore. Now these 12 fungal species isolated from the fresh leaves, 9 fungal species from the senescent leaves, and the 5 fungal species isolated from the mature leaves. So isolates were from 10 different genus, including Ascomycete, anamorphic Ascomycete, that is filamentous fungi, and some isolates were also from the Basidiomycete. Now this fungal community associated with the mangrove forest, mangrove plant, Avicennia marina, differs significantly between the sample locations. And saline diversity values are comparable among all the locations and fungal communities diversity in the leaves. These are the photograph of pure culture plate. These are the microscopic endophytic fungi. Now in this investigation, we have investigated the cellulitic fungi from the different from the different parts of mangrove associated region, and the cellulase producing strain isolated from the different plant parts. Sample were subjected to optimize of median cultivation parameter of cellulose productions. Now, conclusion, so there is a growing interest for the role microorganisms play in all aspects of ecosystem functioning, and success of mangrove restoration project is expected to benefit from the further detailed characteristics of mangrove microbiomes. This study provides scientific authenticated data on the fungal community associated with the various part of mangrove Avicennia marina, and more broadly contribute to a better understanding of the coastal microbiomes. Screening results indicate the potential of mangrove endophytic fungi to produce active intakes and bioactive compounds which are industrially important. The further work will be include more in depth studies regarding the compound present and their structure, elucidation, and biological assay. More concentrated and coordinated cross-disciplinary efforts are required from marines, terrestrial, and atmospheric microbiologists to fully address the technology gap in the research of this nature. These are the references, and I would like to acknowledge, I would like to thank my research guide, Dr. Lal Santiago, for outstanding assistance, grateful discussions, and critical regarding of the manuscript, and also thanks to Principal Dr. Manju Talwani Pathak and Dr. Sasibal Pandey, CHM College, Ultrasonic, for providing laboratory facility, and Dr. Sarita Hanis Madam for her kind support. Thank you.
A very good day, everyone. My name is Sumani Tinsin. I'm a research scholar from the Government Navy, Naval University, Lubani. The topic that I'll be presenting is on the multi-layered cell varieties of shifting cultivation area of Monsoon district now in India. As you all know, the cycle of shifting cultivation, we have a forest area where the vegetation will be slashed and burned, and the resulting cropland area is cultivated for a period of 1 or more years. And lastly, we have the very important fallow period, where basically the land is allowed to restore its vegetation and restore its depleted nutrients. However, if you look at the current situation in the state, we find that there is a massive area of forest under degradation. Nearly 3 million hectares of forest cover are felled every year just for June cultivation. Another problem is that this fallow period has drastically dropped from the optimum 10 to 20 years to just 4 to 6 years, or even lesser in some cases. Another problem is that nearly 100 different indigenous tribes are completely dependent on June or shifting cultivation for their livelihood. This problem is further made worse by the fact that there is a lack of alternative occupations for the inhabitants. So some of the negative impacts of shifting cultivation, as we all know, is that it leads to an increased soil erosion, it leads to watershed dissipation, it decreases the fertility of the soil, it affects the plant's performance. Ultimately, there is a change in the soil physical structure, there's lowering of the soil matter content, there's a lowering of the nutrients and microbes. Ultimately, what we get is a lowered crop yield, which has a very big impact on the agricultural community. So keeping this point in view, this present survey has been conducted in the region of Mokchung, Nagaland, where shifting cultivation is practiced on a very large scale. We look at both the soil data and the fungal data for this work. For the soil data, we incorporate principal component analysis to obtain the soil quality index. The obtained soil quality index is then compared with literature. For the fungal data, we incorporate importance value index and chemical correspondence analysis to know the effect of the soil variables on the population. Onto the result, we find that pH had a value of 5.65, which is quite high. Temperature had a value of 27 degrees Celsius. Soil organic carbon had a value of 1.90%, which is found to be quite low. Nitrogen had a mean value of 325 kg per hectare. Potassium, meanwhile, had a value of 122 kg per hectare, and phosphorus had a value of 23 kg per hectare. And lastly, clay content has a value of 24%, which is also found to be quite low. So for obtaining the soil quality index, what we first do is we have to conduct the principal component analysis on the total data set. The resulting weights of the factor loadings are then incorporated into the soil quality index to obtain the final value. So this is the result of the principal component analysis of the soils from the shifting cultivation side. We find that there is two principal components in the present study. The first principal component explains 72% of the variance, and the second principal component explains 20% of the variance, respectively. As given in the PCA pie plot, we find that under the first principal component analysis, we have clay, pH, nitrogen, moisture, and soil organic carbon. Meanwhile, the second principal component, we have potassium and phosphorus, respectively. So the obtained soil quality index from the current study had a value of 0.74. It is very important to remember that the soil quality index ranges from 0 to 1, with lower soil quality having a lower value, meanwhile a higher soil quality or productive soil having a higher value. So when we compare our obtained soil quality index with the reference soil quality index from different soil type, we find that the soil of shifting cultivation site was found to have a lower soil quality index as compared to the other land use sites. This leads us to the conclusion that soil under the shifting cultivation site was found to be degraded. Onto the fungal data, we find that winter, which generally has lower temperature and lower moisture, had a lower fungal diversity in the present study. With the onset of spring, summer, and autumn, temperature, moisture, and even soil organic carbon increases. So the resulting increase in fungal population is observed in the current study. With regard to the importance value index, we find that the species Aspergillus species 2 had the highest IVI value. This basically means that this species, Aspergillus species 2, has adapted to the practices associated with shifting cultivation. As you all know, in shifting cultivation, there is a practice of burning of soil, removal of vegetation, and the use of chemicals, and also the use of salt. So this species in particular has been adapted to this environment. As a result, this has a higher IVM value. Meanwhile, the other species which are not that adapted to this site have a lower IVM value. With regards to the CCA plot, we find that there are three factors at play. The first is the temperature. As previously mentioned, shifting cultivation employs the burning of soil, the removal of the vegetation, which leads to an increased exposure of the soil surface to this solar radiation. As a result, this increased temperature may be one factor that controls or limits the fungal population. Another factor is the decreased levels of soil organic carbon. Shifting cultivation practices has been reported to decrease the soil organic carbon value in soil. So this decreased soil organic carbon value might be one factor that also limits the fungal population. And lastly, the increased levels of pH value. The practice of burning the soil raises the ash content in the soil, resulting in the increase in the pH level. Also, the spraying of salt by the farmers has been known to increase the pH level. So this increased pH level may also limit the fungal population in the study. So we conclude by stating that three factors: temperature, soil organic carbon, and pH control or limit the fungal population in the present study site. We overall conclude this study by stating that the land under shifting cultivation was found to be degraded, and the decreased fungal diversity is mainly controlled by three factors, that is the soil carbon, temperature, pH. One possible way that we can control this deterioration of the soil quality is by maintaining the fallow period. However, we also stress on the need for community awareness, especially on the land of the fallow period. We also stress on the need of alternative means of farming, such as apiculture, to reduce the pressure on the agricultural land. And lastly, the need for systems such as nutrient manuring, which has been known to increase organic matter in the soil. We'd like to end this presentation.
Good morning, everyone. Respected professors, fellow scholars, and participants, my name is Giacomo, and I will be presenting on morphological identification of resistant fungi isolated from Coffea arabica and its efficacy to solubilize phosphate. Introduction. Coffea arabica, a genus of flowering plants belonging to the family Rubiaceae, is an evergreen perennial shrub. Coffee is the second most consumed beverage after water and the most traded tropical agricultural commodity. About 3/4 of coffee cultivated worldwide are arabica and is considered as king of the coffee. India produces 4.74% of the world's coffee, and the rich northeastern region contributes 0.1% to India's coffee production. Between 2019 to 2020, India has exported more than 2 lakh tons of coffee. It is a source of livelihood for millions of small and marginal farmers and provides employment for millions of plantation workers. Nagaland coffee has unique taste, and it is among the best coffee produced in the world. Coffee grown in Nagaland yields a well-balanced cup, which is redolent with peppery, spicy aroma, contains nuts of carrot, and has the delicious aftertaste of vanilla ice cream. Resistoric microbiota. Resisphere is in the zone of the surrounding plant roots in which microbial populations are stimulated by root activities. It is a unique subterranean habitat for microorganisms. Microbes perform beneficial effects on the growth and development of plants, apart from inducing tolerance to biotic and abiotic stresses are well known. The use of plant-associated microorganisms in biofertilizers, particularly fungi collected from local forests to increase crop yields, is gaining popularity owing to its multiple their positive relationship with ecological environment. The taxonomic and spatial distribution of fungi in the coffee soil of Nagaland is unknown. Therefore, the present study is aimed at isolating indigenous fungal inhabitants of coffee rhizosphere and in vitro screening, that is, phosphate solubilization of the isolates with potential growth promotion. Methodology: collection of rhizospheric soil sample. Rhizospheric soil samples were collected from coffee farm at Kirima, India. The soil adherent to the roots of the plant was carefully collected. Isolation and identification. Serial dilution plate method was followed for the isolation of fungi. Fungal isolates were identified by examining the morphological characteristics of fungal colonies and also through microscopic studies. Qualitative analysis. The isolated rhizospheric fungi were tested for their ability to solubilize organic phosphate in Phytochemical medium. Isolation and identification. A total of 19 rhizospheric fungi belonging to the genera of Acromonium, Aspergillus, Penicillium, Bacillomyces, Coledotrichum, and Scapulorhexis were isolated and identified based on colony morphology and microscopic features. Screening of isolates for phosphate solubilization. The isolated rhizospheric fungi were tested for their ability to solubilize organic phosphates in solid PVK medium. All the isolates showed clear halo zone when cultured in solid PVK. Solubilizing index range from 1.16 to 4.05. These are plates showing mixed culture. These are lists of fungal species isolated from rhizosphere soil. Aspergillus accounted for the most species with 6 species, while Scapulorhexis documented with the list with 1. These are plates showing pure culture and microscopic features of Aspergillus versicolor, Aspergillus fumigatus, Bacillomyces liliaceus, and Penicillium. These are plates showing zones of phosphate solubilization on Phytochemical agar plates. Discussion and conclusion. Rhizosphere is a narrow zone of surrounding plant roots in which microbial populations are stimulated by root activities. A total of 19 rhizosphere fungi belonging to the genera of Acromonium, 2 species, Aspergillus, 6 species, Penicillium, 4 species, Bacillomyces, 4 species, Coledotrichum, 2 species, and Scapulorhexis, 1 species were isolated and identified. Microbes performed beneficial effects on the growth and development of plants, apart from inducing tolerance to biotic and abiotic stresses are well documented. Accordingly, all the isolated resident fungi were tested for their ability to solubilize organic phosphate in solid PVK medium. All the isolates showed clear halozone. Solubilizing index ranged from 1.16 to 4.05. This study evidenced the presence of phosphate solubilizing fungi, which may serve as potential biofertilizers. Screening and application of these phosphate solubilizing fungi can be a better option to mineralize the indigenous phosphate reserves of soil, as well as organic amendments for sustainable agriculture. These are my references. Thank you.
So we have done with the last technical session in biodiversity and environmental science. On behalf of Botany Department, GM Khalsa College, I extend my deep gratitude to Dr. S.R. Joshi and Dr. Kusum Arunachalam for chairing the session in biodiversity and environmental science. It's really a big task to evaluate 28 presentation videos. Thank you so much for sparing your valuable time to evaluate all the paper presentations. Sir, now I request Joshi sir to give your valuable feedback to the participants.
Yeah, very good evening and namaskar, Madam Tandon. It's so nice to see you before. And Sir Tandon, sir, more than attending, this is a pleasure to see you around. Puja ho raha hai hamare ghar pe. It's so nice you came. My sincere namaskar to both of you, sir, madam. We literally sat for the entire day, sir. They're beautiful presentations that you all made. I think some of the youngsters who have gone into the research is beautifully done. I have a couple of advices. Don't take me otherwise. A few things, when you're writing a paper or you're making your presentation, the themes are very, very good. But when you're making a presentation or, taking the research forward, only advice to youngsters who have made the presentation is try finding out the gap area. What is not done in that particular aspect that you're looking for. That will really add to the quality of presentation that you're making and the data that you have collected. This is one I noticed, because let's not repeat what already has been done, because there are a lot of information that is available. Your data are quite novel, no doubt, but then somehow what is the need for doing this particular work and the gap? If you can just add on to that, it will really add to the quality of the presentations. But overall, found it very, very interesting, because I learned personally quite a lot of it. And the second thing is, when you're making presentation, in some of the cases, I am maybe wrong, but then I noticed that you got to be putting your guide together when you're making a presentation. If you're not the sole author, let's make this a habit of your title page must carry the name of your guide. That's very, very important, and it's also ethical to put the name of the guide. That's important. And the third is, when you're talking about methods, materials and methods, you don't have to give detail of what is the mole, what is the concentration of each solution, and you can just mention which method you followed. That will serve the purpose, and you can really go into what is your finding and discuss more on that finding. These are few things that I have, but we had quite a series of presentation. If I go one by one, it's going to take another at least 1 hour, and I really don't want to. I can see Professor Kusum sitting there with few suggestions. I would look forward to her suggestions. Thank you very much.
Thank you, Professor Joshi, for your inputs. And yeah, there was a very informative session. And as the name suggests, the session was on biodiversity and environment. So it was most of the presentation which we heard were covering the entire landscape of India. So we had presentation right from the Ladakh to the southern state of Tamil Nadu, and then we had from northeast and even in the western part of Rajasthan and Maharashtra. So most of the states. So this was really interesting that the studies, the presentation which were made were covering entire landscape of India, and there was really good. All the area, all the aspects of biodiversity was covered. And it was very difficult to even evaluate them, because I feel that most of you have done wonderful work. But as Dr. Joshi has suggested that, yeah, you have to really find out the gap area, because there's already a lot of knowledge is existing now. How to use that knowledge in solving the problem of what the environmental changes are we all are facing today. So as the name of the session suggests, that biodiversity environment. So the problem of in 21st century which we are facing of environmental degradation, so biodiversity can only provide those solution to us. So as we all are also hearing a lot about the nature-based solutions, so nature-based solution will all come from the biodiversity. So all the presentation were covering the different facets of the biodiversity, from microbial diversity to algal biodiversity to the higher plant biodiversity. So it was really interesting and very informative session, and I have really learned a lot. And I'm really glad to see our teachers, both Professor Pramod Tandon and Veena Tandon Madam in this session. So I'm really looking forward to hearing to Veena Madam. Thank you so much.
Thank you, Doshi sir and Kusum madam, for your encouraging feedback.
Namaste, madam.
Namaskar.
Yes, it will really help in motivating the participants, and your valuable remarks will help our presenter to improve certain aspects in the research area. So thank you so much, sir, madam, for chairing this long session. A big thank to the technical team, Ms. Kaveri, Ms. Rutuja, Ms. Pratibha, and Ms. Wrinkle for compiling all the videos and presenting them so nicely. We are grateful to all the presenters for their active involvement at a large number and for timely submission of the presentation video. Thank you, sir. Thank you, madam, again. Thank you, all the participants. Now, over to Pallavi to begin with the valedictory function.
Pallavi.
Pallavi, are you there? Ma'am, I think she's not connected. Viva, you can start.
Yeah, Babita, thank you so much. So I welcome all the participants and I welcome Veena ma'am, who is the chief guest for today's session. Before we begin with Veena ma'am's talk, I invite Kopa ma'am to address us about the entire conference which was being held on 8th and 9th April. Kopa ma'am.
Good evening, all of you. Ladies and gentlemen, that brings us to the end of our conference. And what a conference it has been. I hope that all of you have enjoyed the incredible presentations and the insightful talks from the speakers and all our resource persons, especially Professor Pramod Tandon. Our conference had 4 very important themes. In biotechnology theme, there were 14 presentations and there were 4 plenary speakers. Dr. Malavika Sen, she talked about the lung cancer, which is most common cancer, and what is the reason behind it. And she also discussed about the molecular targeted therapies. Dr. Indira Laisram spoke about the precision medicine. Dr. Aparajita Datta spoke about the drug discovery and development and drug safety. Dr. Rajini gave an overview about the clinical research on thyroid cancer. In biotechnology session, Dr. Anshuman Chattopadhyay was the chairperson and he gave feedback about all the presentations and added his views. In the second session of 8, that is the first day, we had Mrs. Hemali Mathak as a plenary speaker for the nutrition and dietetics theme. She was speaking. She had spoken about the how fortunate we are at having the Indian spices like haldi and black pepper, and how it helps against the Alzheimer's disease. Dr. Meena Mehta was chairing that session, who is from SNDT University, Mumbai. She gave a briefing about the presentations and suggested where are the researchers can improve. Now, in today's session, we started with Dr. Sanjeev Ray, who had given an overview about the, though he is from zoology department, we were so happy to see that he is working with the plants. So he had worked with some allium species. And for that session, Dr. Arnav Sen and again Dr. Sanjeev Ray, who was the chairperson, there were lots of 37 presentations, I think, and they had commented about some few papers, and they were really happy to see lots of MSc students also were involved in presenting their papers, and they were happy that they are really engaged in research. In the next session, we had Dr. Kusumarunachalam and Dr. Kusumarunachalam was the speaker, as well as she was chairing the session. Even Dr. Asad Joshi was chairing the session. Dr. Kusumarunachalam spoke about the ecosystem restoration for biodiversity conservation. Here, the Dr. Debashi Datta was also from here with us from New Zealand. He spoke about how to monitor the impact of browsers on saplings. So we have really enjoyed the main thing. What I most particularly enjoyed was the young presenters, and there were many of them. The presentations were excellent, so well done to all of you. An immense thank you to our chairpersons, Dr. Anshuman Chattopadhyay, Dr. Amina Mehta, Dr. Kusum Arunachalam, Dr. Asad Joshi, Dr. Arnav Sen, Dr. Sanjeev Ray, and especially our participants who knows by now about the how the conference went. Now you all know how the team has been working tirelessly day and night behind the scene. Thank you all who have been presented, participated in the conference, and who have been actually present here as audience member. We have valued your support immensely throughout the organization period and also during the conference. Some of you, for some of you who does not know, we are trying to publish. We have tried to publish the papers which you have sent to us to a UGC recognized journal. All of you might have received by now whoever paper has been accepted. So all the best to all of you whose paper are still the news has to come. So keep well, and we hope to talk to you very soon again. Thank you all of you.
Thank you, Gopa ma'am. Now, may I now invite Dr. Manjari Bhave to introduce the guest of honor today's session.
Hello.
Dr. Manjari.
Yeah. Am I audible?
Yes.
Yeah. Thank you, ma'am. Good evening to one and all. With immense pleasure and with deep sense of gratitude, I take this opportunity to introduce our today's guest of honor, Padma Shri Professor Veena Tandon, who is a widely recognized teacher and a woman scientist of international repute, who has rendered exceptional and distinguished service to the people of Northeast India for more than 35 years. She received her doctoral degree from Punjab University, Chandigarh, and continued her postdoctoral research at University of California. Professor Tandon has made influential contributions to the field of parasitology. Her lifetime contributions have provided a complete spectrum of worm infections prevalent in animal of food value in the northeastern region with the creation of web-based database. She has extensively investigated the efficacy of many medicinal plants which are used in cure against worm infections in the tribal traditional medicinal system. Her research has scientifically authenticated their vermicidal potential and shown that such plant-based cures can offer valuable therapeutic alternatives of affordable healthcare to socio-economically underprivileged and rural societies. I must say research can't be done in watertight compartments of social, economic, scientific and commercial subjects, and what better proof can we have than this? Madam is actively engaged in creating multidisciplinary teams to address national and global research challenges on neglected tropical diseases of worm origin. Professor Tandon is a fellow of National Academy of Science India, Indian Society of Parasitology, Zoological Society of India, and served as a member of many academic bodies and national task forces, including the Scientific Advisory Committee to the Cabinet under the leadership of late Dr. APJ Abdul Kalam. She has received many prestigious awards and recognition, notably Padma Shri Award in 2016, P.K. Janaki Ammal Award in Animal Taxonomy, Lifetime Achievement Award of Indian Society of Parasitology, World Federation of Parasitologists Distinguished Parasitologist Award in 2018 at Korea, Lucknow Management Association's Outstanding Women Achievers Award in 2009 in the category of academics, and recently Outstanding Scientist Award conferred by Chhatrapati Shahuji Maharaj University, Kanpur in 2019. Professor Tandon has published more than 180 research papers and articles in the journals of national and international repute. She has contributed several chapters in parasitology reference books and written 2 books and has edited 4 books as well. During her 40 years of long research career, she has trained a large number of MSc and over 30 PhD students in the subject and has established a strong school of parasitology research in India. I really feel fortunate to get a chance to make her biography known to this scientific gathering today. So here we have a humble, multitasking and research-oriented eminent personality among us as a guest of honor for our valedictory function. We are eager to listen to your words of wisdom, madam. Over to you, Dr. Veena Tandon.
Thank you. Am I audible?
Yes, ma'am.
Thank you very much for your kind words. Good evening, everyone present here. Convener of the conference, Professor Gopa Sharma, along with her organizing team and nominee of the management, Govinderji, chairpersons of the various technical sessions, including Professor Anshuman Chattopadhyay, Dr. Meena Mehta, Professor Arnav Sen, Professor S.R. Joshi, Dr. Kusum Arnachalam, distinguished speakers including Dr. Malavika Sen, Dr. Indra Laisram, Dr. Aparajita Datta, Dr. Rajani Jacob, Mrs. Himali Mathak, Professor Sanjeev Ray, Dr. Kusum Arnachalam, Dr. Devashis Datta, faculty and research students of the college, participants, ladies and gentlemen. I'm truly delighted and so happy to be the part of this important international conference on novel approaches in life sciences organized by Department of Botany, G.N. Khalsa College, Mumbai. Thank you, Professor Gopa Sharma and your team of organizers for associating me with this event and giving me this honor. I'm so happy to see many past scholars from NEHU who are now occupying important positions in academic arena. I have very fond memories of my association with them. Thank you once again, Gopa, for giving me this opportunity of meeting with you all, even though virtually, a rare opportunity. I understand that during this two-day event, you had deliberated on various aspects related to biotechnology, nutrition and dietetics, medicinal botany, besides the just concluded session on biodiversity and environmental botany. In addition, a webinar by NCROM was also organized. I'm sure that you had deliberations with participation of young researchers. The deliberations were truly vibrant and scientifically very challenging. It's really laudable that you made all efforts to organize this conference, even though the COVID pandemic was just hovering around us all during the last 2 years or so, affecting not only the social life but greatly affecting the academic activities of the young generation. Biodiversity, as you know, is essential for the processes that support all life on Earth, including humans. Without a wide range of life forms, microorganisms, plants, and animals, we cannot have the healthy ecosystem that we rely on to provide us with the air that we breathe and the food that we eat. With the burgeoning increase in human population, from the present 6 billion to the estimated 9 billion by the year 2050, one would expect that 1 billion hectares more of wildlands would be converted for food production. There would be massive decline in biodiversity and vital services. About 60% of Earth's ecosystems are in trouble in present. What state will they be in by 2050 is anybody's guess. Climate change is an imminent reality, and we need to work as expeditiously towards mitigation of climate change. Biotechnology harnesses cellular and biomolecular processes to develop technologies and products that help improve our lives and the health of our planet. We have used the biological processes of microorganisms for more than 6,000 years to make useful food products such as bread and cheese, the very common table items in present times, and to preserve dairy products. Modern biotechnology provides breakthrough products and technologies to feed the hungry, combat debilitating and rare diseases, reduce our environmental footprint, use less and cleaner energy, and have safer, cleaner, and more efficient industrial manufacturing processes. I'm sure that you would have discussed some of these cutting edge researches in the area of biotechnology that would lead to a better quality of life to human beings across the globe. I know that with the limitations of facilities at the college level, you may not be doing high-end research, but it is well appreciable that such efforts like the one you have put in for organizing this conference has given a very good platform to the youngsters to familiarize themselves with the present day problems facing our society and lives. Another area chosen by you is related to medicinal botany. Medicinal plants, as you all know, are considered a rich resource of ingredients which can be used in drug development, either pharmacopoeial, non-pharmacopoeial, or synthetic drugs. Apart from that, these plants play a critical role in the development of human cultures around the whole world. Medicinal plants are useful for maternal and child health care, as essential drugs in food and nutrition, for common illnesses and injury, for endemic infectious diseases, and oral and mental health. Worldwide, between 50,000 and 80,000 flowering plants are used medicinally. Of these, at least 15,000 may face extinction due to overharvesting and habitat loss. There is an urgent need to conserve the medicinally important plants on a sustainable basis, ensuring that they are not endangered in their natural habitat. It is also a good practice to introduce some commonly used medicinal plants to children and encourage them to grow them in kitchen gardens. Some minor ailments could be easily handled by home remedies prepared by such plants. We in India have tremendous traditional knowledge of medicinal plants. There is a need to authenticate the active principles in these medicinal plants and carry out researches towards development. Though you may be familiar that uh, in NEHU, a lot of work is being done by researchers at the university uh, relating to the ethnomedicinal properties of plants that are widely used in traditional medicine system. I may just cite one example of Flemigia vestita, a member of the soybean family Fabaceae, and locally known as saflang in Meghalaya. This plant is widely used in traditional alternative medicine system in Northeast India. Its active principles, isoflavones in general, and genistein in particular, have well-established therapeutic roles against breast cancer, and with our group, research group, we could identify the the therapeutic role of genistein uh, against gastrointestinal worm infections. And uh, in the northeastern societies, the plant is widely used for the purpose. World agriculture, we know, produces 17% more calories per person today than it did 30 years ago, despite a 70% population increase. There is enough food to provide for everyone in the world with at least 2,750 kilocalories per person per day. Principal problem is that many people in the world do not have sufficient land to grow or income to purchase enough food. There is an ever increasing number of hungry people in parts of the world. There is a neglect of agriculture relevant to very poor people by governments and international agencies. There are more than 1 million cases of malnutrition per year in India alone. Despite our country's 50% increase in GDP since 2013, more than 1/3 of the world's malnourished children live in India. Amongst these, half of the children under 3 years old age are underweight. The crisis of child malnutrition has often been attributed to historical antecedents such as poverty, inequality, and food shortage. There is a need to improve the health of women, childbearing women, and children to decrease infant mortality in the country. You had chosen to discuss about nutrition and dietetics, a branch of specialization of medical science that deals with the study of various foods, along with their nutritional value and customizing it as per patients' or personal health, fitness and stress level, depending upon age, sickness or work routine, occupational constraints, etc. Dietetics and nutrition course of study is also a part of health and wellness branch. Nutrition and dietetics is one subject which will help in exploring how to design controlled diets. Having said that, I have no comments to make on the so particular and preferred diets of burgers and junk foods among the present generation. I'm sure that these deliberations held at the conference will have definitely aroused awareness among the young generation, and the deliberations will help you prepare concrete recommendations for your college. Maybe simple recommendations which could be workable for planners in the government and at public at large. I would just like to congratulate the organizers once again, having known that the sections have run for a long times beyond the designated times, and you all must be tired by now, but much enriched mentally by the overall proceedings of the conference. My best wishes to all for your future endeavors. God bless you all. Thank you.
Thank you so much, Reena madam. Madam, I feel the words futile to thank you enough. It was really a treat to ears and food for brain to listen to your words of true wisdom. Your speech was filled with gratitude, summarizing the 2 days conference in such a crisp manner. I'm sure all the dignitaries and participants must have received the warmth of your persona even when we are on online mode. You touched upon the areas of biodiversity, environment, and took the participants on the journey of prospective research areas in nutrition, clean energy, and industrial practices. Also, to the medicinal plants as a resource to make the drugs to elevate the health of mankind and their conservation. You brought light on malnourishment of children and stress related to the areas of research in nutrition. I really thank you, ma'am. On behalf of organizing committee of this conference, GN Khalsa College, I express my I feel profusely grateful to you, madam. Over to you, Priyank, for displaying the winners of the presentations. Tandon sir and Tandon madam, now we are going to announce the winners of the presentations in the presentations which were made in this conference. So we have in all 6 winners from different themes. So first one is biotech. In biotechnology category, we have Dr. Gursimran Kaur Uppal. Her topic was in silico studies for the inhibition of proprotein convertase furin using synthetic compound databases to see the effectiveness against SARS-CoV-2 transmission. So congratulations, Simran.
Thank you so much, Dr. Manjri. Thank you so much.
Next, please. In nutrition and dietetics, we have Ms. Akshaya Tare, who talked about nutritious and cost-effective plant-based food colorants and its incorporation in Tutti Frutti. So congrats, Ms. Akshaya. Next, please. In medicinal botany, since we have lot many presentations, there are two prizes. So first one is Ms. Veena Desai, uh, who talked about red blood cell membrane stabilizing potential of Roystonea regia bark extract. And the second prize goes to Shubham Ghadge, who talked about molecular docking studies of selected phytochemicals as potent inhibitors for Parkinson's disease approach to anti-Parkinson's drug. So congratulations, Veena Desai madam and Shubham Ghadge sir. Next, please. We have two more prizes to announce in the theme of biodiversity. Dr. Payal Achrekar, who talked about stomatal index, leaf thickness, and noise absorption studies of some plants, and Dr. Ajita Kumar, who talked about improvement of physicochemical characteristics of soil using biodegradable kitchen waste, Trigonella foenum. So
Thank you very much. Thank you very much for giving me this opportunity, Khalsa College. And I'm really surprised I got the first
thank
prize.
you. So thank you, Payal, and thank you, Dr. Ajita. Over to you, Vibha madam, for final formal vote of thanks.
Our congratulations to all the winners.
Congratulations, all of you. Thank you so much, madam.
Thank you.
Many congratulations to all the deserving candidates. So we are now proceeding with the formal vote of thanks. So very good evening to one and all present here. I feel honored and privileged to get the opportunity to propose a formal vote of thanks at this two-day international conference on novel approaches in life sciences organized by the Department of Botany, Guru Nanak Khalsa College, Matunga, Mumbai. I would like to thank all the honorable delegates who blessed us with their presence. On the behalf of organizing team, kindly accept our deep sense of gratitude to honorable chief guests, Padma Shri Professor Pramod Tandon and Padma Shri Professor Veena Tandon, and to all the chairpersons and the invited speakers who readily accepted our invitation and have enlightened us and provided deep insight knowledge in different theme areas of this conference. Words are not enough to thank for the support and guidance to shape this conference. On behalf of the organizing committee, I would like to convey deep regards and hearty thanks to the management of our institution, Guru Nanak Khalsa College, for all the support extended to conduct this conference. I express my sincere gratitude to our principal, Dr. Kiran Mangaonkar, for giving us an opportunity to organize the conference and for constant encouragement. We are obliged to our convener and the HOD of Botany Department, Dr. Gopa Sharma, for inviting well-distinguished delegates from different fields at international and national level, took initiative in organizing this international conference, and under her guidance, for the first time, our Botany Department hosted it successfully. Her constant help and guidance had radiated a source of energy within us. Thank you so much, madam. Organizing committee highly appreciate IT support team, Mr. Aldrin Jolie, Mr. Shyam Mohan, and Mr. Ashish Jaiswal, who backed us with all technical assistance in facilitating registration, streaming of the conference, and handled all other technical intricacies with deep sense of commitment. I'm thankful to the National Sponsoring Agency, RUSA, and to Ancrom Private Limited. It would have been impossible to organize it without their support. A very big thank you to all the participants for being a wonderful audience. I hope this conference has broadened the horizon of research and encouraged and nurtured young minds to use novel approaches in different areas of life science. Heartfelt thanks to the botany faculty who put their efforts in making this conference a grand success. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to be backed by a team of very motivated and dedicated volunteers of postgraduate students and a PhD student of our botany department. I thank each one of them for their involvement and willingness they have expressed to take on the completion of tasks beyond their comfort zone. A big thank you to the entire team of volunteers. I would like to thank the entire fraternity of Khalsa College and also non-teaching staff members who stood by us in conducting this online program. Thanks to all for making this event successful. Now, before we close the event, I request all the participants to kindly fill in the feedback form. The link will be shared in the Google Meet as well as in the Telegram group instated for this conference. Link will be active today till 8 p.m. Please fill in details carefully and correctly as information will be used for making e-certificates. No request for corrections will be entertained once certificates are issued. I assure that the registered participants will receive e-certificates, but the process may take 15 days. Here, I conclude and request all to please stand for the national anthem. Kindly keep your camera on and mic on mute for the national anthem. Over to you, Priyank, please. Thank you. Priyank, are you able to play?
We can just sing it.
Yes, ma'am. We can sing then. Yeah.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तब शुभ नामे जागे तब आशीष मांगी गाहे तब जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय 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 हे.
Thank you so much.
We can all keep our videos on. We'll take a screenshot.
Yes,
all
ma'am.
of you, please switch on your videos. Whoever is present here, please switch on your video so that we can take a screenshot. Yeah, I have taken. Thank you so much, all of you. Thank you. Thank you so much. Thank you, sir. Thank you, ma'am.
Thank you. God
Dr.
bless you.
God bless
Madam,
you all.
Madam
Thank
Chaitanya, we missed you so much in Nehu.
Yes,
we really missed you. We had a wonderful time with
Yes.
sir.
It's so nice to see a ma'am after such a long time.
know.
I
And
got
I
so
could
Just
emotional.
recall all the classroom memories flashback.
Yes, yes,
It
ma'am.
is really
I know.
nostalgic.
I
Yes,
know.
ma'am.
Thank you,
That's
It's
why
so
Gopal.
I
nice.
called all
So
Nehu
nice.
people so that they
Yeah.
can
Yeah.
meet you all.
How are you,
Yeah.
madam? Yeah. How are
Thank
you,
you,
madam?
sir. Thank you.
Madam Tandon, how are you?
Yeah, I'm fine. How are you, Hiranjit?
Fine, thank you.
Long time no
After
see.
yes, yes, madam. I just met sir a few days back.
Yeah, yeah,
Around
I know.
I
I
was
know.
asking about you.
Gopa ma'am,
You
I'm
always take
stopping the
the lead.
recording.
Yes, Viva, you can stop recording. Now
Yes,
Kusum,
don't,
how are you
don't
and where are
record
you?
our class.
Kusum?
I'm fine, madam. I'm fine. I'm in Dehradun only.
Okay.
I'm in touch with everyone, ma'am.
But she is the person who connects with everyone.
Yes, I'm in
Yes.
touch with everyone.
So we
Yeah.
appreciate it to her.
Wonderful job done.
Yes.
Gopal,
Thank you,
wonderful job
organizer.
done.
Thank you, ma'am.
Thank you. Thank you.
Thank
Okay.
you.
All the best to all of you. God bless you.
Thank you,
you,
sir.
Sir,
sir.
I
namaskar.
think it's
Standing,
Madam,
good.
namaskar. We'll meet
standing,
soon. And thank you,
standing
Dr. Gopa.
invitation to
Thank you,
in
Hiranji. Thank you.
everyone to
Thank
come
you.
to
Thank you,
Lucknow
ma'am.
and see us.
Thank you so much,
Thank you,
Sure,
ma'am.
madam.
sure.
Thank you so
Thank
much.
you.
Please take
Okay,
care and
sure.
stay
Take
healthy.
care, ma'am.
Bye
Take care,
bye.
sir.
Bye.
Thank you.
You
Dr.
all.
Joshi, Hiranjit, Kusumdi, take care.
Thank
All of
you.
you, please
Okay,
take
thank you.
care.
Now
Thank
you
you.
take rest. You must be
Thank you.
too tired
Yes.
now. Okay,
Yeah, so
bye-bye.
tired. Yeah. Chalo.
Chalo.
Take
Nice
care,
to see
Kusumdi.
you.
Bye, bye, bye.
Okay, bye-bye. Bye.
Bye, Anshumanda. Bye.
Bye.
তুমি মিস করলে?
Gopa,
হ্যাঁ, বলো তুমি মিস করে নিলে।
I really appreciate the
Yeah.
way you have connected to everyone in India and abroad.
Yes.
And the number of participants and resource person speaks
And
about
the papers we have received, lots of
and
papers we have received.
and that speaks volume about your organizing.
Thank you so much. Thank you so much.
All the best, and it was a grand success. I have sent my
Thank
picture
you
for
so
it.
Yeah, thank you. Thank you all. Because of all of your support, the presenters and my colleagues, especially my colleagues, my students, they have really worked
It
like
was really
anything. I did not have to say anything to them. They just did.
there
I
was
did not know how it was happening. They were just doing it.
exactly the amount of
Yes,
work
because
needed.
I cannot do it alone. I just said we'll start, and they were after behind me, and they did everything. All the students, all my colleagues, day and night, without me knowing, they were doing.
That was really a grand one. All the best.
And yeah, especially that UGC Kerali's journal we have got, that also has added to this. Yeah.
Okay.
Chalo fir. Bye bye Payal, Shweta.
Thank you so much, Gopa ma'am. Thank you. In the end, it was all worth it.
Congratulations, Payal madam.
Thank you, Vibha ma'am. Thank you.
Call
I
me,
was really
Payal.
surprised to see my name.
No, you should not be surprised.
After you finish.
नहीं I mean suddenly मेरा नाम देखा 2 मिनट के लिए चकित हो गई. मेरा ही नाम है ना.
Good na.
Payal, you call me. I said you call me after this.
Chalo,
sure,
bye
I'll call
Gopa
you.
ma'am. Bye
Okay.
everyone.
Bye,
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 bye.
bye, Vibha.
Okay. Thank you. Yeah. Bye. अभी करूँ मैम?
Yeah.